pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes.
morning and welcome to the second day of CBER track of the Annual Regulatory Education for Industry 2024. We conduct this conference in collaboration with the FDA's Small Business and Industry Assistance Program as an annual educational event on the regulatory topics in medical product development. Now, today you will hear more presentations from 4 different offices from the Center for Biologics Evaluation and Research. It was our intention to make the agenda interesting and diverse in order to share the CBER's experience and provide as much information as we can, and also include references to the available regulatory resources from our website. But before we start the presentations today, let me first again show our CBER conference team, whom I introduced yesterday. Adrian and Ohai will continue managing the interactions with our online audience, and Melek and I will be moderating the sessions today, introducing speakers and running Q&A panels. Let me also take care of the administrative and continuing education information and disclosures. As was already mentioned yesterday, this conference offers over 14 interprofessional continuing education credits. The obtained certificate of attendance may be used in support of credits for the Regulatory Affairs Professional Society, REPS, the Society of Clinical Research Associates, SOCRA, Association of Clinical Research Professionals, ACRP, and the Society of Quality Assurance, SQA, as well as for continuing education credits for physicians, pharmacists, and nurses. In order to claim your CE credits, all participants must attest to their attendance and complete all the required activity evaluations through the FDA Continuing Education Portal, ceportal.fda.gov. And that's supposed to be done within 14 days after the activity ends. Unfortunately, we can't give any exceptions. And the last day to claim your credit for this year's event is June 13th. It's here in red on the slide. We greatly appreciate your feedback, your comments and suggestions that you put in your final evaluations. As you complete them, we take those recommendations seriously when we plan the agenda for our subsequent events. Attention to pharmacists and pharmacy technicians. The FDA Continuing Education team will report your credit to the National Association of Boards of Pharmacy, NABP, but that's provided that you add your NABP ID and the date of birth to your profile on the FDA CE portal in the required format to ensure that you receive credit for this activity. Now, NABP profile number should be 6 or 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month and day format. Pharmacists are recommended to log in 6 weeks after the last session of the activity to ensure that your credit is captured correctly. Now, I also would like to remind everyone again that the CE credits are available only to all real-time attendees who self-identified as healthcare professionals during the registration process and who attend this conference event either in person or through our conference platform. Continuing education credits will not be available to those who view the conference on YouTube after the event. The CE claim codes will be emailed to all attendees, and the email will have the information on the codes and will also contain the surveys and the evaluation links. And now to the disclosure. So I have to say all of this, bear with me, please. Speakers attest that the content they contribute is supported by the best available knowledge and evidence, and gives a fair and balanced view of diagnostic and therapeutic options. Continuing education faculty speakers are reminded to disclose when products or procedures being discussed are off-label or unlabeled. In other words, they're not FDA approved, and if there are any limitations on the information that's being presented. Now, I'm proud to say that all CBER speakers who are presenting today have nothing to disclose. And here, again, to the reminders of the conference logistics, you already know where to look for the relevant conference information at the sbievents.com. All conference presentations and Q&A sessions will be recorded, and recordings will be posted about 10 business days after the event. And then you can submit your online questions using on-screen link or the QR code. And for our in-person audience, we have a mic in the room here for the questions when the time comes. And I again ask everyone to please silence your phone, use internet sparingly so not to interfere with the broadcast. Also, all of our speakers will be staying for the duration of the breaks after their respective sessions, in case people who are present here would like to ask them questions in addition to what was asked during the Q&A. And so with that, we will start our morning sessions, and our first session will be about topics from the Office of Vaccines Research and Review. This is something that's been repetitively asked in our evaluation form, so we were glad to include topics related to vaccines in our agenda this year. And the session will be moderated by Dr. Melek Sunay. And without further ado, Melek, I'd like to turn the microphone to you. Thank you.
Thank you so much, Larissa. Welcome, everyone. Welcome, everyone online. This morning, we're honored to have the representatives from the Office of Vaccine Research and Review to give us our morning session. And so I will begin with the introduction of the first speaker, Dr. Hector Izurieta. He's the Associate Director for Novel Clinical Investigations in OVRR. He has extensive U.S. and international vaccine research and public health experience. He has previously worked at CDC, WHO, WHO, and Medicinas Sin Frontieres and other international organizations. He has authored around 100 manuscripts on real-world evidence studies of vaccine safety and effectiveness, methods for observational study research, and on the national history of pandemics and epidemics. His investigations have been cited extensively and were used for regulatory decision-making and public health recommendations by the FDA, CDC, PAHO, and WHO, as well as multiple other national authorities. So without further ado, we would like to introduce Dr. Izurieta for the first talk of the day. And the title of his talk is Use of Real-World Evidence in Accelerated Approval. Thank you so much.
Thank you. My name is Hector Izurieta. I'm going to speak about the use of real-world evidence in accelerated approval and also in emergency use authorization. This is my own opinion as a scientist. Anything I say will be used against me, not against the Food and Drug Administration. Basically, we would like to improve the understanding of potential uses of real-world evidence in FDA's regulatory decisions using a few examples. I will speak first about the regulatory framework, the emergency use authorization, the accelerated approval of one vaccine, and then try to summarize the So which is our regulatory framework for vaccines? We have, for licensure, we have what we call traditional approval. We also have accelerated approval and animal rule approval. I will not speak much about animal rule approval, but suffice at this point to say that in order to enter the road of animal rule approval, you have to have shown that you cannot go through licensure by either traditional or accelerated approval. Then animal rule approval would be open to you We have IND and emergency use authorization, which are investigational products unapproved in response to a public health emergency. Which is the degree of evidence required for vaccines? What's the evidentiary effectiveness? For traditional approval, we require substantial evidence in humans. For animal rule, substantial evidence in animals. For accelerated approval, we want the data to be reasonably likely to predict efficacy. And for emergency use authorization, it is reasonable to expect. And we have other plans which are expanded access and investigation new drug applications, which we are not going to deal with today. So the example of emergency use, I think everybody's familiar with it. We had a COVID-19 pandemic. In March 2020, the HHS secretary declared that circumstances did justify the authorization of emergency use of drugs and biologics. Basically, in June 2020, FDA issued guidance regarding the development and licensure of vaccines to prevent COVID, indicating that for an emergency use authorization, FDA should determine that the known and potential benefits outweigh the known and potential risk. And please highlight the word potential. Once a manufacturer submits an emergency use authorization request to us, we take into account the totality of the scientific evidence about it. So what happened with the first COVID-19 vaccine, which was Pfizer's? In December 11, we cited the totality of the available data, which provided clear evidence that the vaccine may be effective in preventing COVID-19 and supported the known and potential benefits, outweighed the known and potential risks. At the time, just to frame things, we did not have information regarding more rare events like myocarditis, for instance. That came post-licensure. But that was the information we had at the time. The safety data, including 37,000 and more people, followed up for a median of 2 months. That's not the standard for traditional approval, it's 6 months, and in some cases even longer. The effectiveness data included analysis of over 36,000, including immunogenicity, but also efficacy. And the available data did not allow a determination about how long the vaccine would provide effectiveness, but that is not a requirement. It's desirable to have that information, not a requirement for an emergency use. And participants afterwards continued to be monitored for 6 months, and that opened the door for the licensure path, which we will briefly talk about. In May of the same year, we expanded, the next year, sorry, we expanded to include 12 to 15 years using immunobridging. So we use afterwards basically the same data, but followed up for up to 6 months to approve the first COVID-19 vaccine with substantial evidence of efficacy and safety demonstrated from effectiveness data for approximately 20,000 vaccine and 20,000 placebo recipient, 16 years of age and older, and safety data from 44,000. We did evaluate the surveillance data post-licensure, emergency use authorization surveillance data pertaining to myocarditis, pericarditis, and that was part of our consideration. Now, the vaccine continued to be available under emergency use authorization for the age group for which we did not have enough data, which was 12 to 15 years at the time. Now, the third dose was approved under emergency use authorization. The booster dose, third dose for immunosuppressed people, challenged, if you want, and the booster dose for the general population of elderly individuals. The data submitted by Pfizer in this case did include real-world evidence to support this third dose indication. referencing several observational studies performed, some of them in foreign countries, suggesting waning of protection during the Delta variant surge among two-dose vaccinees. The file includes also safety and immunogenicity data against the reference strain Wuhan from approximately 300 adults. And effectiveness in this case was based on an immunobridging analysis. The VRPAC, the advisory meeting, discussed the booster dose in September 2021. Those presentations, including real-world data on vaccine effectiveness from CDC and data from observational studies in Israel, showing waning of protection from primary vaccination and effectiveness of a third dose vaccine. Please note that we did not review those data, but those data were presented at the VRPAC. FDA asked the committee then to vote on whether licensure could be issued, and the vote was negative, 16-2. The second question was on an emergency use authorization. Did the advisory committee agree that the data was sufficient to support an emergency use authorization of this third dose? And the vote was 18-0 in favor. Thus, in September 2021, based on the declaration of emergency and considering the totality of the data available to FDA to support the effectiveness of the third dose and the known and potential benefits evaluation compared to the known and potential risk, we authorized the third dose for individuals 65 and older and for individuals 18 to 64 at high risk. So let's move to, let's move from the emergency into a more traditional path. We are no longer in an emergency, but we have other options which require reasonably likely to predict that the benefit outweigh the risk. This is accelerated approval. How does it work? You need a serious condition. We need to agree that the condition is serious. This product, in this case a vaccine, should have meaningful advantages over available therapies, be that vaccines, drugs, or any standard of care. You have to demonstrate effect on a surrogate endpoint, which is reasonably likely to predict clinical benefit, and I will show you a few examples, one in particular. And you have to perform an adequate and well-controlled post-marketing study, and I put studies because we tend to ask for more than one study in this case, particularly if real-world evidence is used. It should be conducted with due diligence. What do we mean? We don't want to wait 10 years until we have the results of the study. We have a presumption of a favorable benefit risk. So I would say something close to 5 years would be acceptable. Further than that, it will be difficult. I will give you an example. In November 2023, we, under accelerated approval, approved Valneva's chikungunya vaccine for prevention of disease caused by the chikungunya virus in individuals 18 years of age and older who were at increased risk of exposure to chikungunya. Why this requirement of increased risk of exposure? Because that was a group for which we were clear that the potential benefits outweighed the potential risk. For people at low risk, this being a live attenuated vaccine with a rate of around 1.6% of adverse events that require a medical office visits or the encounter with a medical doctor was not so clear. So we added some warnings: severe or prolonged chikungunya-like adverse reaction, potential for vertical transmission unknown, and of course, syncope, which is frequent in almost any vaccine. The surrogate endpoint. What was the surrogate endpoint here? It was effectiveness based on evaluation of seroresponse. And the zero response was defined as anti-chikungunya virus neutralizing antibody levels above the threshold. The threshold was derived from a non-human primate model, but we accepted it, so this is not an animal rule approval. The zero response rate was determined by an assay among 266 vaccinated, PR and T, and 96 placebo recipients. I will give you an example of the, I would say, impressive results in regard to that. 28 days after single dose, 98.9% were seropositive among the vaccinees and 0% among placebo. And 180 days, 6 months later, that's after that single dose, 96% were still seropositive among the vaccinees and of course zero among the placebo. This was the surrogate. Then we asked for post-marketing confirmatory studies. The first one was a test negative case control observational study to assess effectiveness of the vaccine in the prevention of symptomatic laboratory confirmed chikungunya after a single vaccination in adolescents and adults in Brazil. As I said, we wanted the report to be submitted in time, and here are all the timelines we required. So it's less than 4 years. The second study is a pragmatic randomized study to assess the effectiveness of chikungunya vaccination in, again, the prevention of symptomatic laboratory confirmed chikungunya after a single vaccination. These timelines reach up to 4 years. It does not need to be specified in regard to being in Brazil or any other country. So we have these two studies. We also asked a 10,000 people safety study among vaccinees with the comparator. So in summary from this, we at FDA provide multiple pathways for vaccine access, which may allow the use of real-world evidence. That is not the preferred pathway. You have to demonstrate that this is the only pathway you have available, and we need to agree with that. Emergency use authorizations provide an expeditious pathway for larger access to investigational products. There are other options, expanded access programs in which you can, research the data, of course, investigation and new drug applications, which most of you probably know, if you need to fill an unmet need. During this pandemic, under emergency use authorization, FDA used real-world data to aid in the decision-making for COVID-19 vaccines. One of the examples is the third dose authorization. For accelerated approval, FDA instituted it to allow for earlier approval of drugs that treat serious conditions and fill an unmet medical need based on a surrogate endpoint. A confirmatory study is still required to confirm the anticipated clinical benefit. FDA used the accelerated approval for the approval of a number of vaccines, including this example of chikungunya. In this example, the sponsor offered 2 post-licensure studies, which were both using real-world evidence. Now, let me see if I can get into the challenge questions, which which one of these answers is wrong regarding accelerated approval? And I would like some show of hands for the correct answer, which could be the wrong answer, by the way. The condition needs to be serious. The vaccine provides a meaningful advantage only over existing vaccines, regardless of other existing treatments. Demonstrate effect on a surrogate endpoint, and adequate and well-controlled post-marketing confirmatory studies. Anybody thinks the condition needs to be serious is wrong? Nobody. Anybody thinks the vaccine provides a meaningful advantage only over existing vaccines is wrong? Yeah, raise your hands if you agree with me. This is a hint. So the second question, which answer is wrong regarding emergency use authorization? For an emergency use authorization, FDA must determine that the known and potential benefits outweigh the known and potential risk. The second one, FDA evaluates the request and determines whether the relevant statutory criteria are met, taking into account the randomized trial data submitted to FDA. This is about emergency use authorization. The third one, FDA evaluates the request and determines whether the relevant statutory criteria are met, taking into account the totality of the scientific evidence available to FDA. So who agrees that number one is wrong? Anybody? Good. Who agrees with me that number two is wrong? Good. Guys, we are improving here. I'd like to acknowledge a number of people from the FDA from whom I have stolen most of the slides I have shown to you. Kerr Prutzman, Sudhakar, who is here and will present after me, Peter Weina, who is also here and will present today, Maria Allende, Douglas Pratt, our boss, David Caslow, and all the review teams for all the vaccines we license. Thank you very much. Should I wait for questions at this point or not?
Thank you so much for that great talk. And if anyone has any questions, please jot them down, make a note, and we'll have our Q&A session at the end of the speakers' talks for this session, okay? And there is a place online where if you are thinking about it online, our online guests, you can just fill out the form so you don't forget. And then the folks in the room can also jot those down. So moving on to our next wonderful speaker this morning, Dr. Peter Weina. He is the Associate Director for Medical Countermeasures and Scientific Affairs in the Office of Vaccine Research and Review. Prior to his position, Dr. Weina actually served as a colonel in the U.S. Army, where he served for 46 years. And he was last assigned as the Director of the Office of Research Protections for the Defense Health Agency. He received multiple awards during his military service, including the Expert Field Medical Badge, the Order of Military Medical Merit, the Bronze Star for service in Iraq during the first year of Operation Iraqi Freedom, the Soldier's Medal for Heroism, Legion of Merit Medal, and the Defense Superior Service Medal. Currently, Dr. Weina is clinically active infectious disease staff at AT Augusta Military Medical Center and a full professor at the Uniformed Service University of Health Sciences. He's a fellow of the American College of Physicians, a fellow of the Infectious Disease Society, and he has more than 75 articles in peer-reviewed journals, 2 books, and 5 book chapters. As a result of his work, he's received several distinguished recognitions throughout his career. In the most recent years, he's received the BEYA Modern Day Technology Leader Award in 2020 and the Federal Laboratory Consortium Excellence in Technology Transfer in 2021. So without further ado, I would like to introduce our speaker for this morning, Dr. Weina. Thank you so much.
So good morning, and good morning to, we've got people here in the room, and I just want to acknowledge the thousands of people that we have online watching this as well. Hopefully I'll be able to provide some good information for you. Being the relatively new guy on the block, I got the short straw and got to talk about artificial intelligence challenges for vaccine development. And that's either a good thing or a bad thing, depending upon how you look at it. It's a good thing because there's not a whole lot to talk about because it's relatively new. It's a bad thing because there's not a whole lot to talk about because it's relatively new. So there's a lot of information that I hope to be able to give you of where we are so far. So these are our challenges. Just want to start off by saying this is an informal communication, my own best judgment, which some people have questions about quite often, but we'll do our best, and this does not bind or obligate the FDA in any way, shape, or form. So artificial intelligence challenges. Well, in one word, it's evolving. And if anybody has been tracking what's happening with artificial intelligence, it is moving so fast that trying to keep up on exactly A, what it is, and B, what it can do is a very, very difficult task. So like the technology itself, the regulatory science for trying to figure out how we can work with the groups that are out there, with the sponsors that bring things to us, that regulatory science is evolving as well. So where are we today in our thought process? First of all, because of this continuing education, I have to say our learning objectives, which is to appreciate and describe the current approach to AI challenges in regulatory oversight. So we have a couple of guiding principles that we are working with for artificial intelligence. So first of all, you can see how new this is. The very first one that we have for it is using artificial intelligence and machine learning in the development of drug and biologic products. That was in May of last year. So only a year ago is when we have our first guiding principle about artificial intelligence, and it's moving very quickly. And this was a discussion paper that was requesting feedback from stakeholders on the use and how do we actually regulate artificial intelligence. It's a very difficult thing to do, and we're like, well, you guys tell us what we should be doing and how we should be doing it. The next thing that happened was in October of last year, the executive order came out on safe, secure, and trustworthy development and use of artificial intelligence. And it was basically a broad oversight of how we as the federal government should be dealing with artificial intelligence. And this was to advance and govern the development, and actually had 8 guiding principles and priorities, which I'll go over in a few moments. And the last but not least, you heard Dr. Califf, if you tuned in yesterday, you heard Dr. Califf talk about a product that came out called Artificial Intelligence in Medical Products in March of this year. And this is how CBER, CDER, CDRH, and OCP are working together to give us some kind of guidance with artificial intelligence. And it was a publication that came out, very easy to find online, and gave us some broad general ideas as to how to move forward with this and how to have a safe, secure, and ethical deployment of artificial intelligence in the medical products development arena. So let's talk first of all about what the executive order said. October of last year, the executive order came out and it had 8 guiding principles and talked about how we as the federal government should be dealing with artificial intelligence. First of all, it should be safe and secure. Well, that kind of makes sense with all of this issues that we have with making sure that we don't have cyber attacks and making sure that artificial intelligence isn't a way in to break into our systems. Another one was to promote allowing the U.S. to lead a promotion to actually have the U.S. lead the way with artificial intelligence. Let's not stand back and see how it develops, but step out front and lead the way. Make sure that the development and use supports American workers. AI policies have to be consistent with equity and civil rights. And this is actually a fairly important one. And it's, it took a little bit of thought process and a little bit of understanding what's happening with artificial intelligence to understand where this comment is coming from. And the comment is coming from the idea that artificial intelligence has to be trained somehow. And that's one of the biggest concerns that we have about artificial intelligence. It's got to be trained, so how do you train it? What databases do you use to train it from? How do you make sure that there aren't inherent biases that are introduced into the artificial intelligence in its training program? And this is one of the biggest concerns that we have with artificial intelligence, is to make sure that not only do we understand where it's being trained from, but making sure that we aren't accidentally introducing biases into the outputs from artificial intelligence, and also make sure that we have a human check to make sure that when it does provide an output, that it's something that makes sense. The interests of the Americans must be protected, as you would expect from an executive order coming from the White House. Privacy and civil liberties must be protected, and manage the risks from governmental use of this. So we can't just willy-nilly say, well, this is really easy. We can, you know, if we give this project or this task to artificial intelligence, it's going to make it a whole lot easier for us and make it a lot faster. We have to make sure that we're doing it in a responsible way. And the federal government should be in the lead on how we use artificial intelligence. And by being out front, we make sure that we kind of, A, understand the risks associated with it, and B, help set the rules for this. So another guiding principle that came out, we talked about, was this, that Dr. Califf talked about, was this publication that came out as how we as the federal government, how we as the FDA are going to work across CBER, CDER, CDRH, all of our other groups, all of our other centers, and use AI responsibly. So first of all, we want to make sure that we foster collaboration to safeguard the public health. And this is making sure, I mean, everybody looks at the FDA as a single agency, but it's actually a collection of different centers. All of them have different personalities. All of them have different ways of doing things. All of them have different priorities that they find are really important to how they govern the products that come through their center. And we have to make sure that for things like AI, that we're working together as a group and doing this collaboratively rather than continuing in our little pathways of different groups. Make sure that we advance the development of regulatory approaches that actually support innovation. We don't want to make sure that, or we don't want to provide regulatory rules that basically shut down the innovation of AI. And it's a relatively easy thing to do if you think about the idea that when there is uncertainty, the easiest thing to do is to make sure that you clamp down on it so that you have a good handle on exactly what's happening and how things are going forward. But that kills innovation. And we want to make sure that we actually introduce things that are going to support and encourage innovation rather than closing down on it. We want to promote the development of harmonized standards, guidelines, best practices, and tools. This is nothing new for AI. This is, as any sponsor, large sponsor will tell you, trying to navigate the differences between EMA and the European Union or Health Canada, and then comparing that to the US FDA versus maybe Japan's PMDA versus all of these different regulatory agencies have got different rules and different ways forward. And why do they have those different rules and different ways forward? Is because we have different laws and different things in which our governments have told us we can do and cannot do in the registration and in the promotion of products that are produced in the various different countries. And because of those different laws, we have different pathways. Some people wonder why a drug may be allowed in France, but it's not allowed in the United States, or why it's allowed in the United States, but it's not allowed in Canada. And the reason is because we all have different laws, we all have different rules. One of the things that we want to try and get out ahead of and get forward thinking about is those laws that underlie the regulatory science, try and get out ahead of that so that we do have harmonization between our different groups, rather than the chaos that is sometimes run into by sponsors when trying to market a product across different countries. Oops. But there we go. And to support the research related to the evaluation and monitoring of AI performance, we want to make sure that we're not that we're not just blindly allowing things to come forward, but that we understand exactly what's happening as well in that, and by doing the research that's necessary. So one of the things that we have within CBER is the Artificial Intelligence Coordinating Committee. And on that committee, one of the things that we do is we talk about staff education, we communicate across the various different offices within CBER. We look at the landscape, we review the data that's out there and understand how AI is being used. We want to make sure that we align the AI strategy within the FDA so that regulations and ways forward that we come up with in CBER aren't totally different than the way they do it in CDER or in CDRH or in any of our other centers across the FDA. So one of the things that we did is we looked at all of the IND submissions that have happened between 2016 to 2023. And you heard Dr. Califf talk yesterday. Again, if you paid attention to what he was talking about, one of the things he said was, we have hundreds and hundreds of applications of AI within our agency. And that's true. Across all of the various different agencies, we have AI that has creeped in already across CDER, across CDRH, and CBER. But within CBER specifically, we identified 60 different submissions just within the INDs that involved the use of AI. And that use of AI was growing every single year from 2016 through 2023. And we anticipate, of course, that it's going to just continue to be more and more prevalent in the submissions that come in. Most of these are in oncology and in the infectious disease product areas. The vast majority of them had to do with predicting what was happening, classifying what was happening, clustering the data, and trying to look for anomalies. Now, CBER, when we look at packages, we don't just look at the clinical side of it. We don't look just at the efficacy or the safety, but we also look at manufacturing processes that are taking place as well. And a lot of AI is being introduced into the manufacturing side of things to make sure that the quality is where it needs to be. But again, you know, when you train the AI, the biggest thing I think we have concerns about is that the output may be based upon a little bit of a black box. We know that it's trained in a certain way. We don't quite, you know, of course, with AI, one of the things is it's not a standardized static algorithm that goes from point A to point B, but it becomes a little bit of a black box as to how that iteration takes place and how the AI gives its input or gives its output dependent upon the input. So we want to make sure to understand that. We also have a regulatory review subcommittee under that coordinating committee, and it explores review approaches to AI containing submissions. How are we actually on an individual basis looking at each one of these submissions that come in, and how is that being done within the various different groups? And we apply framework and standards as appropriate. We're trying to identify best practices, how the reviews are being done, and whether or not things are being missed that maybe should not be missed. Or, as again, on the other end, making sure that we're advancing innovation and not squashing down on the innovation. And we do a risk-based review approach to this, and the subcommittee then makes recommendations to the coordinating committee for the output. So how do you engage with CBER as a business, as a sponsor? How do you engage with us when it comes down to AI? First of all, we have a website, and these are available in the slides. If you click on the link, it'll take you directly to the site and give you the information. So we have one on artificial intelligence that gives you the broad overview. We also have the CBER websites that take you to that document on artificial intelligence and machine learning, how the, again, the artificial intelligence and medical products for the FDA in general. How do you request formal meetings with the office? And for broader applications, we have a thing called the CBER Advanced Technologies Team, the CAT, which actually meets and discusses ways forward for advanced manufacturing, which we'll hear more about in just a moment. And then for general AI inquiries, we actually have a site that you can send questions to and ask about AI. So again, for continuing education purposes, we have some challenge questions for you to assess if I communicated very well or if you were listening, one or the other. Under the executive order, includes one of the following guiding priorities: develop and use, support global, the development and use supports global usage. The AI policies are consistent with market forces. Privacy and civil liberties must be protected, or individual industry sponsors should lead the way. And I'm seeing some, indications from the audience here that the obvious one is privacy and civil liberties must be protected. Of course, on the first one, development and use supports Americans. The AI policies should be consistent with civil liberties and privacy, and that the government should lead the way in AIs. So, challenge question number 2, which of the following statements is not true? Use of AI in vaccine development and production was found to be growing every year in a recent analysis. Use of AI was primarily found to be growing in neurology and pediatric diseases. A recent survey found that there were around 60 submissions for AI, and AI uses in vaccine development is growing faster than regulatory science can accommodate the regulatory documents to assist sponsors. And again, indications here from the audience are the use of AI was primarily found to be growing in neurology and pediatric diseases. It's actually oncology and infectious diseases. So, in summary, most AI regulatory activity within CBER has been a relatively recent occurrence, as evidenced by the fact that most of our documentation and guiding documents are only a year old, and probably have only been worked on for about the last 2 or 3 years. And we would like sponsors to engage the agency early in the process to assure success. And working together, AI can be a very powerful and useful tool to speed the development and availability of biologics. We'll be getting to the questions when we get to the panel. Just as a closing thought, AI and its use in vaccine applications is evolving, and sometimes evolving so quickly that we're playing catch up with what's going on, and we need to get out ahead of this. And a plea is to engage the FDA early. Get to us early in the process. I think after several decades of working with the Department of Defense, mostly in product development of drugs, vaccines, and in devices, one of the things that I would go back and do differently is rather than trying to second guess the FDA, I would engage with the FDA much earlier, ask them questions, and make sure that we got some kind of feedback from them, even if the feedback was, it's too early to have a conversation with us. When it comes to AI, it's never too early to have that conversation because we're still learning, and what we need to do is to work together as partners with sponsors to make sure that we have good information and good data that we're working with to develop our regulatory science. So with that, thank you so much.
Thank you so much, Dr. Weina, for that great talk on AI. It's quite timely given the recent developments in research in this field. So we're very excited to have some questions for you later for that. And for our final talk for this morning's session, I'd like to introduce Dr. Sudhakar Agnihothram. He's the Assistant Director of Office of Regulatory Initiatives at the Office of Vaccines Research and Review. He has extensive experience in the product lifecycle regulation of vaccines for prevention of infectious diseases. He served as the chair of the FDA Multidisciplinary Review Committees responsible for emergency use authorization for the Moderna and Janssen COVID-19 vaccines. He's also a review committee member for Moderna COVID vaccine for Spikevax biological license application. He is also the office lead in managing several programmatic regulatory initiatives, including activities on frontline COVID-19 vaccines, and has been involved in advancing stakeholder partnerships in these areas. So we're very excited to have him talk today, and we're going to hear his talk on CBER's advanced manufacturing programs for sponsors. So thank you very much and welcome.
Well, first of all, I like to thank everyone for the opportunity for inviting me to present here. And then, with excellent presentations from my colleagues before, I hope to keep the momentum going. So I am the Associate Director of Office Regulatory Initiative, Sudhakar Agnihothram, and then I manage several regulatory initiatives, including COVID-19 activities at the office level. And today I'm going to talk to you about advanced manufacturing program for CBER regulated products. So, and because this is for continuing education purpose, the learning objective would be to understand the framework and approach on advanced manufacturing program for CBER regulated products. So obviously, this is the disclaimer. My comments here are an informal communication and represent my own best judgment. By no way, means, or form it obligates or nor it binds to FDA. So this is the website, that you can go and find about advanced manufacturing for public health, emergency preparedness and response. We have just gotten out of COVID-19 pandemic. And then I'm not going to go through every single link on this website, but the intent here is to give an overview in terms of the resources that are available for anyone who is interested in looking at how FDA is involved in advanced manufacturing, fostering approaches for advanced manufacturing, how FDA works collaboratively within the agency as well as with other public-private partnerships. So this website is basically just like a one-stop shop for everything. And up front, I just wanted to put it out there for anyone who is interested in going and learning more. But throughout my talk, what I would be doing is letting you getting educated in terms of the important points that are available on how FDA works on fostering approaches for advanced manufacturing technologies and how those are important in the context of our bread and butter, the regulatory review. So what is exactly advanced manufacturing? So integrating novel technological approaches for manufacturing. So there is a new technology that helps in improving a current manufacturing process or solve a current manufacturing problem that definitely comes under the umbrella of advanced manufacturing. The next one is you already have a technology that has been used for manufacturing of drugs or biologics or devices, but you're using an established technique in a new or an innovative way. Like, for example, like, you're improving that technique or using that technique for a different purpose. Like that again qualifies for advanced manufacturing. And if you are developing a production method in a new domain where there are no defined like best practices or experience, that, for example, also qualifies under advanced manufacturing. And as we always say, this is something like, that you must have heard from a lot of FDA speakers, like, so it's case by case basis. So every field has a different set of production techniques that are considered advanced, and like, one size never fits all. And then we always review on a case by case basis in terms of the technology that is presented, like, how that is helping solve a current existing problem, or like, how the technology would be helpful in a particular context. So what are the benefits of advanced manufacturing, right? So we keep talking about this advanced manufacturing. The first one is to improve drug quality. So obviously, like, the mission of FDA is to protect and promote public health. And then, like, needless to say, what comes in that is like to ensure that every drug or biologic or a device, like, that is approved or authorized or whatever it is, like, it's of the best quality. So improvising the quality of drugs, strengthen domestic drug supply. So, and as you might hear, like, during upcoming discussions after this talk, like, there might be, like, during the pandemic preparedness or like for a pandemic preparedness, or you did see during the COVID-19 pandemic that there was a lot of disruptions in terms of, like, drug supply. So utilizing advancing manufacturing technologies can mitigate some of those issues and strengthen drug supply domestically as well as, like, internationally. Which takes to my next point, which is prevent drug shortages due to quality related issues. Like, for example, like, if you have like one sole manufacturing facility, like, and then, like, you identify a quality issue there. And then if that manufacturing facility alone serves as, like, sole resource for supply of a drug or a biologic, then using advanced manufacturing technologies and then, establishing like smaller manufacturing facilities, like, that are geographically, like, placed appropriately in terms of supply chain, that can also help in terms of, like, preventing drug shortages. And obviously, like, as you did see during the COVID-19 pandemic, like, the speed at which vaccines or drugs or devices are brought to use by the public is, like, very, very essential. And then one of the benefits of the advanced manufacturing is to expedite drug product development and then the speed at which the products are brought to the market. And obviously, like, all of this will result in greater agility and resilience in response to a public health emergency. So just an overview in terms of biological products regulated by CBER, like, so Dr. Peter Marks is, like, our center director. And then so under CBER, like, we have several offices, like, that works on several different products, like, blood and blood components and derivatives, like Office of Blood Research and Review, vaccines, which is where, like, I'm from, Office of Vaccines Research Review. We regulate preventive and therapeutic vaccines. And then the newly formed Office of Therapeutic Products, which is a super office, like, they work on tissue, cell and gene therapies, transplantation. And allergenics obviously comes under the purview of Vaccines Research and Review. And then there are also related devices, including IVDs, that we regulate. So these are, in a nutshell, some of the products regulated by CBER. So I'd like to give an overview about the CBER Advanced Technologies Program. One of the missions of the CBER Advanced Technologies Program is to fund advanced research and development projects to support regulatory science and innovation. As an example, recently there was continuous manufacturing of mRNA vaccines, a project that's headed by MIT that was funded by FDA for about like $82 million, which I think, like, is one of the integral components in terms of, like, getting mRNA-based vaccines and mRNA-based therapeutics to the market in a rapidly scalable fashion, and also like, you know, in an expedited fashion in terms of like, you know, delivery. Build internal scientific and regulatory expertise, obviously, like, you know, so within our team, we meet very often in terms of like, you know, discussing the issues related to the advanced manufacturing. And then like, you know, we also invite external speakers to talk about advanced manufacturing technologies that are developed by academia as well as industry. And then through these processes, like, you know, we build internal scientific and like, you know, end of the day, it all matters in terms of what we review in terms of the product life cycle of a product. So we do build regulatory expertise for advanced manufacturing. And there's a CBER Advanced Technologies team, or CAT, that has representatives from like, you know, each offices within CBER that apparently spearheads all of these activities. So promote dialogue, education, and input among CBER staff and between CBER and prospective developers, advanced manufacturing technologies, to encourage their implementation in the manufacturing sector. So pretty much like, you know, everything that I just spoke about in terms of working internally as well as externally through public-private partnerships in terms of fostering advanced manufacturing technology is like, you know, forming the core in terms of what this CBER Advanced Technologies team does. So it consists of a small cross-functional group representing CBER leadership, relevant policy review and inspection programs, and all of the CBER offices, including Office of the Director, Office of Vaccines Research and Review, Office of Blood, Office of Therapeutic Proteins, and Office of Compliance and Biologic Products. Like, you know, all of these, like, you know, offices have, each of them have their representatives in the CBER Advanced Technologies team. So how does the CAT work towards like fostering advanced manufacturing? They provide early access to interactions with CBER, which I'll be talking in the next few slides, prior to filing a regulatory submission. And they discuss technical and regulatory issues related to the implementation of innovative manufacturing and control strategies. Because anytime, as I told you before, when you come up with a new technology, or when you use an existing technology for a new purpose, or when you're using an existing technology in a new domain, there will be inherent challenges that would have to be discussed. And as my colleague Pete Weina just mentioned about, it's never early to engage with FDA. So one of the key focus of this CAT like team is to have early meetings with manufacturers in terms of assessing their technologies, and then like assessing the regulatory readiness, and then also looking at the problems like, you know, that these technologies may bring, and then come up with mitigation strategies in terms of like, you know, how to resolve those issues. So these are all like what these CAT technologies team do. Talking about early engagement, so there are several phases. I mean, as some of you may already be very familiar with this product lifecycle in terms of like pre-IND, IND, and then phase 1, phase 2, phase 3, and then like licensing of a particular product, and then there is post-marketing, and then improvement of a product, like, you know, in a post-marketing sector, either via manufacturing technology or whatnot. But even before we go into the product life cycle, like, you know, this CAT interaction with the CAT team provides non-binding regulatory advice in terms of chemistry, manufacturing, and controls, and innovative approaches to product developments. I mean, these are not PDUFA regulated or anything like that, but these are more, you have a media, or you have a platform to submit requests to the CAT technologies team, and then like, you know, this team really evaluates all of these requests in a non-binding and provides like, you know, regulatory advice. There is also this interact meeting that you can go find on the FDA website, which might be very specific to a product and an indication for human use, that covers the 3 key areas in terms of like CMC, preclinical, and then pharm tox, and like, you know, the clinical. So it's a pre-IND. It's even before you come in to submit a pre-IND application is what this interact meeting is about. These are all non-binding regulatory advice, but then once you enter into the zone of like, you know, binding regulatory advice, like, you know, so you come in through an established pre-IND application, which is basically like, you know, you have a lead product, you want to take that into the clinic, you have a plan that you have charted out on the whiteboard in terms of what your CMC looks like, what data you have from your preclinical studies to support, and then what are your plans for clinical development. So in that area, like, you know, you also have specific focus in terms of each of these CMC, preclinical, farm docs, and then clinical, where you could ask questions to FDA through a pre-IND meeting request. And obviously, we always encourage sponsors to submit IND application because it's free. And then, like, even if you're developing a product, which is, for example, for an infectious disease indication, which is not endemic in US, we still like request that sponsor submit an IND application because that helps you grant a lot of like and utilize a lot of meetings, including end of phase 2 meeting, for example, where you can bring in the data that you have generated from phase 1, phase 2, and then talk to the agency about how that supports your phase 3 development plans in terms of like how you scaled up your manufacturing to meet the commercial needs, like, you know, how your animal studies supported, like, you know, your safety or like, you know, other immunogenicity endpoints that you were looking at that was further evaluated in phase 1 and phase 2. And then what are your plans for like, you know, commercial manufacturing, and then what are your plans for lot release, et cetera, before you go big on your phase 3, which apparently like, you know, is your pivotal study to support the licensure of a product, which can go through, as my colleague, Dr. Hector Izurieta described, several approval pathways, traditional, accelerated, annual rule, et cetera. But the crux here is that like early engagement with FDA under an IND provides you with all of these opportunities for engagement with FDA and to ask right questions throughout the clinical development so that you can have your product developed in a right way within a reasonable amount of time and then get it to the market to meet the unmet medical need. So advancing innovative manufacturing technologies through extramural funding. So what's the role of like, you know, extramural funding in terms of advancing innovative manufacturing technologies? Since 2018, CBER has awarded several grants and contracts to support research products. I just gave you an example about continuous manufacturing of mRNA vaccines that's headed by MIT, which is an $82 million program. And then this funded research addresses knowledge and experience gap identified for emerging manufacturing and testing technologies and support the development and adoption of such technologies in the biological product sector. Basically, this is an evolving areas, and then you would obviously need a lot of funding in terms of testing out new technologies, developing new technologies, and then assessing the readiness of these technologies for commercial scale manufacturing, et cetera. And then extramural funding helps very much in this regard. And we at CBER, we at FDA have programs where we fund for fostering these technologies. So some of the examples: 3D bioprinting for tissue engineering, novel manufacturing approaches for cell therapy products, including the continuous production. I mean, in terms of vaccines, obviously, like, you know, continuous productions for vaccines. I mean, there was a recent publication from Meghna University in terms of a continuous manufacturing process using a recombinant vesicular syndromitis virus vectored SARS-CoV-2 vaccine that apparently reduced the manufacturing time in half. Obviously, that's when you talk about a pandemic and when you talk about a public health emergency and the pandemic response, reducing your manufacturing time in half in terms of commercial manufacturing is something that's really huge. So that's something we do as well. And then obviously process modeling and simulation, and then non-destructive analytics for evaluating product quality. I mean, these are going into deeper dive in terms of looking at quality of a product, how you assess quality of the product, and then what type of approaches that you bring in, including analytical approaches for evaluating quality of the products that can also be implemented in lot release, etc. So the scope of these CAT meetings are novel technologies with significant impact on product development, manufacturing process, and control strategies can be submitted under the CAT meetings to engage FDA. Manufacturing and analytical methods for which CBER has limited experience. I mean, if someone brings something new and then they want to discuss the applicability of the new technology in terms of commercial scale manufacturing, like, you know, supporting a product approval, etc., they can do that as well. And not necessarily like, you know, these are like very product specific discussions. These are more technology specific discussions that apparently can be applied for several set of products. For example, if you take continuous manufacturing, it does not necessarily need to be applied for one particular vaccine, but the technology that you develop, depending upon what you develop, can be applied for vaccines that can be developed against various infectious diseases. So how do you actually submit the CAT meeting request? This is the link to the website that you can go and access the meeting request submission and how you do that. But basically, it's just a 2-page, including figures and tables. It's a backgrounder that provides the following information, which is description of your technology, why the technology or product class is very novel and unique, and what's the impact of this technology on that product class, for example, and then summarize your manufacturing or development plan in terms of what you envision doing using the technology. So these are the specifics that apparently go into these CAT meeting requests. And questions regarding perceived regulatory, technical, or other challenges for implementation. As I told you before, our bread and butter is to regulate products throughout their life cycle, to support licensure, and to bring efficient and effective reviews and approaches where we can actually engage on early with sponsors in terms of assessing their technology and then how they can be carried through the product life cycle to support a product approval. So the review process, once the request is submitted, it's initially triaged by CAT coordinators, and then it's assigned to the relevant review officers depending upon what technology, what is the product class that is addressed by that particular technology. And then there are recurrent CAT meetings, that apparently are that occur where you discuss the specific request specific technology. It might not be all done in one meeting, but you might need recurring meetings in terms of addressing your questions as well as addressing your problems. So the possible outcome could be after they go through the meeting request, obviously you may get your meeting request granted, and then you may get your responses to the submitted questions and recommendation to request other meetings for product specific discussions, which I just talked to you about in the previous slide, which are more going along the product lifecycle development for a specific product that includes pre-IND through pre-BLA, end of phase 2, that type of meetings. So some of the examples or technologies that were discussed, including continuous manufacturing, automated, scalable, and remote control systems for manufacturing cell therapy products, improved cell lines for vaccine antigen production, and adeno-associated virus vector manufacturing. And my colleague Pete Pina just spoke about the use of artificial intelligence and advanced imaging technologies for real-time product quality and assessment. AI technologies are also used in advanced manufacturing for real-time product quality assessment. So we work on reviewing that as well. Multi-product manufacturing facility design. So you might have one facility that apparently manufactures multiple products. I mean, it could be drug, it could be a biologic, and then but these facilities might have different suites in which these manufacturing might be carried out. And obviously genome editing has become something really big in terms of the rising wave of genetic engineering as well as gene therapy over the last few years, which also falls under the umbrella of the technologies that are discussed under the CAT request. So I just wanted to give you a very brief overview in terms of the Center for Advancement of Manufacturing, Pharmaceuticals, and Biopharmaceuticals. So this is apparently established by the FDA, where again, so each of the individual center, CBER, Office of Counterterrorism and Emerging Threats, as well as CBER and CDRs, they evaluate advanced manufacturing technologies on their own, but we also work very collaboratively through the Center of Advancement of Manufacturing, Pharmaceuticals, and Biopharmaceuticals to accelerate the development, implementation, and evaluation of advanced manufacturing technologies. Because so you could have a biologic product that apparently uses a device to deliver that biologic product or that drug, and then that would require a collaborative approach within different centers within FDA. So that is what we do under the center, and obviously advanced drug product development science. And train a world-leading regulatory workforce. Again, our bread and butter is to make sure that all of this ultimately results in an efficient and effective regulatory review of products to enable timely licensure of products, like, to meet the unmet medical need. So in this process, like, as I told you before, partner with public, private, like, we have a lot of public-private partnerships that we work with in terms of evaluating and then getting engaged, as well as, like, getting knowledgeable with advanced technologies that are out there. Oops. So one of the example here that I wanted to give is, like, so there was a workshop that was conducted in 2023, the FDA PQRI, which addressed the utilization of artificial intelligence in advanced manufacturing, as well as discussed several advanced manufacturing technologies. When I talked about the communication in the last slide, so this is one example of how we get out there and then talk to small business industries, as well as, like, established manufacturers in terms of how we regulate or how we engage in terms of evaluating advanced manufacturing technology. And this is one example of that. So I just wanted to make sure that, like, folks also got visibility on the ICHQ 13, which was established by FDA for continuous manufacturing of drug substance and drug product, where the objective was to provide a harmonized guidance. There is a guidance document on this ICHQ 13, continuous manufacturing of drug substance and drug product, that talks about clearly the development, implementation, and assessment of continuous manufacturing technologies used in the manufacturing of drug substance and the drug product. And the scope of this is to apply the continuous manufacturing of chemical entities and therapeutic proteins, and the conversion of batch manufacturing to continuous manufacturing. Obviously, continuous manufacturing is something that is integration of stepwise manufacturing processes into a single process that can be controlled by machine, so that, like, the manufacturing can happen over time, and it clearly allows to meet the supply demand. And then when you look through this guidance document, what you will find is that the nuances in terms of how to integrate these approaches for drug substance and drug product manufacturing, et cetera, is clearly outlined here. So I just wanted to, before I finish my talk, also wanted to bring the attention of audience to the Advanced Manufacturing Technologies designation. This guidance, the link for this is provided here. This is in draft form, and then the final guidance is expected to be published by the end of the year. And then just the key highlights of this, right, is what does this guidance address in terms of advanced manufacturing technology designation? Is the process for submitting an AMT designation request. What is the eligibility criteria for your technology to be designated as advanced manufacturing technology, and then the data and the information to be included in the designation request, how FDA will communicate in terms of receipts, and how would we determine, like, whether a technology qualifies an advanced manufacturing technology designation request, and how we assess that, as well as the process by which we engage with holders of designated, like, advanced manufacturing technologies and applicants. Then what are the benefits of this program? Again, this is a standalone program which is separate from, for example, the CAT request that I talked about, which is very specific to Center for Biologics Evaluation and Research. And then you can go read this guidance at your leisure to find out and expand on all of these specifics. So just to summarize everything, we are committed to accelerating the adoption of advanced manufacturing technologies. Again, like, as my colleagues spoke before, engaged with FDA early on to discuss the regulatory and technical issues associated with your innovative technology. I mean, when someone develops an innovative technology, they might think they have solved the problem of the world, but apparently when it gets into the regulatory realm area in terms of developing, like, process controls, analytical controls, in terms of scaling up, in terms of assuring quality, assuring stability, there are a lot of parameters that go into assessing the quality of the product, like, that has to be built in terms of developing a technology. So all of these are nuances that need to be developed once you have a technology down. So really, like, encourage everybody who is interested in this to engage as early as you can with FDA, and we collaborate internally and internationally to build the scientific expertise and regulatory framework necessary to evaluate like, emerging technologies. And I just gave you a few examples in terms of the center of excellence that we have, as well as the ICH Q13 guidance that we published. So for the purpose of continuing education, my challenge question number one, which of the following best fits the description of advanced manufacturing? Use of established technique efficiently, use established techniques effectively, use established techniques in new or innovative way, and substitute established techniques using other techniques. So yeah, I just see Matt raising his hand for B. Think the answer is like, use established techniques in a new or innovative way. That's C, which if you remember in the initial slide of advanced manufacturing technology, what fits in that, there was that answer for that. So challenge question number 2, which of the following is an example of advanced manufacturing? I think you guys should be able to get this. Manufacturing of drug substance and drug products in a GMP facility, use of continuous manufacturing, example, vaccines, adeno-associated virus vectors for gene therapy, NMR for advanced research, 2D bioprinting. The answer is use of continuous manufacturing. So I'd like to thank everybody again for taking time to listen, and I'd also like to thank the CBER Advanced Technology manufacturing team, our center, our office, my colleagues, Pete and Hector here, Meghna, and our colleagues from OBPV, Craig, and also the organizers for giving me this opportunity. And my contact information is provided here if you want to follow up with any questions, but we'll also have a panel for discussion. Thanks very much.
So I would like to invite the 3 speakers back up to the discussion table. And I would like to invite our audience members who may have questions for the talk to make their way to the microphone at this time. I have heard from our online team that the online chat has been buzzing, so we also have some questions from our online audience as well. But we can begin as soon as everyone gets nice and settled. All right, well, we'll start with our first question in the audience, please.
Good morning. Thank you for taking my question. Eric Sanchez, University of Puerto Rico School of Pharmacy. To Dr. Sudhakar. Actually, when I was hearing your talk, thinking about your counterpart at CBER, the emergency technology team, is working on the assumption that advanced technologies might be graduated in the sense of use. So once you have a new technology that is being started and is being used for X amount of time, they graduate that technology and no longer takes additional submissions sort of saying for evaluation for the team. Like, right now, process analytical technology using NIR, chemometrics are things that are done routinely right now from the advanced technology initial perspective. I wonder if CBER is working on their same assumptions so that, once an advanced technology is being used for X amount of time, that will be graduated and there will be, no need for further involvement of your CAT team in the future on those submissions. So that being said, it would be good to know which technologies are graduated as they are evolving on your principles, and then understanding that those should be used by companies in a routine manner. So that would be the case.
Yeah. If I understand your question correctly, what you're asking is like when you come in with the technology and then you present a problem or you want to discuss a specific approach or a specific issue. Once that is resolved, would there be further engagements in terms of like fostering the technology further down the regulatory pipeline for manufacturing? I mean, is that exactly your question? Or
yes, particularly on new submission from other companies, maybe.
Yeah, absolutely. I'll first answer and then I'll ask my colleagues to chime in as well. As I presented to you before, I mean, like these CAT meeting requests, they are like when you come in way early on, way initially, they are non-regulatory, non-binding, where you just present the technology, discuss the approaches, discuss the applicability of it. And then once you have a path forward in terms of utilizing those technologies, like then you take that forward for your product development, and then you come in with either like a pre-IND meeting where you describe the technology, and then if your question is also whether you can reference your previous discussion, I don't see why not. And like, I don't think it hurts. There is anything like, I don't think there is anything that prohibits you from actually referencing your previous meeting with CAT. And then when you present it in the context of a specific product, like, the stability of the product, how you're assessing the quality, how you're using the technology to take that product through phase 1, phase 2, commercial manufacturing, and using that for analytical comparability instead of a lot to lot. I mean, all of those are done under an IND pathway. And then, so the focus shifts to a specific product at that time. So, Pete.
Yeah, I actually sit on the CAT, and so I've been on it for about the last year and a half. And as a lot of these technologies that are coming in are very immature, and that's why these are not regulatory binding meetings that we have. And we get tons and tons and tons of applications, and obviously we can't look at all of them, and some of them are actually, as you brought up, technologies that have already been looked at or are considered, considered routine. So that's one of the ways that we provide feedback to sponsors that come in with what they think is an advanced technology, but it's actually not. Another way that the agency is actually, I think, getting really good at doing this is to actually have designation programs. And you see that. I also sit on the committee that is looking at the new guidance for platform designations and things. We also have one, for example, for animal models. And all of these are then put into databases that sponsors can look at and say, oh yeah, there's this one that's already got a designation. And that then the technology itself or the animal model or whatever actually has been given a designation is then available to the public through the access system. So that there are ways that we do provide feedback to individuals so that they're not kind of spinning their wheels.
Okay, so let's take one question from our online audience.
Oh, good morning, Dr. Sudhakar. So please, can you advise if there are collaborative activities with other regulatory organizations, such as the European Medicines Agency, to improve the efficiency and speed of new product development via the use of advanced manufacturing technologies?
Absolutely. I mean, so I'm not going to go into a specific of the advanced manufacturing itself, but we do work very collaboratively with our partners, EMA, Health Canada, and then other national regulatory agencies. And then so we at the Office of Vaccines, for example, we do have like quarterly cluster meetings, where we meet and discuss product specific issues in terms of where a particular product is. I mean, these are all done under memorandum of understanding, confidentiality agreements, and then there are limitations in terms of what specifics we could share. But in that realm, there could be advanced manufacturing issues that can be shared on a specific product. For example, if it's a quality related issue, stability related issue that impacts the distribution of that particular product globally. So there are definitely avenues where we work with global partners. And Pete or Hector, if you want to add anything.
Okay, thank you so much. And we'll take the next question from our audience member.
Hi there. Thank you all for speaking with us today. This question is mainly for Dr. Weina, as you said, you pulled the short straw. You mentioned that the treatment of algorithms or the functioning of AI and machine learning models was somewhat of a black box. So I was wondering, from a regulator's perspective, what is your sorry, a moment. What is your view on any unique challenges on the validation and verification of AI models? And do you have any recommendations for those of us in the industry who are using AI?
Yeah, that's a really great question about the idea that, AI is sometimes viewed as this little bit of a black box, but I think the validation and the verification of how it produces an output should have some kind of predictability to it. It's supposed to, I think that in best case scenario, it's smarter and faster than what a human could do it, but come to approximately the same output as a human. So part of that validation is testing it, in the system, making sure that the outputs are going to more or less agree with what the output of maybe a group of experts may potentially come up with. I think that it's going to get harder and harder to validate it as these algorithms become more and more opaque and really hard to look at. So I think that what's going to end up having to happen is that in the initial development, we're going to have parallel systems in which humans are still intimately plugged into the system and verifying the output of what's happening. As we become more confident in the output and that biases are not being introduced, and that's really one of the biggest issues and one of the biggest concerns about AI systems in use. Once we've gained some confidence, I think that there's going to be more reliance upon the output of that particular system. But as the system matures, that's going to have to be, spot checked every once in a while. It's just like, we can't check every single pill that comes off of a production line. We do a sampling of it to make sure that the quality is there. That same type of check is going to have to be in place for AI.
Thank you.
All right, we'll look to our online audience for another question from them.
All right, this question is for Hector. Excuse me. Please elaborate on what level of evidence, I'm sorry, that's the wrong one. What, if any, modifications for emergency to use authorization and risk-benefit analysis are in process or under considering arising from COVID-19 vaccine experience? Are there any lessons learned being implemented from the real-world evidence?
In an emergency, let me give you a hypothetical example. Let's assume that between January and February of the year 2020, a new disease appears in our borders and starts spreading rapidly. And we need to use something, let's call it emergency use authorization, to authorize vaccines for that new disease, for which we got the genetic, the genetic material very late in the process from the country in which the disease started, I have to admit. Would you imagine that we would have authorized a vaccine with very strong standards of evidence by December of the same year? We regulators, the heavy ship called regulator, that's called emergency use authorization. In the emergency use authorization, we need to accept something that is reasonably likely to predict benefit, and the benefit risk balance should be positive. To give you a few examples, we authorized these vaccines, and very quickly, once the vaccines were started to be used, we identified through post-licensure surveillance myocarditis, pericarditis, following the mRNA vaccines, and very rapidly identified risk at the groups at higher risk, males, adolescents, young adults. The rates were still fairly low compared to the dreadful effects of the vaccine. Remember that, to give you another example, in nursing homes, a significant proportion of the populations of those nursing homes initially died during COVID-19. Almost 3,000 deaths per 100,000 nursing home residents, and I can give you figures from other age groups. When we authorized the Janssen vaccine, it was in February 2021, we authorized it using reasonable evidence for the emergency use authorization. But in April, very shortly afterwards, we identified TTS, which is a disease with a rate of, in this case, in the case of Janssen, the Janssen COVID-19 vaccine, less than 4 per million. And we did it in collaboration with our partners from other regulators, public health agencies, very early. And then we issued recommendations with CDC to restrict the utilization. And then eventually the vaccine was taken out of the emergency use authorization as requested by the sponsor. So I would not say we are not evaluating our emergency use authorization regulations. We are, and we will continue to. But in my technical opinion as a scientist, and this is not the opinion of FDA, our emergency use authorization regulations work very well, and our collaboration with other partners and with other regulators of public health agencies, including CDC, was excellent. I would say superb, considering the severity of this pandemic. I can elaborate. I am asking when I need to.
Yeah, I'd just like to add 2 more points. One is the emergency use authorization enables you to bring a drug to the market quickly, like, under emergency use. But for example, if you take COVID-19, the standards under which EUAs were approved were very rigorous, so both of the mRNA vaccines were approved using clinical endpoint efficacy studies, which apparently the length of the safety follow-up, was a little different compared to what you would have for a biological license applications, which is 6 months of safety follow-up. But the safety database that we had was pretty equivalent to, in fact, more than 3,000. I mean, it was in several thousands. And contributed for a substantial evidence in terms of authorizing those vaccines. But still, as my colleague Hector mentioned, once these vaccines go out, post-market, you find out a lot of adverse events, including myocarditis, thrombosis, thrombocytopenia syndrome. We work very tightly with our colleagues who are sitting here at OBPV as well, like CDC, etc. And then the systems are so very tight and then very heavily networked that we identify these and we immediately label these. And for example, with Janssen, we even came down with the temporary halt in terms of vaccine administration and proceeding clinical trials, etc. So lessons learned from COVID really worked well. Yep.
Okay, we'll take our next question from our audience.
This is for Hector. Russ Coleman. I'm interested in the need for real-world evidence, blurring the lines between animal rule and a classical clinical regulatory pathway. What do I mean by that? So formally with the DoD, and we were concerned about adversaries weaponizing biological agents, troops getting exposed to aerosolized agent, for which there's no clinical patient population. So animal rule. I've heard recently of discussions with the FDA where, well, this is going to be animal rule, but we'd love to see some surrogate clinical endpoints. So for example, ventilator-required pneumonia to augment that animal data. So is the FDA thinking on animal rule evolving with this desire for real-world evidence?
Yeah, we are evolving animals as artificial intelligence evolves. It has demonstrated we also evolved. So let me give you some perception, my perception of how I see animal rule at this point, and I will ask Pete afterwards to elaborate on Sudhakar. We have a stronger standard, which is traditional approval. So you will have to demonstrate to us that traditional approval is not a viable path. If you pass that hurdle, then we will require you to try something else. Accelerated approval, it would be the next option in line. For accelerated approval, you would need a surrogate. You will have to show to us that you cannot have a surrogate in humans in order to go to the next step. So if you don't have the viability of surrogate in humans, then we can consider animal rule, which could be a high standard of benefit in an animal model. But we have to go stepwise. Now, if we get into an emergency situation, if the HHS declares an emergency, then emergency use authorization would be your next option. And that adds a little more flexibility. But I will let Pete elaborate.
Hey, Russ. As far as the use of the animal rule, I think that as Hector has already brought up, we are evolving. As new data comes to us, we evolve our guidances. And the guidance, that's why we're not going to use a guidance from, 2000 unless we've looked at it and said, hey, yeah, all of this stuff still is in place, but the guidances are going to evolve. But as far as animal rule, we may approve a product using the animal rule so that we can actually field it. But once we actually start putting it into humans, it's really good to start getting some feedback as to whether or not this is doing what it's supposed to do, if the safety is the way it's supposed to be, if the efficacy actually turns out to be what the animals predicted. And we all know from, those of us that have done a lot of product development know that, we can only go so far with the animals. A good rule of thumb that I always learned very early on, whether it's true or not, is that the animals do pretty good about 80% of the time, but 10% of the time, when we get something good from the animals, turns out not to work in humans, and 10% of the time when it doesn't work in animals, it will work in humans. So we're going to miss the mark part way out there. So the animal rule is in place so that we can have a product out there and available, but we still need that follow-on data. And we do that with all of the other approvals as well. We even do that with traditional, in which we have the highest standard, and we continue to provide feedback through a variety of different surveillance systems like VAERS and things like that, in which we want to make sure that we look for adverse reactions and say, hey, this is starting to show up on our radar when it might not have shown up in the clinical trial. So even as Hector's brought up, there's kind of a gradation. We would have our traditional approval, accelerated approval, and then animal rule down there. But all of them have got kind of backup systems to make sure that maybe what we saw at first blush, it isn't exactly the entire story, and we need to read further in the book before we get that output.
Just to be clear, I was talking about clinical efficacy data, not the safety data.
Yeah, but same thing for clinical efficacy data. Again, the animals are only going to show us so much. And the approval in the animals give us, best possible when, we can't do a clinical efficacy trial in humans. But once we start putting it into humans, we should be paying really close attention to what that data is. And real world data is the way to do that.
We'll look to our online audience for questions.
Sure, this question's for Peter. What does CBER means by AI containing submissions, or the data is AI generated? The text of the application is AI generated?
Yeah, so AI is starting to creep into just about everything that we do. When we say that AI can really insert itself just about anywhere into that process. It can be AI for an AI containing submission may have AI in the manufacturing process and getting, the quality, or it may adjust the amount of product, different products that go into a combination product based upon feedback down the line in the manufacturing. So AI may be used for that. Or it may be something simple as AI generated minutes for a meeting. Hector just forwarded something to me yesterday in which we had a meeting and AI actually produced the minutes, and we, he sent it out, and when you look at it, the minutes are like, I didn't actually say that. But AI generated minutes interpreted that given the totality of the conversation, that's probably what I meant to say, but that's not really what I said. So, the idea of AI can creep in anywhere in the submission, and we need to understand where that AI comes in, because that's going to be a variable that we need to account for in the output of the review of the submission, and whether or not that's going to potentially change what a human looking at it and doing that work may have done.
I'll just give one specific example. For example, in an investigational new drug application, which you're developing for broadly protective vaccine, AI might come in a space where AI might be sampling all of the sequences, like several thousands and millions of sequences of this pathogen in the past, and then come up with a consensus sequence, like, or a group of cocktail of sequences. Flu is just an up-top example that comes up to the mind. And then that gets submitted to an application, and then based on that, a sponsor makes a claim that, this vaccine is going to be broadly protective. And I think someone in the audience asked before about validation, et cetera. But the reality comes to play when you actually do an assay, when you have an immunogenicity endpoint using a validated assay to prove that basically the cocktail of antigens that you have selected or the sequences that you have selected by the AI, based on serology data, epidemiology data, virus surveillance data, no matter whatsoever, that apparently results in a measurable endpoint that can function as an evidence that it is indeed broadly protective against several strains of pathogens. So that's one example of AI coming in. Another example is just in advanced manufacturing, controlling the continuous manufacturing process, and then processing control. So these are very specific examples, but like Pete mentioned, anywhere.
Yeah, so we have a principle at FDA, first do no harm. In fact, if I remember well, that principle came from Isaac Asimov's Rules of Robotics, many, many years ago. Basically, we need to know that you are using AI and how you're using it and for which purpose. And then we will know how deeply we have to get into that to be sure that first there is no harm. And then we will see how beneficial it has been. This is going to be an evolutionary, very steep, very steep stair, and it is complicated, and we have to be careful.
Yeah, so getting back to the question, AI containing submission, it can be anywhere in the submission at any point, and what we need to do is have you identify where was AI used.
Okay, well, we just have about a minute left in the session. So I think that we could just give the speakers a big round of applause for all of their presentations and their answers today. And thank you so much for coming and entertaining all of the questions so eloquently. And for now, we will go on a break for the next 10 minutes. And our next session of the morning will start promptly at 10:30. Thank you so much.
Well, good morning again, and welcome to the second session of the day. And the second session will be about post-approval safety of biological products. And the first talk in our second session is probably a good segue from the topic of vaccine development, because we will touch upon post-marketing evaluation of medical products, which obviously include vaccines and other biological products regulated by CBER. So the first presentation will be really looking at post-marketing safety from a slightly different angle. We will be talking about use of biological products in pregnancy, which is an interesting topic on its own, and I'm sure we'll have a good discussion afterwards. So the first presentation will be delivered by Dr. Meghna Alimchandani, who serves as the Deputy Director of the Division of Pharmacovigilance in the Office of Biostatistics and Pharmacovigilance in CBER. Her work is focused on pharmacovigilance planning and passive and active post-marketing safety surveillance for products regulated by the center. She also actively participates in the design of post-marketing studies and REMS programs, and oversees the pharmacovigilance reviews for biologics license applications. Dr. Alimchandani is a 10-year FDA veteran and served in various review and supervisory roles. She received her medical degree from the Albert Einstein College of Medicine in New York and completed residency in anatomic pathology at the National Institutes of Health. So please join me in welcoming Dr. Alimchandani.
Thank you, Larissa, for the introduction. Good morning, everyone. My name is Meghna Alimchandani. I'm in the Division of Pharmacovigilance in CBER's Office of Biostatistics and Pharmacovigilance, or OBPV, and I'll be talking to you about post-marketing surveillance for product safety in general, and then, as Larissa said, more specifically, product safety in pregnancy. So these are the learning objectives for this presentation. I will start by going over a couple of org charts to give you some context for our pharmacovigilance discipline reviews in CBER. Then I will broadly cover several topics. I will describe the importance of safety throughout product life cycle, and then focus on post-marketing surveillance strategies, including passive surveillance and active surveillance. We will discuss signal evaluation and risk management. Then we will briefly touch on the pregnancy and lactation labeling rule, and I'll end with some examples to bring all of the above together for you, and we'll end with some summary and conclusions. So this is an agency level org chart. It shows the different centers. So we are located in the Center for Biologics again. This is your CBER track. Now, I wanted to point out that many of the guidance documents that I'll be referencing are CBER and CDOR guidance documents and are applicable to both centers, and there's a lot of collaboration between the centers as well. This is our CBER org chart, and we are located again in the Office of Biostatistics and Pharmacovigilance. We work closely with our product offices in CBER, and we are responsible for doing post-market surveillance for all of CBER-regulated products. And as was mentioned before, this includes vaccines, therapeutics such as cell therapies, gene therapies, etc. Okay. So the next couple of slides are to show you the importance of safety throughout the product lifecycle. This slide is just to remind you again of the various stages of development, and other speakers have already touched on this. So really, development begins with preclinical research as the pre-IND stage, goes through the early phase human studies, the IND stage, the larger clinical trials, and then culminates with the biologics license application and approval decision, and where our focus is really in the phase 4, the post-licensure setting. Now, product safety is critical throughout the life cycle of the product. And FDA ensures that products are safe for their intended use. These are the slide lists, many of the activities that are occurring throughout the life cycle, again, starting with preclinical research, early phase human studies, inspection of manufacturing facilities, monitoring of lot release, inspection of clinical sites for compliance with GCP, and then post-market adverse event surveillance. So why does CBER conduct post-marketing safety surveillance? Now, we are mandated to do so. It's codified in the Code of Federal Regulations, and there are several limitations of the pre-market safety database that makes it critical to have robust post-marketing surveillance practices in place. Pre-market safety database may not detect safety issues that arise when products are marketed to the general population. Post-marketing surveillance may reveal interactions with comorbidities, interactions with other concomitant products. Often, pre-market safety database may exclude certain vulnerable populations, such as pregnant women, which is, again, the theme of this talk, and I'll talk a little bit more about that on the next slide as well. Pre-market clinical safety database may be limited by small sample sizes. This is especially true for rare diseases, and observation periods and follow-up may be limited as well, and may not be able to pick up the adverse events that have longer latency periods or that are more rare. And finally, unless a trial has a dedicated safety endpoint and was powered for a certain safety outcome, inferences about safety are limited by concerns about post-hoc analysis with multiple comparisons. So for all of these reasons, post-market safety surveillance is absolutely critical. So I'm going to be referring throughout the presentation to this 2019 guidance, and the link is provided below, and this is some of the information from that guidance as well pertaining to pregnant women during clinical development. So as I mentioned before, during clinical development of most drugs and biological products, pregnant women are actively excluded from trials. So at the time of licensure, you may have little or no human data really in the label for the product. If pregnancy does occur during a trial, the usual procedure is to discontinue treatment, monitor pregnant women to assess pregnancy outcomes, and again, at the time of marketing, you are left with very little data. For the next few slides, we're going to talk about post-marketing surveillance for product safety and pregnancy. I'm going to spend some time going over this slide because it introduces a lot of concepts that we'll talk about throughout the rest of the presentation. So this display, it's a slide displays for you the pharmacovigilance plan on the left side of the slide, and on the right you have pharmacovigilance actions. So the World Health Organization defines pharmacovigilance as the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other medicine vaccine related problem. So it is a very broad definition. It includes medication errors, administration issues. Now on the left side, you have the safety specifications. Now these are based on the clinical safety database, the available data that is available for that product at the time of our review of the application. Now the important identified risks, potential risks, and missing information are based on the sponsor's assessment of that data. So it's very important to understand, we review that, we review their proposed safety specifications. We may or may not agree with them. We provide feedback to them frequently, but FDA is not approving the pharmacovigilance plan. So unlike a USPI, unlike the label that we approve, we're not approving a PVP. We acknowledge that this is the sponsor's assessment. We are free to disagree, provide our recommendations. Now on the right side of the slide, you have the pharmacovigilance actions. Now these are determined by our assessment of the safety data. So I'm going to go over each of these. So what do I mean by routine pharmacovigilance? This is routine safety surveillance. Again, this is mandated by the Code of Federal Regulations. Biologics are under 21 CFR 600.80. There is mandatory adverse event reporting by manufacturers for any serious and unexpected or unlabeled adverse event. The manufacturer has to submit a 15-day or expedited report. For all other adverse events, so serious and labeled and non-serious events, they have to submit non-expedited reports. In addition, the manufacturer submits to us something called periodic safety reports. These are aggregate assessments of all of the safety data that is available to the sponsor during that reporting interval. The periodic safety reports are submitted to us quarterly for the first 3 years post-licensure and annual thereafter. Now, in addition to the routine PV, FDA may ask for enhanced pharmacovigilance. And you may see that sometimes in approval letters as well. And what I mean by enhanced pharmacovigilance, it's still passive surveillance, it's still spontaneous adverse event reporting, but we may ask the sponsor that for a certain adverse event of special interest, we want you to submit 15-day reports, and we may put a time period on it. We may say for 3 years after licensure, all reports of myocarditis or all reports of whatever AESI it is must be submitted as 15-day reports, regardless of seriousness or label status. So that is sort of another layer that we can add to the routine pharmacovigilance piece. Now, the next arrow is pointing to safety-related post-market studies. So these come in different flavors. So safety-related post-market studies can be conducted by the sponsor, or can be conducted by academia or by the US government. So it depends on who is conducting the studies. If they are sponsor-conducted studies, they may be post-marketing requirements, or they may be post-marketing commitment studies, or they may be voluntary studies that the sponsor is initiating on their own. And then in terms of the study design, they may be targeted studies looking at a specific serious risk or a specific risk, and be powered for that safety outcome. So you could have targeted studies, or you could have general safety surveillance studies, which is looking at all safety outcomes, a catch-all for safety data and conducting follow-up of patients for a certain period of time. So there are many flavors to these safety-related post-marketing studies. And then finally, we have something called REMS, or risk evaluation and mitigation strategies. There is a high bar for REMS because it is tied to the benefit risk balance for the product. We only require REMS when they are necessary to ensure that the benefits outweigh the risks. And we have a few products in CBER with REMS programs as well. Okay. So post-market surveillance product safety in pregnancy, how does the information that I presented to you in the last slide tie in with pregnancy specifically? So use, as we said before, use in pregnancy lactation is often not studied in the clinical trial. So this is often categorized as important missing information in the sponsor's pharmacovigilance plan. We will have passive adverse event reporting. We may also ask the sponsor to perform enhanced pharmacovigilance. We also have active surveillance. As I told you, we have safety-related studies. So these are post-approval pregnancy safety studies. These may be sponsor-conducted studies, post-marketing commitments, requirements, and voluntary studies, or U.S. government-conducted studies, or academia-conducted studies as well. And we'll go in a little bit detail with all of those categories. So this slide goes over what we want you to report in a pregnancy exposure report. So this is the information. These are the factors that we will consider as we assess pregnancy exposure-related adverse event report. So we ask that this information be included in that ICSR, or individual case safety report. So please describe the adverse outcome with as much clinical detail as there is, the exposure description, what was the product, what was the biologic, the therapeutic or the vaccine product, the dose, the frequency, the route of administration, the duration of treatment, was it a one-time treatment, is this a chronic treatment? The gestational age at exposure is really an important factor that we consider. The maternal age, medical pregnancy history, use of concomitant medications, supplements, other substances, and exposures to known or suspected environmental teratogens. Now, these kind of reports, these pregnancy adverse event reports, and any adverse event report is coming to us under the broad umbrella of passive surveillance. So with passive surveillance, we are doing continuous safety monitoring for all of our licensed products. These are spontaneous adverse event reports. So these are voluntary for, in terms of healthcare providers and the public, it's voluntary adverse event reporting. But as I mentioned before, for the manufacturers, the sponsors, this is mandatory adverse event reporting. And this is codified under 21 CFR 600.80. They must submit expedited or 15-day reports for all serious and unlabeled adverse events. And these must be submitted to us within 15 days. The regulatory definition of seriousness is also displayed on the slide. All other adverse events would fall under the category of non-expedited reports. And then, as I mentioned, we also have periodic safety reports that the manufacturer submits to us quarterly for the first 3 years, annually after that. And again, this is the sponsor's assessment based on all the data that they have for that reporting interval. Okay. So this slide goes over the different pharmacovigilance databases. So we have the Vaccine Adverse Event Reporting System, or VAERS. Now this was established in 1990. It is co-managed between FDA and CDC. And there are a lot of vaccine surveillance activities that are complementary and between these two organizations, between our organization, FDA and CDC, for vaccine safety. So those all the vaccine adverse events reports are going into VAERS. For the therapeutic biologics, those reports are going into the FAERS, or the FDA Adverse Event Reporting System. You also have the manufacturer's global pharmacovigilance database. Again, this includes their data from not only for the US adverse reports, but outside US as well, and for other indications that may not be approved in the US. So this is a very rich database as well that is maintained by the manufacturer. And then we have other databases as well maintained by other organizations. We have the World Health Organization's FIGI-base, and Vigilance, an example of EMA's database. So foreign regulatory agencies have their own PB databases as well. So this is the web page for VAERS. It's a screenshot of their web page. I wanted to also mention that the VAERS data as well as the FAERS data are publicly available. The data sets are publicly available as well. And anyone can submit a report. There is online reporting on the VAERS website. This is the MedWatch screenshot of the MedWatch website. Again, anybody can submit a report to VAERS. So the next couple of slides are going to go over some of the strengths and some of the weaknesses of passive surveillance databases like VAERS and FAERS. So pharmacovigilance database like these accept all reports regardless of the plausibility of the product causing the event or the clinical seriousness of the event. Now, some of the strengths of these databases is that they can rapidly detect potential safety problems. Again, they are capturing a huge amount of data. And because of this, it may lead to potential detection of rare adverse events. It is open-ended for hypothesis generation, and there is great geographic diversity. We are getting reports from all over the US. We are getting reports from the US and outside US as well. We have foreign reports coming in. So we are capturing an enormous geographic diversity with these reports. We also may have the capability to monitor production lots. Now, we may be able to do this if the lot information is available in the report. So this really depends on whether that information is provided or not, but many times we undertake lot analysis as well using the available information on lots. This slide goes over some of the limitations of these pharmacovigilance databases. There is missing or inaccurate data. Their reported diagnoses, again, are not verified. Anybody can submit the reports. There is underreporting because, again, from the public, it is voluntary reporting from their side. There may be reporting bias. There may be stimulated reporting. There is an absence of control groups or comparator groups. And because of these limitations, we may not be able to assess causation. Also, we may not be able to detect long latency adverse events. It's less likely to get reported. Okay. Now, this slide goes over the role of product utilization data. So unlike with passive surveillance, we do not have denominator data. Unlike a setting where you have a study, you know, the number of patients you've enrolled, we don't have that with passive surveillance data. So as a surrogate for that, we often use sponsor distribution data to provide context for adverse event reports. And this accounts for total amount distributed in the US or outside US, worldwide distribution. We may ask for lot-specific distribution data if we are doing lot analysis for certain adverse events. And also we may ask the sponsor to provide distribution in specific age groups, for example, women of childbearing age, if that information is available. Now, some things to keep in mind is that we cannot calculate incidence rates from passive surveillance data. This is always a limitation of FAERS or VAERS data. We may be able to calculate reporting rates, but that's not incidence rates. And then not all doses distributed were administered. So again, this is a surrogate and not actual denominator data. This is some language that is from this ICH draft guidance that is cited at the bottom of the slide. And I just wanted to touch on reporting requirements for pregnancy exposure. So are sponsors required to submit ICSR or individual case safety reports for pregnancy exposures that are not associated with any clinical adverse event? And the answer to that is no, they are not required as per this guideline. However, if the pregnancy exposure is associated with the clinical adverse event, then that is required to be submitted. Sorry about that. We're just having some technical challenges. Okay, so thank you. So we come to a couple of challenge questions now. So pregnant women are often excluded from clinical trials, and use in this population is categorized as missing information in the pharmacovigilance plan, or PVP. True or false? I think everybody is going to get that. It's a true statement. Again, drives home the importance of post-marketing surveillance for these reasons. The next challenge question. Post-marketing surveillance for product safety in pregnancy includes A, adverse event reporting in accordance with 21 CFR 600.80, B, enhanced pharmacovigilance, and this would be specified by FDA, by CBER, submission of periodic safety reports at quarterly intervals for 3 years post-approval, at annual intervals thereafter, post-approval pregnancy safety studies, all of the above, and again, the answer is all of the above. Okay, so switching gears from passive surveillance to active surveillance. So this is this guidance, and the link is provided. I already referred to it previously. This is our 2019 guidance of post-approval pregnancy safety studies. It has a lot of information on post-marketing surveillance for product safety in pregnancy, including passive and active surveillance, and it's a great resource for that discusses different study designs as well. So before going any further, just wanted to focus on a very simple, very basic definition of safety-related post-market study and what we mean by that. So remember, any study, any clinical trial, any post-marketing study is always going to be collecting safety data. There is no study that does not collect safety data. But when we are talking about a specific safety post-marketing study, what we imply by that is that the study is being conducted specifically to evaluate safety or further investigating a safety issue associated with the product. So the primary safety endpoint is really what defines a safety study. So in this guidance, again, there are 3 approaches that are outlined for pregnancy safety surveillance. This includes pharmacovigilance, pregnancy registries, and database studies. Now, these are not mutually exclusive. These are all complementary and help to provide a comprehensive, holistic surveillance of product safety during pregnancy. So pharmacovigilance, we have already mentioned. By that, we refer to the passive surveillance and spontaneous adverse event reports. We'll talk a little bit more about pregnancy registries and database studies. So pregnancy registries are usually prospective observational cohort studies to collect data on product exposure during pregnancy and associated maternal and infant safety outcomes. And it's great when they have a comparator group. It could be an active comparator. If an active comparator is not possible, then we can use historical controls as well. Pregnant women are enrolled and followed until the pregnancy outcome. Infants are followed for a specified time period, usually 6 months or to a year after birth. And then database studies are electronic healthcare, are using electronic healthcare data. This is administrative claims data or EHR data, electronic health records. It requires mother-infant linkage. Now, the big advantage of these database studies is they can leverage very large data sets, but there is sometimes a lag time, and it requires additional infrastructure to be built in, like establishing mother-infant linkage. The advantages with pregnancy registries is that you can put them in place right at the time of approval, right post-approval. You can start enrolling patients. There is no lag time. The data can be very rich. You can get patient-level data, and again, you're following them prospectively. Usually database studies are retrospective. However, with pregnancy registries, one of the issues that we run into very often is low enrollment, and that can impact the feasibility of pregnancy registries. So this slide goes over what we are looking for in terms of study protocols. So usually the full study protocol is submitted to us for post-marketing requirement or commitment studies in the post-approval period. And these are all the things that we're looking for: description of the study population, the outcomes, the control and the comparator group, rationale for sample size, data collection methods, data analysis plan, statistical analysis plan, and the study milestone dates. And there are 3 study milestone dates that are always included in approval letters, again, for PMRs and PMCs. We track these milestone dates, the dates for the final protocol submission, the dates for study completion, and then the date for the final study report submission. In addition, for the PMR and PMC studies, we are reviewing them on an ongoing basis throughout the life cycle of the study. So after the protocol, we're looking at annual PMR, PMC status updates reports, interim reports, any protocol amendments that come in, and then, of course, when the study finishes, when the final study report is available, we look at that and try to decide if additional regulatory action, labeling change is going to be prompted by the final study report or not. So I touched on this a little bit earlier. So post-approval pregnancy safety studies, again, we have sponsor-conducted studies, or in the other category, you have U.S. government or academia conducted studies. For the sponsor-conducted studies, they may be PMRs, PMCs, or voluntary sponsor studies. And I'll show you the criteria for these different types of studies. So a post-marketing requirement study, as it says in the name, it's required by the FDA, and it's required under the Food and Drug Administration Amendments Act of 2007, which gave us the regulatory authority to impose PMRs. These are targeted safety studies to assess a specific serious risk. And I'll go over the study purposes again under the regulations for these PMRs. But very important to keep in mind that they are for a specific serious risk, usually. And then the post-marketing commitment studies, that they are defined as being agreed upon study between FDA and the applicant. These are usually general safety surveillance studies, so they're not powered for a specific serious outcome, but we didn't have enough data at the time of the pre-market review, and we want to collect data on all pregnancy exposures in the post-marketing period. So we often have post-marketing commitment for general safety surveillance studies. And you'll see this in many of the vaccine approval letters when the vaccine is indicated for women of childbearing age. And then the third category is when the sponsor initiates a voluntary study. Now, the voluntary study is, again, any safety signal from any source the sponsor has to report to us, but for the voluntary studies, we don't track them as closely as we are tracking the PMRs and PMCs. The PMRs, PMCs, as I mentioned, we track the milestone dates. We post publicly information on the study status, whether it's ongoing, whether it's completed. The voluntary studies, the sponsor has initiated and is collecting data, and they may give us updates in the periodic safety reports and voluntary studies as well. Okay, so this is the guidance on PMRs. These are the study purposes under the regulations that would trigger a post-marketing requirement study to assess a known serious risk related to the use of the drug, to assess signals of serious risk related to the drug, or an unexpected serious risk when available data indicate the potential for a serious risk. So we have a lot of flexibility under the regulations, but at the end of the day, they're usually targeted safety studies. And in the examples portion of this presentation, I think I have a couple of examples of PMRs as well. This provides a snapshot of the agency's PMRs and PMCs, specifically for post-approval pregnancy studies. This was a slide that was presented publicly last year in a workshop. It collates data from both CBER and CDER for these pregnancy studies, and you can just see that the vast majority of the PMRs and PMCs were established in the past 10 years. Okay. So switching gears now from the sponsor-conducted studies to the U.S. government studies. So we have studies that are conducted by the FDA, by CBER, and this is under the BEST program or the BEST initiative. The acronym stands for Biologics Effectiveness and Safety Initiative. And then we often have vaccine studies that are conducted by our colleagues at CDC, and they use the Vaccine Safety Data Link. Now, these are population-based data sources. They're using electronic healthcare records data, they're using claims data, and they often complement the sponsor studies. Now I think we come to the next two challenge questions. Post-approval pregnancy safety studies may be conducted by A, sponsor post-marketing requirement studies, B, sponsor agreed upon post-marketing commitment study, C, a sponsored voluntary study, and D, study conducted by US government or academia. And as you have guessed, the answer is all of the above. These are all different types of post-marketing pregnancy safety studies. So this is the last challenge question that I think I have in this presentation. So post-approval safety study designs may include A, observational cohort for registry design, B, database studies using electronic healthcare data, and this refers to administrative claims data and AHR data, spontaneous adverse event reports, A and B, all of the above. So the correct answer is not all of the above. The correct answer is actually A and B, because remember, spontaneous adverse event reports, those are not studies. That's part of our passive surveillance data. So really when we refer to studies, we're talking about registries or database studies. Okay, so the next portion of the talk will focus on our principles for signal evaluation and risk management. And this is a best practices document that was posted earlier this year, actually in January 2024, and goes over some of the principles that I'm going to go over. So this is how signal is defined in that best practices document, and I'm not going to read it out for you, but it's there for reference. What I want to point out is that a signal is meant to be hypothesis generating. It does not mean that there is causal association. It means further action is needed to verify the signal. So the first step is signal identification. The second step is signal evaluation, which is a really important step to try to gather information, use all our available post-marketing surveillance data sources. This would include passive surveillance, this would include active surveillance, data from ongoing studies or completed studies, data from literature if we need, then collaboration with other foreign regulatory agencies, information from them. So all of that goes into our signal evaluation process. So this is again from the guidance on post-approval best practices. Signal evaluation in regards to pregnancy exposure is really important because it helps us identify a product's potential for adverse developmental outcomes, including teratogenicity. And it's important because product-associated adverse developmental outcomes are potentially preventable. And these listed over here are some of the outcomes of interest that we're looking for, including preterm delivery, small for gestational age, pregnancy complications, miscarriage, stillbirth, abnormalities of the immune system, other infant outcomes. This slide hopefully helps to put it all together. So with we have at the top of the slide, you have the sources of safety data. You have clinical sources from pre-market, clinical trials, sources of data, including post-market, data from spontaneous reports, fairs and fairs, data from electronic healthcare databases, population-based data sources, and then again, medical literature, foreign regulatory agencies. So all of these are the sources of data that we are reviewing and screening. It helps us identify signals, and then we evaluate signals. The next step is signal verification, and then it culminates in regulatory action. And regulatory actions can include communications, label changes, recalls, and withdrawals, etc. So as part of signal evaluation, we are doing both qualitative review as well as quantitative review. The qualitative review includes review of individual case safety reports. We conduct case series analysis. These are the questions we're asking ourselves as we're performing signal evaluation. Is the adverse event unexpected? Is it labeled or unlabeled? Are there unusual patterns or demographic clustering, geographic clustering? Is this event new? Is there biological plausibility that would support a causal association? Is there a dose response? Is a positive rechallenge or a dose response? Absence of alternative etiologies, alternative explanations, and how do reporting rates compare with background rates? We also perform disproportionately reporting analysis to see if a particular product adverse event pair is reported more frequently than other product event pairs in the passive surveillance databases. And then finally, we are reviewing data from other sources, including literature and post-marketing studies. And this slide really lays out the different risk management options. So after signal identification evaluation, the risk management options would include things like communications. We have a safety and availability web page on CBER where we can post safety communications. One of the most powerful tools of communication are to have label changes. These could be voluntary label changes by the sponsor or mandated label changes that the agency is requiring the sponsor to do. We can also initiate withdrawals or recalls if that is warranted. Inspections, new safety information may trigger new post-marketing studies, post-marketing requirements, and FDA may also require REMS if there is new safety information to support that. I'm going to touch very briefly for a few seconds on the pregnancy, lactation, and labeling rule. So the goal is to provide the prescriber with the relevant information for decision-making when treating pregnant or lactating women. The PLLR format improves presentation of the currently available data. There is animal data included. This is section 8 of the label. There is human data is added when available. So often when we complete post-marketing, when the sponsor complete their post-marketing commitment or requirement study, we review the final study report, and that may need to lead to additional label changes and incorporating that human data under section 8 of the USPI. And when there is no data available, the label will explicitly state that no human data was available. I'm going to go through the examples fairly quickly because I think I'm a little bit short on time, but these are really there for your references, and it goes through some of the concepts that we talked about in the rest of the presentation. So this is a publication of the vaccine safety data link, their infrastructure to look at pregnancy safety active surveillance. Again, the VSD is a collaboration, our studies conducted by the CDC, is a collaboration between CDC's Immunization Safety Office and integrated healthcare organizations. This is another publication by CDC using VSD data, looking at influenza vaccination and safety in pregnancy. And this is another example of monitoring pregnancy safety during COVID-19 vaccine exposure. And this is a nice paper because it talks about different pharmacovigilance systems. So it talks about passive surveillance data, VAERS data, as well as active surveillance data looking at COVID-19 vaccines in pregnancy in the US. These are a couple of papers that go over, again, for your reference. These are BEST studies or building infrastructure in the BEST system, the CBER BEST program, to look at maternal and fetal outcomes. This is an example of a research study that we did to see how effective the PLLR labeling format was to convey information about risk in pregnancy to providers who perform different focus groups. Again, section 8 is the section of the label that has information on pregnancy safety. Okay, and this is from an approval letter, and the link that's all posted publicly, again, goes over some enhanced pharmacovigilance requirements that I've talked about before when we asked the sponsor to specifically submit expedited reports for certain adverse events for a certain period of time. And then we also over here listed are some post-marketing requirement studies, which are, again, targeted safety studies for a specific serious risk. The VRBPAC meeting is another forum where we can exchange information. The VRBPAC is an example of one AC. We have other advisory committee meetings for therapeutics as well, where which we can use to discuss safety issues and brainstorm ideas for post-market safety monitoring. Then I just have one slide, I think, for my summary and conclusions. So despite rigorous safety evaluation during pre-market phases of clinical development, post-market safety monitoring is necessary due to the limitations of clinical trials. Post-marketing surveillance includes many approaches, as I've gone over during this presentation, including passive and active surveillance strategies. FDA may require post-marketing studies by manufacturers. FDA and the applicant may have agreed upon post-marketing commitment studies as well. New databases have expanded population-based surveillance capabilities. FDA and CDC share many vaccine surveillance activities. Again, the BEARS is co-managed by FDA and CDC. And our goal is to ensure safe and effective products for the U.S. population, including populations of interest such as pregnant women. Thank you so much. Over to you.
Thank you very much, Meghna, for the comprehensive overview of the post-marketing safety evaluations that are available, and including examples, particularly with COVID vaccine and other in pregnant populations. All right, so our next presenter is Dr. Craig Zinderman, who is the Associate Director for Medical Policy in the Office of Biostatistics and Pharmacovigilance in CBER. In his role, Dr. Zinderman provides expertise and advice to office and division leadership on medical product safety surveillance and the management of post-marketing safety issues. He also serves on a variety of inter- and intra-center working groups developing FDA regulations and guidances on post-marketing safety and adverse event reporting for regulated industry. Dr. Zinderman has over 2 decades of experience at the FDA with a lot of knowledge on public health issues and post-marketing safety. Now, many of you know that FDA participates in developing and harmonizing guidelines for medical product development through the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, also known as ICH. And so today, Dr. Zinderman will talk about the recent update for the ICH E2D guideline for post-approval safety data and standards for reporting of individual case safety reports. Dr. Zinderman, please.
Thank you, Larissa. Okay, so good morning, everybody. As Larissa said, I'll be talking about ICH E2D guideline. I'm the Associate Director for Medical Policy in CBER's Office of Biostatistics and Pharmacovigilance. Oops. Okay, so first we'll go over the learning objectives. I'll start with an overview of what this guideline is. To be really brief, this guideline discusses the management and reporting of post-approval safety information by industry. And then we'll talk about some background information on why it was decided to revise the guideline in the first place. Then we'll highlight some of the biggest changes in each section of the revised guideline, notably some key new definitions and terminology. We'll talk about the different types of individual case safety reports that are discussed in the guideline. There's spontaneous reports and solicited reports. And most importantly, we'll go through the section of the guideline that talks about the different sources of information, things like digital platforms, patient support programs, literature, and we'll talk about what's changed in the new draft for each source. So what is E2D? As Larissa said, ICH is the International Council for Harmonization. It's a collaborative group of regulators and industry from what is now a large number of countries and regions. And they get together and develop harmonized guidelines on various aspects of scientific and technical aspects for pharmaceuticals. The title of this guideline, Post-Approval Safety Data Definitions and Standards for Management and Reporting of Individual Case Safety Reports, basically says what it talks about. This guideline is part of the efficacy group of ICH guidelines. That's what the E is for in E2D. It provides harmonized procedures, foundational definitions and concepts, and guidance to industry. ICH guidelines refer to industry as marketing authorization holders, or MAHs, and I'll use that term throughout this talk. But in brief, it provides guidance on how marketing authorization holders should collect and report post-market safety information to regulators. For example, it defines what is an adverse event or an adverse drug reaction, what makes an adverse event or adverse drug reaction a serious event, or what makes an event unexpected. There are also definitions for other fundamental terms like primary source reporter, consumer, and healthcare provider. I should note that the original E2D title was, or the original E2D guideline was limited to expedited ICSRs, and the title, and had expedited as part of the title. In the new version of the guideline, we've expanded the scope to include all ICSRs, because really these concepts and definitions apply to all post-market individual case safety reports and not just expedited ones, and we've changed the title accordingly. So by way of background, the ICH E2D guideline was originally written and published by ICH in 2003. There are several new or emerging post-market safety information sources that are more and more, either they're brand new or they're more and more frequently used, and a lot more frequently used than they were back in the early 2000s. And these sources typically vary in their characteristics, and they differ in how much they contribute to what we know about the post-market safety, what we know about a product in the post-market setting. But they can have they can be really large volume, particularly something like digital platforms, which include social media, and can be burdensome for marketing authorization holders and regulators alike to monitor and to manage. So ICH stakeholders wanted to address the burden presented by these new emerging data sources. And ultimately, ICH determined that the definitions, and in particular the guidance provided in the original guideline, is no longer sufficient to provide guidance on current pharmacovigilance practices and needs given these new sources. But there was also a need to update the definitions in the guideline, clarify the management and reporting of cases from literature, and to generally update the terminology in what is now a 20-year-old guideline. So an ICH expert working group, EWG, was formed in 2019 to come together and update the guideline and bring it in line with current post-market safety surveillance. I do want to note that the recommendations in the revised guideline are harmonized to the extent possible, but there are a number of differences in post-market safety reporting requirements across the different countries and regions in ICH. So it's not always possible to provide guidance, provide a recommendation that's harmonized, that's consistent in all the different regions when their reporting requirements differ from each other. Some of those regions have changed their reporting requirements, changed their regulations since the original guideline was published in 2003. So those countries now differ from other regions in ICH, and they differ from the original guideline. So we had to navigate these various differences. So in places in the guideline where it's needed, where it's applicable, we have a note where there may be local or regional exceptions to the guidance that we provide in the guideline. And we refer MAHs to refer to the local or regional requirements to know what they need to know about that particular concept or term for that country or region. Despite the regional variations, the EWG believes that this new version of the guideline provides what is now a harmonized framework for best practices in managing post-approval safety data, particularly involving reporting terminology and involving the emerging new data sources. I should note that these slides highlight only some of the significant updates and changes made to the guideline. One should refer to the complete document for all editorial changes and updates, and there's a link there, and there'll be more links at the end of the slides. These are the groups that made up the expert working group, or that still make up the expert working group. There was fairly wide representation across both regulators and industry, with 9 different regulators participating and 5 pretty large industry stakeholder groups participating. The plenary working party included MHRA from the United Kingdom and Health Canada. There are several brand new definitions added to the revised version of the guideline. Some of the important definitions that we're going to discuss. One is individual case safety report, or ICSR. And this definition includes a portion that defines the minimum criteria for reporting a case, an ICSR, to regulatory authorities. And we'll go over what those criteria are. ICSR did appear in the original guideline, but it only appeared once. This is a term that wasn't used very often back in 2003. When it appeared in the original guideline, it was just talking about the ICH E2B electronic transmission standard. That's the ICH standard for submitting a case, actually transmitting a case, the format that a case gets transmitted to when industry sends it to regulators. But otherwise, it wasn't used in the original guideline. Since that term is used much more frequently now, and it's in FDA regulations, it's in many countries' regulations, we now give it an explicit definition in the guideline. The guideline also provides definitions for expedited report, and primary source, which is the individual who provides information on a case to the MAH. So ICSR. An ICSR is a description of an adverse event or an adverse drug reaction or other observation that's occurring in an individual patient at a specific point in time. As I said, there's another part to this definition in the guideline, and that's the minimum criteria for reporting an ICSR to a regulatory authority. So there's 4 minimum criteria. A case has to have an adverse event or an adverse drug reaction. There has to be a medical product. The patient has to have been exposed to something that we regulate. An identifiable patient and an identifiable, at least one identifiable reporter. Okay, as I said, there's a definition for expedited report, and we define this broadly as an ICSR that meets the requirement for reporting as soon as possible, but not later than 15 calendar days after day 0. So we just define it with a timeline, any ICSR that meets these local requirements. The specific requirements for which ICSRs need to be submitted within 15 days are defined by the local or regional authority. So we just use this 15-day definition, and we refer those specific requirements or leave those specific requirements and criteria to the local regulations. In order to understand this definition, of course, you have to know what day 0 is. Day 0 is the date when any personnel of the MAH, the company, the marketing authorization holder, when any personnel, and this includes third parties acting on behalf of the MAH, when any personnel obtains sufficient information to determine that a case report fulfills the minimum criteria for reporting. So once you have all of those criteria in the definition of an ICSR, then that's when that reporting time clock starts. Okay, I want to take a minute to talk about the US FDA regulations. So throughout my talk, you'll see some slides that have a US flag and US map on them. And so on this slide, we're talking about US regulations. We're not talking about the guideline. And Meghna already presented some of these. I'll go over them again. For US application holders, an adverse event that's reported to the applicant by a consumer or a healthcare provider has to be reported as an expedited ICSR, so within 15 calendar days, if that adverse event is serious, and seriousness depends on the outcome of the adverse event, and those outcomes are defined in the regulation, and they're listed here on the slide. So it has to be reported in 15 days if that adverse event is serious and it's unexpected. And unexpected means unlabeled. It just doesn't appear in the label at the time that they're processing this adverse event. Other ICSRs, in other words, non-expedited adverse events, those adverse events that are non-serious, or adverse events that are serious but they're already labeled, those adverse events have to be submitted at the time that the applicant's next periodic safety report is due. So a PADER for drugs, periodic adverse drug experience report, or for biologics, a PAER, periodic adverse experience report. PADERs and PAERs are due quarterly for products that are less than 3 years old, or annually thereafter for older products. Okay, back to the guideline. So some other definitions that we've added to the guideline are added really to support guidance on managing some of these newer or emerging sources of data that are becoming more and more commonly encountered by MAHs. Digital platforms is defined in the guideline as software and technology used to enable transmission of information between users. So it's really broad. It includes lots of things. In fact, we list that includes social media, websites, internet forums, chat rooms, and software applications. This term digital platform that appears in the new guideline replaces the term and the section for internet in the original document. Organized Data Collection System, or ODCS, that also is presented in the original guideline, but it's presented just as a series of examples. Things like an observational study, a registry like the studies that Meghna was talking about, a clinical trial, a patient support program. These are all organized data collection system, all examples of organized data collection system. In the new guideline, we provide an explicit definition of ODCS, and the new definition includes a set of documentation, just some basic documentation that a company, that an MAH should have if they're going to do an organized data collection. Things like what are their objectives in doing this data collection? What's the data set that they're using? What's the time frame of the data that they're collecting? What's their methodology for reviewing that data? And how are they managing and processing any adverse events or adverse drug reactions that they encounter in that data? We also have definitions for patient support program and market research program. Those are new definitions in this version, and we'll talk about those in a moment. All four of these new definitions were areas of specific focus for the EWG to come together and revise the guidelines. So again, it's all about these new data sources that we wanted to address. Chapter 3, Section 3 is a new section in this guideline. This section includes the concepts of spontaneous and solicited reports. These are old concepts. They're not new. They've been in the guideline before, but they were listed as individual sources, as discrete sources of safety information, but they're really not sources in and of themselves. They're really meant to describe the context in which an MAH collects an adverse event, in which they became aware of that adverse event report. For instance, a spontaneous report is a direct communication by a healthcare provider or a consumer to an MAH, to a company or a regulatory authority. It could be directly to the regulatory authority or another organization. That describes an adverse event or an adverse drug reaction in a patient who is exposed, obviously, who's exposed to a medicinal product, and it was not gathered as part of an organized data collection. And that's the part that makes it spontaneous. An individual, whether that's the patient or their healthcare provider or some other consumer, is spontaneously on their own volition contacting the company or contacting the regulator and volunteering that information. So it's a spontaneous report. In contrast, solicited reports are any reports that are derived from an ODCS. So if you have an organized data collection like a patient support program, or you're doing a study and you're going out and gathering that information actively, and that's how you find out about an adverse event, then that is a solicited case, a study report. For the purposes of reporting, when you're transmitting an ICSR, when a company is transmitting an ICSR to a regulatory authority in E2B format, in that ICH electronic transmission format, solicited reports should be classified as reports from studies. So I'll kind of use those terms interchangeably, study reports and solicited reports. And in fact, to talk about FDA guidance for a minute, in FDA regulations and guidance, we don't talk about solicited, we use the term study. Solicited reports should have a causality assessment and should only be submitted, of course, if a causal relationship between a medicinal product and the adverse event is at least a reasonable possibility. Okay, the next section, maybe the most important section in the guideline, goes through the various sources of post-approval safety information that companies encounter where they gather their adverse event information. The literature section has extensive updates to clarify important topics, and we'll discuss those in a moment. We'll also go through the digital platform section, and then we'll talk about the sections on patient support programs and market research programs. There's also a section on regulatory authority sources. So many regulators and countries make adverse event data from their national or their regional pharmacovigilance database available to the public or to companies. Meghna talked about how you can go on the VAERS website and access the VAERS data or the Wonder website, and VAERS reports are fully available there. Many times this can include anonymized redacted data at the case level. The VAERS and the Wonder websites have just that. In fact, on those sites, they include narratives that have been redacted. So the REvIDE guideline provides guidance on how marketing authorization holders should handle cases if they go to a national, a a country's publicly available adverse event database, and they collect cases from there. It talks about how to handle those cases. Okay, updates to the section on literature. This section includes more harmonized guidance on screening of medical and scientific journals. It clarifies the start of the regulatory reporting time clock for cases identified in literature reports. Oftentimes, literature articles will identify the product that's involved in a patient's adverse event just by the name of the substance or the name of the active ingredient, and not include the actual product's trade name or brand name. And so the company won't know if it's actually their product if it's actually their product, especially if that product, there's other products of the same ingredient made by other manufacturers. So we clarify some expectations around the obligations for companies to report if the trade name is unknown or if it's ambiguous which product actually was involved in the event. The guideline also provides recommendations that marketing authorization holders should include important findings from literature in their periodic safety reports, even if those cases don't, or if that information doesn't qualify to be included in an individual case safety report in an ICSR to submit, which is what this guideline is about. If there are still important findings that the company finds in literature, they can be included in periodic safety reports. Okay, so we'll talk about US FDA requirements again on this slide. For US applicants must submit cases from literature that they find in scientific literature. In other words, these are cases that the applicant receives or otherwise obtains from a literature article. If that case is expedited, again, so it's serious and unexpected, and if the applicant finds if the case was found in scientific literature where the literature article is either a case report or it's the result of a formal clinical trial. So there's some distinction there in the regulation about the type of literature article for which reporting is required. So I'm going to go on to talk about the digital platform section. And as I said, this section is replacing the section on the internet. This section defines what is meant by digital platforms as a data source, and importantly, it makes a distinction for managing cases from digital platforms based on whether the marketing authorization holder, the company, has responsibility or ownership of that digital platform. And what we mean by responsibility or ownership is that the marketing authorization holder controls the content of the information on that website, on that chat forum. If they control the content, then they have responsibility for that site. As it was in the original guideline, companies have no obligation to go out and screen digital platforms for which they don't have ownership, for which they don't have responsibility. If they don't control the content, various social media sites, Facebook, whatever, they don't have to go out and screen social media or other digital platforms just to find adverse event, adverse drug reaction cases. There's no requirement to do that. However, if a digital platform is under the MAH's responsibility, so again, meaning that they control the content, then the MAH does have to regularly screen those sites to see if anyone has posted adverse event information, adverse drug reaction information about their products on those sites, and then they have to manage those cases as appropriate. Those cases could be spontaneous or solicited depending on the context in which the MAH got that information. So for instance, if a company has a product website and someone's able to post information about an adverse event that they had in their own experience on that website, well, obviously the company controls the content of that website. So that digital platform is under that MAH's responsibility. That MAH has to screen that website regularly, and they're responsible for identifying that adverse event report. Since that patient was voluntarily, spontaneously posting information about their case to the company's website, then that's a spontaneous report. On the other hand, if a company is using a digital platform to, say, conduct a registry study or conduct a patient support program, and they identify adverse event, adverse drug reaction cases that they've solicited through that study on their digital platform, then that case should be managed as solicited because of the context in which the company got the case. Well, MAHs aren't required to go out and screen external digital platforms, right? We said they have no obligation to do that if they don't control the content. This still often happens, and they encounter a large volume of information on these websites. And so the guideline gives some guidance on how MAHs might be able to manage, might be able to handle information on these sites. For one, they can screen data on digital platforms using an organized data collection system. So remember, we talked about a little basic documentation that you need to have if you're going to do an organized data collection. So the objectives, what are your methods for reviewing the data set, what exactly is the data set that you're going to collect, what's the time frame of that data, and how are you going to manage adverse events, adverse drug reactions that are identified in that data in the context of conducting your planned analysis. So if you use an ODCS and you go out on an external site, a digital platform for which the MAH does not have responsibility, you still have to appropriately manage and appropriately report adverse event, adverse drug reaction cases that you identify on that site. If you identify those cases in the context of conducting your planned analysis that's specified in your documentation. You don't have to go out and screen the rest of that data set or other parts of that digital platform site just for the purpose of finding other adverse event, adverse drug reaction cases beyond conducting your planned analysis as part of your organized data collection. The guideline also clarifies the time clock for regulatory reporting of cases identified on digital platform sites. And then perhaps most importantly, we propose in this draft to include a new value in E2B that would identify cases that were sourced from a digital platform using an organized data collection system. Currently, all solicited cases, all study reports are identified in E2B simply as report from study. And there isn't a way to tell the difference between which cases came from an observational study like a registry, which cases came from a patient support program, or which cases came from, for instance, social media websites. So these flags would allow you to tell, which cases came exactly from a digital platform. The next section that we'll talk about is the patient support program section. So the new guideline provides a definition for patient support program. This is a really widely used term. I think most companies that I've encountered have patient support programs. Some companies have dozens of patient support programs, even for a single product. So it's really widely used throughout industry, but it's not a well-standardized term. And companies consider different things, different kinds of programs to be patient support programs. So we provide a definition. What is a patient support program? These are company programs in which patients enroll for the purpose of supporting their use of that company's product. So they might get financial reimbursement from the company for receiving for taking the product. They might get educational assistance about their disease, their medical condition for which they need the product, or they might get education about the product itself. Or they might get assistance with adherence, compliance support, so that they can regularly take the product when they're supposed to. Or they might even get clinical assistance with administering the product, like in-home nursing support to help teach them how to use it or to actually administer it for them. So all of those different things can be part of a patient support program. All of these programs include a mechanism for two-way interaction between the company and the patient or the patient's healthcare provider. So you can imagine if a company is going to give financial reimbursement for a product, they want to be sure that the patient needs that product and that the provider is actually administering that product. So they're going to collect some medical records to verify that the patient has the conditions for which it's necessary to have their product before they give financial reimbursement. So there's that two-way interaction of information. They're getting medical information from the patient through those medical records. If a nurse goes to the patient's home to help deliver, help administer, or teach the patient how to administer a product, you can imagine certainly that in the course of that conversation, the nurse might learn things about that patient, about their medical history, or about adverse events that they're having. How are you doing today? Oh, I had a headache since the last dose when you were here last. So that, that represents an adverse event that then the nurse has to report to the company, and that becomes an ICSR. Importantly, so there's a few important concepts in this definition. One is that we consider all patient support programs to be ODCS, organized data collection. So I talked about how there's this mechanism of two-way interaction that's built into the nature of these programs. And so because of that two-way interaction, the cases are considered solicited. because there's already an interaction between the company and the patient's not really spontaneously reaching out to the company to provide this information. It's learned in the setting of that interaction. Part of the definition is that to be a patient support program, a program must include either collection of medical information, so I talked about those reimbursement programs, or it must be set up in such a way that collection of medical information, or that it's likely that the program will receive medical information. So a nurse going to a patient's home is an example where it's very likely that the nurse will learn medical information about the patient in that interaction. Another important aspect of the definition is that it excludes certain kinds of programs that are often called patient support programs by some companies. A program where they strictly do delivery service. So they're not providing financial reimbursement. They're not providing any sort of education. All they're doing is delivering it to the patient, just like Amazon or FedEx. So that's not a patient support program. That's just delivery. A program where patients can sign up on a website or something and get a voucher, a coupon, so that they can get a discount when they take their prescription to the pharmacy to get it filled. That's also not a patient support program. It's just a coupon that the company's offering. And they don't have, those kind of programs don't have the mechanism for two-way interaction. So as I said, these cases are all considered solicited because the adverse events are garnered in this in the context of this two-way interaction between the representative from the company and the patient or their healthcare provider. And again, just like digital platforms, we propose in this new draft that there's a new value in E2B for cases that specifically designate them as having come from a patient support program. And that would delineate them from other study type of reports. So we'll take a moment to talk about US FDA regulations again, not the guideline. So according to FDA guidance, applicants should manage patient support programs as study reports. So I said we use that word study in FDA jargon. According to FDA regulations, applicants must submit adverse events from post-marketing clinical studies. So we're saying manage it as study reports. So you follow the regulations for a study. If a case is expedited, so again, here's that serious and unexpected criteria, and the applicant concludes that there's a reasonable possibility that the product caused the adverse event. So cases that meet these criteria that come from a patient support program would have to be reported as an ICSR. The last source of data in post-market setting that's described in the guideline is market research programs. As with patient support programs, the guideline provides a definition, which you see here. And these are basically activities that companies conduct. They're planned collections of data where they're collecting consumer insights or healthcare professional insights, feedback, sentiments. Sometimes they're called sentiment analysis. And usually these collections are conducted for business or marketing purposes, not usually safety purposes. As with patient support programs, you should manage cases in these settings as solicited, again, because of the way the data is collected. And again, we propose a value to identify cases from market research programs. Chapter 5 discusses which ICSRs should be reported to regulatory authorities, especially with respect to seriousness and expectedness. So should all serious cases, only serious and unexpected, non-serious cases, what has to be reported and which case has to be reported as expedited events. The new guideline was updated to be harmonized. So it really refers to local and regional requirements to determine which cases exactly have to be submitted and which cases have to be submitted as expedited. This section also includes a note about important safety findings. These are findings which do not qualify for ICSR reporting, but which may lead to changes in the known risk-benefit balance or impact on public health. And these should be communicated as soon as possible to regulatory authorities if in accordance with local or regional requirements. And there's also a section on other observations. This section was expanded to include several scenarios and clarify the reporting obligations for each of those scenarios. So when we say other observations, we mean a group of different scenarios that don't represent adverse events in and of themselves, but they're different observations. Lack of effect, overdose, abuse, misuse, medication error, or occupational exposure, use of medical products in pregnancies, Meghna touched on this, and then off-label use. All of these things don't have to be reported in general, unless reporting for that specific event is required by local or regional requirements. So again, I'll take a minute to talk about FDA requirements regarding these scenarios. Off-label use, medication error, and use in pregnancy are all the same as recommended in the guideline. These are not part of the definition of an adverse event. So they're not adverse events, adverse drug reactions in and of themselves. So they don't have to be reported as ICSRs unless they occur along with an adverse event, unless the patient in the case who's you administer a product off-label and that patient has an adverse event, then it has to be reported. But if there's no adverse event and it's just one of these observations, it doesn't have to be reported to the FDA. However, regarding lack of effect, the definition of an adverse drug experience in FDA regulations, any adverse event associated with use of drug in humans, whether or not considered drug-related, including, among other things, failure of expected pharmacologic action. So if you do have a case of lack of effect, that does qualify for ICSR reporting for FDA. Chapter 6 talks about good case management, topics like patient and reporter identifiability, case evaluation and follow-up. We also add a section on duplicate management, which stresses the importance for marketing authorization holders and regulators to identify and manage duplicates when reporting and when managing their own databases. As a reminder, please refer to the complete document for all changes and updates. So this is an exciting time for the expert working group and for ICH regarding E2D. The guideline was approved in draft in February. So after 4 years, we finally finished a complete draft. That draft was published for public comment in the US Federal Register in March, mid-March. And that comment period has now closed, but the guideline is still published and open for comment on the ICH website. So any stakeholder can go to the ICH website and provide comments directly to ICH. It's also still open in several of the participating regions, most of the regions really, so stakeholders can provide comments to other regions still. The public consultation is expected to conclude this summer, and the expert working group will review and revise the guideline depending on comments. And we forecast a final guideline in May of 2025. So in summary, the revised guideline includes new and updated definitions, things like ICSR, Organized Data Collection System, and Patient Support Program. We clarify and we add new guidance for key information sources, especially these new or emerging sources that I talked about, like digital platforms and patient support programs. And then we update guidance on the other safety observations, things like lack of effect, off-label use, and those other scenarios. The draft is open for public comment, like I said, in several regions. So we'll do the two challenge questions with our last couple of minutes. The main reason for ICH to update E2D was to compare and contrast the use of large language models like ChatGPT, BERT, and Llama for post-market safety reporting. New information sources have emerged or are used more often, which vary in characteristics and contribution to post-market safety surveillance, things like patient support programs and social media. New participants, Health Canada and MHRA, have recently joined ICH. Or to align with related ICH post-market reporting guidelines, which have also recently been updated, ICH E2B R3 and ICH E2C R2. So anybody who, so if you said B, you'd be right, new information sources. An ICSR is an adverse event or other observation in an individual patient at a specific point in time. Which of these is not one of the 4 minimum criteria for an MEH to report an ICSR to a regulatory authority? At least one adverse event or adverse drug reaction, at least one suspect or interacting medicinal product, at least one serious and unexpected, that is unlabeled adverse event, an identifiable patient, and at least one identifiable reporter. Got it. So if you said C, you'd be right. It's not one of the 4 minimum criteria. Those are some of the criteria for reporting an ICSR as expedited under FDA regulations. But the 4 criteria for ICSR in the guideline are the other 4. Thank you.
Great, thank you very much for this deep dive into the modernization efforts for BCSR reporting. Thank you. Please don't go away. Stay at the podium. And Meghna, please come up to the podium. I would like to ask both presenters to be here at the stage, and we will start our Q&A panel. Any questions from the room? All right, well, how about online?
Here you go. Right. This first question is for Greg.
Okay.
Greg, how will FDA make stakeholders aware when the final version of the guidelines is available?
So ICH guidelines, when they're finalized, are adopted by FDA as an FDA guidance. So the guideline will be, essentially reformatted and then republished by FDA as an FDA guidance document. So it'll be announced in the Federal Register just like any other guidance document.
Thank you.
Okay. This question is for Mega. Are sponsors required to report pregnancy exposures not associated with clinical adverse events? What about under enhanced pharmacovigilance?
So as we went over, if there is no adverse event, if it's just a pregnancy exposure, they're not required to report it as an ICSR. However, under enhanced pharmacovigilance, we could ask for additional reporting. We can ask them for an assessment of all pregnancy exposures in the periodic safety reports, for example. So there could be differences from product to product based on our requirements for enhanced PV.
Thank you. It looks like we have some online questions.
This question is for Greg. Greg, will the guidance provided in ICH E2D supersede existing FDA rules or guidelines?
Yeah, so like I was discussing about having variations across regions and countries and having to account for that, one of the principles of ICH is that a guideline has to be consistent with the guidances and regulations in all the participating regions. So it won't supersede FDA guidelines. In other words, it won't contradict with existing FDA guidances or regulations. It'll be an FDA guidance when it's adopted as a guidance, but that everything in it should be harmonized and consistent with what's in other regulations and guidance.
Do we have more questions? Okay, great.
Yes, we do. This question is for Mega. Can pregnancy registry studies include retrospective reports?
So usually pregnancy registry studies, we think of them as prospective observational cohort studies, but we do have sponsors sometimes include retrospective reports. And, there's always bias with retrospective reports as well. So when that happens, we ask them to analyze them separately and, include that in their final study report.
Thank you. Craig, do you have anything to add?
No, that's
all right. Okay, great. Are there more? Okay, why don't we go ahead and...
This question is for questions. How frequently are sponsors required to search literature?
Okay, so I kind of alluded to this in my talk. Under US FDA regulations, there's actually no requirement that sponsors search the literature at all. The guideline doesn't give a frequency, and part of the reason that we don't give a frequency is because in some regions like the US, there's no requirement to search the literature. The guideline does say that we encourage that MAHs search the literature, but we leave it to local or regional regulations to determine, specifically what frequency.
Thank you. Thank you.
All right. Yes, we do.
Okay.
We have a question from Mega. What should the sponsor do for pregnancy registry post-marketing commitment, PMC, that has a low enrollment and is not able to reach target enrollment?
This is a very, very common question.
Very common, yes. Very common scenario. So very good question. So I mentioned, we have many strengths of pregnancy registries. We can collect patient level data in a prospective manner, but sometimes they run into difficulties with reaching their target enrollment. And it just becomes infeasible. The study just becomes infeasible. So in those scenarios, we ask that the sponsors for PMCs, and this applies to PMRs also to an extent, to communicate early with us. They have two options for PMCs. They could either ask for a release from the PMC, and we would consider that request, again, depending on the data and the justification they provide. Or they could ask for a release and replacement of the PMC. So if it was a pregnancy registry, they could come up with an alternate study design. Maybe they feel it's feasible to do a database study, and we could release and replace the pregnancy registry with a database study as well.
Thank you. These are good real-life options.
I just wanted to add a couple of things. Usually that happens after the product's been out for some time, because the sponsor's been trying, hopefully, had a good faith effort to do the study and to get the enrollment that they need to get. So sometimes there's other data available at this point in time that there wasn't at the time of approval. So some of the things that we consider when we make that decision about, should this be released, should there be a different study, we can look at VAERS data, and what other post-market data, or maybe other studies have been done, or maybe the sponsor has other INDs underway for other indications. So we can look at all the available data at that point in time, which might be better than the case at approval.
Thank you. Any more questions?
Yes, we do. This next question is for Greg. Greg, how does the guidance on handling PSP reports differ from current FDA requirements for these reports?
So it doesn't really differ. Like I said, all the guidance in an ICH guideline has to be consistent with FDA requirements. I think what's new is, first of all, is that it'll be a new guidance, and the guidance, the only place that it really mentions how to handle a patient support program is an old 2001 FDA draft guidance. So it's not really well addressed in FDA regulations and guidance. So, for one, it'll be a new and recent advice about how to manage patient support programs. They'll still be managed as study reports, so it's not changing. And the sponsor still has to collect anything that they receive or otherwise obtain through a patient support program. Still has to be appropriately managed and reported as an ICSR if it meets the requirements for reporting an adverse event case. So those rules aren't changing. The other thing that's new is the definition. So I talked about how we have this definition of what's included in a patient support program and a couple of things that are excluded. And that's actually a big change. What we learned from industry that's participating in the expert working group is that the exclusion of delivery services, the exclusion of coupon card services is actually a big change for them. So it could somewhat change the scope of what's considered a patient support program.
Thank you. That's actually very helpful to know, I think. Any other questions?
Yes, we have one for Meghna.
Great.
What should a sponsor do for a pregnancy study post-marketing requirement, PMR, that has been delayed?
So again, some of the options are similar to what you would do for a PMC. You would provide justification for the delay. Now for a PMR, they also have to submit a good cause request for a delayed milestone. And they have to lay down their rationale for that good cause. It has to be something that was not under their control. So again, if they were not able to reach target enrollment, they have to show us, that they made good faith efforts, they tried, but it was not feasible. So that is another aspect of PMRs, to submit that good cause request.
Thank you, Meghna. I think we're almost at time. There's less than 1 minute left. I would like to thank both of our presenters. Thank you so much for giving the informative talks and for answering questions. Let's give them a round of applause. And we will be back in this room at 1 o'clock after lunch for another session about blood products. So thank you.
afternoon session. This afternoon, we'll have talks from the Office of Blood Research and Review. And just a reminder that we'll have 2 speakers for this session. And if you have any questions, please jot them down, or if you're online, just fill out the questionnaire form, and we'll have a Q&A session at the end of the time that both speakers have given their presentations. So for our first speaker this afternoon, it is my pleasure to introduce Dr. Orieji Illoh. Dr. Illoh joined the FDA in 2009, and currently she's the director in the Division of Blood Components and Devices in the Office of Blood Research and Review. She oversees regulatory activities related to blood and blood components for transfusion, plasma for fractionation, blood collection, containers and storage solutions, and devices used in the manufacture of blood and blood components, plasma volume expanders, and oxygen carrying solutions. Prior to joining the FDA, Dr. Illoh practiced clinical pathology and transfusion medicine at the University of Virginia, Charlottesville, and then at the University of Texas Medical School in Houston. So it's with great pleasure that we invite her for her talk today, and we look forward to hearing your presentation. Thank you.
All right, thank you for the introduction. So as was mentioned, my name is Dr. Orieji Illoh. Happy to be here today. Welcome to our on-site participants as well as our online participants. So we're really grateful to the organizers for having us to give you a snapshot of what the Office of Blood Research and Review does. So I will be speaking first and followed by my colleague, Dr. Lathrop. So this works. So my topic today will be about blood regulation and safety. So hopefully the learning objectives today will be to describe briefly the functions of our Office of Blood Research and Review, OBRR, and give you an example of some of the products that we review. An understanding of our approach to the regulation of blood and blood components and donor safety, blood donor safety, and then briefly describe our process for obtaining blood licensure. So first of all, I'll talk about our functions and products that we review. So first of all, I just want to give you a quick overview of the structure of our office. Our office is led by Dr. Ann Eder, and I forgot to mention that in the next few weeks, I'll be transitioning to the Deputy Office Director position. And then we also have 2 main divisions, the Division of Blood Components and Devices, which is where I am now, and also the Division of Emerging and Transfusion Transmitted Diseases, where Dr. Lathrop resides also. So within these 2 divisions, there are several branches and also lab research groups that do a lot of mission-related research as well. So our functions include, and our core functions really include regulatory review of different devices, blood components, so on and so forth. But of course, we do regulatory review of blood and blood components, several biologic products such as albumin and hemoglobin-based oxygen carriers, and then a lot of devices related and drugs related to blood manufacturing. Our office also conducts mission-related research in different topics relating to emerging infectious diseases, and also looking at different topics relating to things like pathogen reduction of blood components, storage of blood components, looking at oxygen-based carriers, and many other questions that are relevant to our regulatory work. We are also involved in a lot of policy related to blood, and also we are involved in drafting regulations and guidance documents. And as appropriate, we also develop safety communications that we share with the public relating to any issues that we want people to be aware of. We also are involved in inspections and a few compliance activities together with our sister office, the Office of Compliance, including things like reviewing fatality reports related to donations or transfusions. We also conduct inspections of blood establishments, and I'll expand on that a little bit more later. So this slide just gives an example of some of the products that we review. Of course, I mentioned that we review blood and blood components for transfusion or further manufacture. And what we really do in this aspect is really working with the different manufacturers or blood establishments or blood collectors. So the American Red Cross, you might have heard of that, Vitalant, or in this area, Nova, all that kind of, those kind of places. We work with them to ensure that they're manufacturing blood in accordance with the regulations. We also oversee devices used in the manufacture of blood and blood components, and this is a variety of things, including collection equipment like apheresis equipment, or other devices, blood bags that are used for blood collection. We also oversee blood collection containers and additive solutions, pathogen reduction devices for blood, and blood donor screening and supplemental tests for relative transfusion transmitted infections. That's what RTTI means, such as HIV, HBV, and HCV. We also, and I think Julia, Dr. Lathrop might expand on this, also review a couple of retroviral assays that are also used for diagnostic use. In addition, we also review immunohematology reagents and compatibility tests that are used for blood banking. So I'll hone in more on blood products because that's where my talk will focus on. So when we talk about blood products regulated by OBRR, we're talking about blood and blood components that are collected and processed for transfusion, or source plasma, which is used for further manufacturing. And further manufacturing means that these are typically used and developed into derivatives, for example, such as IVIG, albumin, and products like that that are plasma derived. This source plasma can also be used for in vitro diagnostic tests. So for source plasma, they come in different forms, but mainly most collectors collect normal source plasma, which is just from normal qualified donors, or they may collect what we call disease-associated or disease state source plasma. Disease-associated means a donor has recovered from a particular disease and they have an antibody of interest that can be used either for therapeutic purposes or for in vitro diagnostic use. Disease state means it's a donor who has a particular disease, but their plasma is of interest maybe to use as controls in the device. And so we also oversee that because it's a more detailed program, and we have to make sure that the firms doing this are doing this in a way to protect the health of the donor as well. We also oversee the collection of source plasma from donors who are immunized to produce a particular protein, usually an antibody of interest, that can be further manufactured into a derivative. So just to give you some context about our the use of blood and blood components in the U.S. I'm sharing here the NBC US survey results from 2019. And you can see that in the U.S., about 14 million blood donations for transfusion occurred, and about 15 million blood components were distributed and transfused. So blood, especially whole blood, can be further processed to make cryoprecipitate, plasma, platelets. So that explains the 15 million compared to the 14 million donations there. So you can tell from the graph here that there was a peak in blood collections and donations that has kind of tapered over the last few years. And so it continues to remain that way and probably has plateaued so far as we can tell. So the point here is that there are lots of blood donations that occur. There are a lot of blood that is processed for use in patients. So hence the importance of maintaining a safe blood supply and also making sure blood is available for our patients. In terms of source plasma, we continue to see an increased demand for source plasma for use in the manufacture of derivatives. And from this graph here, which is obtained from the PPTA website, you can see a continuous increase in the amount of source plasma collected. In 2019, there were about 53 million collections. And you can see the numbers here that the centers that are opening are licensed for source plasma collections continue to increase. Of course, in 2020 and 2021, there was a slight dip in the collections because of the pandemic. But we're continuing to see an increased amount of source plasma centers opening to meet the demand for source plasma. So I'll stop here at this point and ask our audience to participate here. I have my first challenge question. And so my question is that OBR regulates all the following products except, and I'll list them. A is devices for HIV testing, blood and blood components and source plasma, intravenous immunoglobulin preparations, and D, immunohematology testing devices. So which one do we not regulate? And I'll give our colleagues online also a moment to think about this. Anybody vote for A, B, C, D? Okay, so the answer here is C. We do oversee blood components and source plasma that is used for the manufacturer of derivatives, but the derivative itself, like such as IVIG, is regulated by our sister office, the Office of Therapeutic Products, OTP. All right. So my next section of this talk will be about FDA's approach to the regulation of blood and blood components for transfusion and further manufacture. So first of all, I'll talk about the laws and regulations that are used for oversight of these products. So under the Public Health Service Act, the PHS Act, blood is defined as a biologic, and therefore a license is required for distribution in interstate commerce. Biologic products must be manufactured in a manner to ensure the safety, purity, and potency of the product. And then in addition, blood is subject to regulations to prevent communicable diseases, which is section 361, of the Act. And then what is unique to blood is that blood is also defined as a drug under the Federal Food, Drug, and Cosmetic Act. So therefore, blood is a drug because blood components are intended to cure, mitigate, treat, or prevent diseases in humans. And under this act, blood manufacturers are subject to current good manufacturing practice regulations, prohibitions on adulteration and misbranding of products. So we expect blood components to be labeled in according to the regulations, and if it's not, we consider that as misbranding. An inspection of manufacturing facilities. So my next two slides will just give an overview of really the regulations related to blood and blood components. And this is found in 21 CFR in the 600s of the CFR. And so blood and blood components are subject to Part 600, which has general requirements for biologics. 601 that also has general requirements for licensing biologics and also reporting manufacturing changes. And I'll expand on that later. And then Part 606 is pretty detailed in terms of current good manufacturing practice, including things like labeling, bacterial risk control for platelets, and fatality reporting. And we have requirements for fatality reporting if a transfusion is found to be a cause of death in a patient, or if a donation caused a death in a donor. That has to be reported to FDA. We also require blood establishments that manufacture blood and blood components, including source plasma, to register in our database and list all the products that they manufacture. And this is something that is supposed to be updated every year. Under 610, there's general biologic product standards, but specifically there are areas that also address testing of blood components for relevant transfusion-transmitted diseases. That section also addresses dating periods for blood components and expiration dates. Under Part 630, that details the requirements for donor eligibility and donation suitability, so what to do to screen donors for blood donation. And then Part 640 gives additional standards to required for specific blood components such as red blood cells, platelets, plasma, and source plasma. So you will note if you look at these regulations that, we're not just focusing on collecting a safe product for our patients. We also want to make sure our donors are safe. And so we have specific regulations to protect the health of the blood donors too. And these are typically found in CFR 630.10 and CFR 630.15. So examples of some of these regulations that we have to protect donor health include donor questioning for medical conditions and medications that they may be taking. And donors are typically deferred if we feel that if there are conditions where that might affect their health. We also have requirements for physical examination, and this includes taking a blood pressure, pulse, temperature before collecting blood from the patients, from the donors, and also a requirement for hemoglobin measurement. And we've also established donation frequency, because frequent donations can lead to complications such as iron deficiency in our blood, in our donors. So we want to do the best that we can to mitigate that. We also have requirements for the source plasma collections, especially for total protein measurement and serum protein electrophoresis periodically on these donors, just to make sure that they remain healthy. And so in addition to our regulations, which are binding, we also have guidances that we put out to industry. And the first bullet here really just gives you the link to those guidances, blood guidances that you can look at. So generally, guidance documents explain FDA's current thinking on the regulatory issue. And we may use that to clarify or explain how manufacturers may comply with the statutes or regulations. Or we may also explain how and establish good manufacturing standards for blood products. So we have lots of guidances that we put out there, whether it's related to testing, how to handle the results, whether further testing is needed, how to dispose the units of blood that may be collected from donors, and labeling guidances, and many others to address the manufacture of blood and blood components. Okay, so I've talked about the basically the legal framework for blood and blood components. And now I'm going to talk about our blood licensure requirements and the review process. So first of all, I want to introduce the branch that does a lot of this work. This is the blood and plasma branch, which is under the Division of Blood Components and Devices. This group consists of about 15 staff, including the branch chief and team leads, who all have expertise in the area of transfusion and blood collection. And so they oversee blood establishment registration. They review applications for the manufacture of blood components intended for transfusion or for further manufacturing. They conduct inspections. They assist with development of blood policy. And a lot of their work also includes advising blood establishments on regulatory issues. So this slide just kind of gives you an idea of what we have right now in terms of registered and licensed blood establishments as of April 2024. You can see that there are about 765 registered only blood establishments. So this includes hospital blood banks, so hospitals that collect blood for their use, and also transfusion services. They don't necessarily collect blood, but they do compatibility testing, storing of blood products, and issuing of blood products to patients. So if they do things like washing red cells or irridating red cells, they're considered as manufacturers, and so they have to register with FDA. And then we have about 948 blood collection establishments and 1,152 source plasma establishments, which I'm sure has gone up as of today. We constantly are inspecting and licensing more and more source plasma establishments. And so the biologic license application, or BLA process, under 601, the regulations, blood establishments that want to get licensed are supposed to submit a BLA application. And so this application includes a cover letter, the Form 356H, a list of the blood components that will be manufactured, a description of the manufacturing process, including submission of written SOPs. And so typically there are a lot of SOPs that are submitted with the application. And then as applicable, validation data summary, and also quality control data should be submitted. In addition, we typically also review the blood component labels to make sure they're compliant with our requirements and the regulations. The regulations have really specific information about what the label should look like when it's submitted or used for blood components. So this list is not an all-inclusive list, but these are some of the things that should come in the BLA application. We typically advise manufacturers, especially if they're new to this process, to contact FDA, maybe request a meeting with us, or just send an inquiry asking about what they should include in their BLA application. That way, there's less likely a chance that the application would be incomplete and prolong the whole review process. So those who are already licensed under the regulations 60112 must submit changes to their manufacturing process in an approved BLA. So how they categorize the change depends on the degree of the change, the degree of the complication of the manufacturing change. And so if it's a major change, that comes in as a prior approval supplement or PAS. If it's a moderate change, it comes in either as a changes being affected in 30-day supplement, a CBE 30, or a CBE, which means that the change can be implemented immediately while FDA is reviewing the supplement. And then minor changes come in an annual report. So there are a lot of things that blood establishments do in terms of manufacturing. And so what we have out there is an FDA guidance, and I put the link down on the slides here, that kind of assist blood establishments on understanding where their changes fall into, which category it falls into, and how they should submit it. So when we get either a BLA supplement or an original BLA application, the first thing we do is to make a decision about whether the application can be filed. And then we issue a filing letter if it can be filed. This has to occur within 60 days of receipt of the application. And if we find the application to be incomplete, we will send the manufacturer a refuse to file letter. The refuse to file letter will typically explain to the manufacturer why we're not able to file the submission. So from my experience for BLA supplements and BLA applications related to blood and blood components, typical issues that we find when we do refuse to file, which is not very common, is either we did not receive sufficient SOPs, so incomplete SOPs were submitted, or nothing was submitted at all, or applicable data such as QC data were not submitted. Okay, so if the application can be filed, the review continues. The review typically is very interactive with the sponsor. And so along the way, the reviewer may send several questions or what we call information requests to the sponsor, clarifying things maybe about their SOP, their labels, so on and so forth. So once the review is complete and found to be acceptable, the reviewer will draft a memo. The Office of Compliance will also conduct a compliance check to make sure that that facility has an acceptable compliance history. And if everything's acceptable, we issue an approval letter. If there are deficiencies in the submission and are unable to resolve them through an information request, for example, we will issue a complete response letter, which will stop the review clock. The letter will include a description of the deficiencies in the submission and what we need to address those deficiencies. And typically sponsors are given about a year to respond to complete response letters. And if they fail to respond to their complete response letter or withdraw their application, FDA will proceed with withdrawing the application. So typically we'll issue a pre-withdrawal letter first to the sponsor. And if we don't hear anything, we'll proceed with withdrawing the application. So in this slide here, I just give an outline of the typical timelines for our common submissions. I want to highlight that blood and blood components are not user fee products. So unlike user fee products where they're subject to specific timelines, these are not user fee products. That being said, we do adhere to timelines similar to our user user fee products. And so for original BLAs that come from new manufacturers, which is really a detailed submission reviewing tons of SOPs, going back and forth with this manufacturer and conducting an inspection, typical timeline is 12 months. For BLA supplements, and obviously that depends on the category of the supplements, for a PAS, it's about 12 months. Our target usually is 6 to 9 months. A lot of these supplements, or a couple of them, will also include inspections, hence the long timeline, especially if people are opening new centers under their license. So PASs involve review of major changes. And then for the review of moderate changes, the CBE 30s and the CBE, that takes about 6 months. So in this slide here, I just give you an overview of the applications reviewed by the Blood and Plasma branch in 2023. So you can see that we have very few original BLAs. So they look few, but they're very detailed when we do get them for review. Like I mentioned earlier, that also entails inspections and detailed review of the SOPs, since the manufacturer is getting into the licensed product manufacturing business for the first time. So last year we had 5. Most of those were for new source plasma centers. And then we had 146 PAS submissions. A lot of them also were for new source plasma centers. And if you, only new source plasma centers. And then you can see the number of CBE 30s and CBEs, couple of annual reports. And then the last line here highlights product correspondences. And these are basically communications from firms informing us of changes that may not necessarily fall into any of these categories, such as we're closing our facility or we're selling our facility to another facility. We still have to process those and send letters occasionally to address those. Okay, so I'll go a little bit into the inspections and what we do. So our goal when we conduct inspections is to determine that products are manufactured in compliance with the regulations. So for the original BLA submissions, we will conduct a pre-license inspection. And for the BLA supplements that include a new facility, we will conduct a pre-approval inspection. So for example, if you're a licensed source plasma center and you're opening a new center, we will need to conduct a pre-approval inspection in addition to reviewing your package. So blood establishment pre-approval and pre-license inspections are conducted by our staff in the Blood and Plasma branch. They are mainly domestic inspections within the US, and typically the inspections take anywhere between 2 to 3 days. So in this slide here, I'm just outlining the number of inspections that were conducted by the Blood and Plasma branch between 2020 to 2023. The numbers continue to increase, and you'll note that in 2020 and 2021, of course, we were in the pandemic. So a lot of these inspections were done as record reviews in lieu of actual on-site inspection. But from mid-2022 to date, we've gone back to on-site inspection. Most of these inspections are of new source plasma collection facilities. However, we're beginning to see an uptick in the number of blood collection facilities, so blood collection for transfusion as well. So inspection decisions, if something is observed that is objectionable during the inspection, the inspector may decide to issue a Form 483 listing the objectionable conditions. Typically, firms will work on addressing those situations and communicating with us what they've done to address those conditions. And then, of course, final inspection actions are determined by our Office of Compliance. And it's either there's a no action indicated, which is perfect, an NAI, or there's a voluntary action indicated where there were objectionable conditions identified, but the agency may decide not to take administrative or regulatory action, those are the VAIs, or they may decide to take official action, OAI, when there are objectionable conditions identified and they decide that they need to take administrative and regulatory action. So note that inspection findings are considered when making regulatory decisions on applications. So if we have an application under review, maybe for a new center, and the place is inspected and a 483 was issued, we will take that issue into consideration when making a final decision on approval or not approval of an application. So typically firms will communicate with us their plans on addressing the 483 issues that were identified. So in summary, I hope I've been able to share with you OBR functions, which include efforts to maintain the safety and availability of blood products for transfusion and for further manufacturing. Blood and blood components, including source plasma, are subject to both the PHS and FD&C Act. And manufacturers must obtain a biologic license before placing a product in interstate commerce. And our BLA application includes the review of SOPs and related material, QC data, and inspections. So in this slide, I've put a little bit of contact information here that you can use. For active regulatory submissions or something that you want to submit to our office, we suggest contacting our Director of the Regulatory Project Management Staff, Cherry Germanimo. For any inquiries related to blood manufacturing, we do have a mailbox that our staff in the blood and plasma branch respond to. And I put we call it the BPB inquiries mailbox. I put the address there. And then if you're unsure where to send your questions to, you can always contact CBER. And I put 2 contact informations in there. And they will share the information with the appropriate group to address. So I have my last challenge question. So which of the following statements is not true? So the blood licensure project process for blood establishments may include inspections to ensure compliance with the regulations. Applicants found to have submitted incomplete applications are issued a refuse to file letter, identifying the reasons for preventing a full review. Failure to address the deficiencies in a complete response letter may lead to withdrawal of the application. And finally, licensed blood establishments do not need to report changes in manufacturing to FDA. Go ahead. C? Yes. So C is the answer. It is not true. Even though an establishment is licensed, any changes in manufacturing must be reported to FDA under 21 CFR 601-12. So thank you. And go to our next speaker. I think we'll have a Q&A session after this.
Thank you so much for that great presentation. Very informative about the Office of Blood Products. Our next speaker is Dr. Julia Tait Lathrop, and she is the Associate Deputy Director of Emerging and Transfusion Transmitted Diseases in CBER. She's responsible for regulating retroviral diagnostics, including all HIV devices, as well as devices for screening blood donors for infectious disease. And in her current position, she's responsible for developing and implementing regulatory and review policies for the division. She joined the FDA in 2012 and has held roles for the Center for Devices and Radiological Health, Office of In Vitro Diagnostics and Radiological Health, and as a scientific reviewer and acting chief of immunology and flow cytometry branch in the Division of Immunology and Hematology hematology devices, until 2016. She holds a PhD in biology from the University of Virginia. So thank you so much, Dr. Lathrop, for coming out and giving us your presentation today. We look forward to hearing.
Well, good afternoon, and thank you for the invitation to speak. As I was introduced, I'm Julia Lathrop from the Division of Emerging and Transfusion Transmitted Diseases in the Office of Blood in CBER, where, to repeat my bio, we have the responsibility for regulating devices that are used to test blood for evidence of transmission or transfusion transmitted infections. So that's HIV, HCV, syphilis, babesia, as part of ensuring the safety and quality of blood, which is a biologic. We also have the responsibility for regulating devices used for retroviral testing. So those are tests for HTLV1 and 2 and all tests for HIV. Other infectious disease tests are regulated by the Center for Devices, or CDRH. But even though we're in two different centers, we're bounded by the same statutes and regulations and guidances. So we communicate quite frequently, and many of us worked in CDRH. And we try to align our approaches and policies as much as possible between the centers, given the different intended uses of our devices. But fundamentally, all blood donor screening tests and all HIV tests are regulated by CBER. So hopefully after this session, we'll know something about the history of testing for HIV infection, how FDA evaluates in vitro diagnostics, reclassification, what is it and what does it mean, what is the current status of HIV IVDs, and how FDA is looking to expand testing for HIV. So first, we'll cover the history of HIV testing. So in this timeline, you can see that the first AIDS case, what came to be known as AIDS, were reported in the MMWR in 1981. And because they were reported from people in the gay community, the condition had a number of different names: gay cancer, gay pneumonia, gay-related immunodeficiency. And it wasn't clear for some time as what was actually causing this condition, if it was a virus, if it were lifestyle factors, and how it was being transmitted. But about a year later, the first reports of individuals with hemophilia who contracted what came to be known as AIDS from contaminated coagulation factors appeared in the MMWR. And it became quite clear that coagulation factors, which are produced from pools, as Dr. Illoh noted, pools from hundreds to thousands of individual blood donations, and then the coagulation factors for plasma-derived factors are purified from those, that the coagulation factors were contaminated with the causative agent of AIDS. But it still wasn't quite clear. But near the end of 1982, the first reports of transfusion transmission of AIDS were reported. And in this case, it was an infant who had no other risk factors other than a blood transfusion. So it became quite clear that the causative agent was present in blood and could be transmitted by blood transfusion, and that the blood supply and the coagulation factor supply were contaminated with the infectious agent. So HIV was discovered in 1984, and just under a year after that, the first donor screening test for screening blood for presence of HIV was licensed by CBER. The first diagnostic test wasn't approved for a couple of years after that, and you might ask yourself, why was donor screening first when there were so many people who needed to be diagnosed? And the reason for that is because of the slope of this line, which you can see prior to 1984, first there was nothing. Then the risk of having an infectious unit of blood was 1 in 100 in some parts of the country. And so 1 in 100 units of blood were contaminated with HIV. And this is in the context of millions of units of blood transfused every year. So it wasn't a situation of being concerned about possible contamination of the blood supply. The blood supply was contaminated, and coagulation factors were contaminated. Most lots of factor VIII were contaminated with HIV. And part of the reason that blood is so infectious is it takes, one, very few virions to transmit an HIV infection, but two, the sheer volume of blood that's transfused into a person is, massive compared to other incidental ways of transmitting. So the risk of infection was incredibly high. So the blood establishments, before the virus was even identified, started deferring donors from populations that appeared to be at risk of carrying the infection. So that was, of course, men who have sex with men, people who inject drugs. Other populations at the time were deferred for life from donating blood. At the same time, there are also some establishments that tested for hepatitis B, because most people who were infected with HIV were also infected with hepatitis B. So it started in blood transfusion, which is why CBER regulates those testing devices. But HIV IVDs detect different markers of infection, which appear at different times following infection. So at first, after infection, there's a few days called the eclipse period, where the infection is present and blood is infectious, but it can't be detected by any test. Fortunately, that's just a very small number of days. But then different markers appear at different times following infection. The RNA comes first. So there are nucleic acid tests. The P24 antigen appears. It later gets complexed by antibodies. And then we have IgM and anti-HIV IgG antibodies. So there are a number of different IVDs that look for different markers of infection. So from the early days when blood was contaminated, lots of people were being infected with HIV. To the present, well, to 2017, which is now a few years ago, we had authorized a large number of HIV IVDs. First, there were the donor screening, and then came different types of diagnostic tests. So we had rapid tests, viral load monitoring tests, point of care, home tests. And at the same time, HIV treatment in a public health triumph has changed what remains an almost invariably fatal disease to one that can be treated, and people who are treated have a near normal lifespan. But this is all dependent on them being correctly diagnosed. So you can see the paramount importance of the quality of HIV diagnostic tests to make sure people who are infected can get the life-sustaining treatment that they need. But so treatment of HIV advanced, and the one piece of evidence of that is that donor deferrals went from lifetime deferrals for some populations to now individual deferrals, and no particular populations are deferred from donating. So in 2017, having 30 years of experience with these devices and knowing where they fell apart and what worked and what didn't work, we started to think that perhaps it was time to consider reclassifying them, because leaving aside donor screening tests, which are BLAs, and we're not touching those, diagnostic tests were all class 3 devices and require submission of a PMA. And why that's important and how we think about it and why we want to reclassify, I want to go through and talk about how FDA thinks about IVD review fundamentally and device classification. So for people with experience with FDA, this is all going to be very redundant. It's all stuff you know. But there may be some people out there who may not be quite as experienced. So this is a very fundamental, what do we think about IVDs? First of all, in vitro diagnostics includes all tests, not just tests for diagnosis. So tests for screening, monitoring, prognosis, they all fall under the umbrella of an IVD. And they are defined in the Food, Drug, and Cosmetic Act as instruments, reagents, and systems intended for use in the diagnosis of disease or other conditions to cure, mitigate, treat, or prevent disease, and in the collection, preparation, examination of specimens taken from the human body. And this will be evident as what that means and the implications of that in a few slides. So review of all IVDs is based on the balance of the benefit of the test versus the risk of the test. But there are very few IVDs where actually running the test is risky to the individual. Usually risk is considered the risk of a wrong result to the patient. What happens if you get a false positive or a false negative result? So we look at the balance of risk, of benefit versus risk, and then the data has to provide a reasonable assurance of safety and effectiveness of the device. Safety, that it's not going to hurt anybody, what are the risks from a false positive or false negative, but also effectiveness. You have to generate information that you can do something with. The result has to mean something that's actionable. A test that gives you a number that you can't do anything with, that doesn't mean anything, is not an effective test. So the device has to be both safe and effective. So this is all, so how do we decide how to think about those? The intended use of the device determines the benefits and the risks. So the intended use is what it's intended to do, but all the nuances of an intended use can really have a big effect on the risks associated with the device. So for example, is the test intended for self-testing, home test, or is it going to be run by trained professionals in a laboratory? Is it for antibodies to HIV? Is it antigen? Why are you running the test? Is it diagnosis? If a person goes for a diagnosis, they have signs and symptoms of a disease. Something is wrong. They go to the doctor, something's wrong with me, let's figure out what it is. They're under the care of someone who's going to keep looking if they can't figure out, hopefully, keep looking to see what is actually going on. Versus a screening test, no signs, no symptoms. You go in, you get tested, you get the answer. If it's negative, you're good to go. The implication of the wrong result in that situation is very different from someone who is already under care of a healthcare provider. What's the indication you're testing for? Is it HIV infection? Is it flu? Is it syphilis? Is it cancer? All of those have different risks. And who are you testing? Is it newborns? Is it pregnant women? Is it everybody? All of these different components of an intended use statement have meaning when it comes to interpreting what are the risks and the benefits of this particular device. And so this is an example of a generic intended use statement for an HIV diagnostic test. And you can see it, I've bolded some of the elements that go into our consideration of what are the risks and benefits of this test. So when I say we look at the benefits and risks of a test, it's the benefits and risks of this device. You sometimes hear people say things like, well, they're all class 2 devices, they're all moderate risk, it should all be the same thing. But in fact, the classification of a device only has some meaning in terms of what are the risks and benefits of the device. So for example, for HIV testing, we think about what are the benefits of a true negative. You've been tested, you're reassured, the individual may want to consider if they have risk factors in their life they may want to mitigate. A true positive. Nobody wants a positive HIV test, but you can immediately start life-saving treatment, entry into care, being taken care of and being followed, and have access to the very effective drugs. A false positive, unneeded treatment. The drugs may have few side effects, but any drug that's used when it's not needed has unneeded side effects. There's a lot of stress. If you're a woman in labor who gets a diagnosis of HIV while she's in labor, that's a pretty stressful event. So the implications of false positive can be quite serious. But of course, in HIV testing, we worry a lot about false negatives. You got denied needed treatment. They may have no further testing. The individual went, they got tested, they got a negative result. They may inadvertently transmit the infection to their partners. So the impact of the impact of different risk factors can vary depending on the type of test that's actually being evaluated. So IVD device, there are 3 classes of IVD devices, class 1, class 2, and class 3. And how we decide what class a device falls into is based on the risks of the patient and how or if those risks can be mitigated. So first, we look at the intended use statement and we ask ourselves, can general controls alone, are they sufficient to provide a reasonable assurance of safety and effectiveness? These are provisions in the Act that are basic, they're generic, they apply to almost all devices, and they include things like labeling. You have to say what the test is for. Manufacturers have to have quality systems in place. Prohibitions against adulteration, misbranding. You can't say your test does things it doesn't do. You have to report adverse events. They apply to pretty much devices across the board. They're not specific. If you have a tongue depressor, which is a medical device, general controls are sufficient to mitigate the risks for that device. Special controls are device type specific controls that are in the regulations that, along with general controls, provide the reasonable assurance of safety and effectiveness. And if you read the regulation about special controls, it's pretty non-specific. It doesn't define what they are. And that's because they are device type specific. They are necessary to provide this reasonable assurance of safety and effectiveness, and they're going to vary depending on the device type. But they can include things like performance standards, patient registries, complaint reporting. So we look at the intended use and we ask ourselves, are general controls alone sufficient? The answer to that is yes. It's a class 1 device. Then we say, do we need to actually see the data, or can they just go ahead and market it without us looking at it? In which case, if the answer to that is yes, the device is exempt. That means it's exempt from pre-market submission, and they can go ahead and market it. If the answer to that is no, then it requires a 510K. The majority of class 1 devices are exempt. Note that they still have to meet the general controls, and there has to be evidence that these general controls are met. It doesn't mean you're not regulated. It doesn't mean that at all. It just means you're exempt from pre-market review. But if the answer is no, general controls are not alone or not sufficient, then we ask ourselves, do we have enough experience with this device type? Can special controls be established to provide the assurance? And the answer, do we know enough about it to mitigate the risks associated with the device? If the answer to that is yes, it's class 2. Then we ask ourselves, do we need to see the data for this? If the answer to that is no, we don't, it's also exempt and can be legally marketed. In this case, though, if it's exempt, it has to meet general and special controls, if there are any, whether or not it actually comes in for review. This device has not been cleared or approved, it's exempt, but the evidence has to be available upon inspection that the special controls have been met. If it's not exempt, and most class 2 devices are not, that requires submission of a 510K. But if the device has such risks that we don't know how to mitigate them with special controls, or we've never seen a device like that before, then it's a class 3 device and requires submission of a PMA. So just to summarize, class 1 device, they're generally very low risk. Most of them are exempt from pre-market submission. Class 2 devices are moderate. They can also be high-risk devices. It's often said kind of, class 1 low, class 2 moderate, class 3 high. But you can have a high-risk device that's class 2 if you're able to mitigate the risks sufficiently so that it can be safely regulated as a class 2 device. Those require a 510K. In class 3 devices, high risk cannot be mitigated. Those require submission of a PMA. So each class has its own review paradigm, and this is just covering some of the terminology that's associated with the different classes. As I mentioned, for class 1 devices, I'm not going to go through every cell of the table, but it's important to remember these devices have not been reviewed, they are not cleared, they are not approved, they are legally marketed, but it would be adulteration and misbranding to claim that they had been cleared or approved. Exempt devices have not been cleared or approved. Class 2 devices, 510Ks are cleared. De novo petitions, which I won't get into, but basically it's another way of looking at a device that should be class 2. De novo petitions are granted. The review paradigm for a 510K is substantial equivalence. They have to meet general and special controls. We look at analytical and clinical data for both 510Ks and class 3 devices. And PMAs are approved. So when you're thinking about reclassification, taking devices from class 3 to class 2, one of the considerations is what difference does it make what the classification is? And there can be some pretty substantial differences in benefits to manufacturers. So the performance standard for a 510K is substantial equivalence, which means it's substantially equivalent to an already cleared device that's already been shown to be safe and effective. PMAs are standalone. They have to have the data to show they're safe and effective. Some 510Ks require clinical studies, some of which are extensive. Almost all PMAs require clinical studies. The review of the analytical data, it's also called preclinical data, precision, reproducibility, is the same. Software and instrumentation review is the same. We clear draft labeling for 510Ks versus final labeling for PMAs. There's no review of manufacturing in a 510K. That's quite a substantial part of a PMA, so the documentation that has to be submitted is a lot smaller. However, evidence of meeting the quality systems has to be in the manufacturer's internal documentation, whereas CNC sections are reviewed in a PMA. There are rarely inspections for 510Ks, sometimes maybe for de novo. PMAs, inspections are customary, although they can be waived if there's a lot of been inspections of that facility recently. Post-market inspections, adverse event reporting is burdensome. Those provisions are the same, whether it's a 510K or a PMA. What really matters in terms of classification is if you're making a change that may affect the safety and effectiveness of your device. In 510K, you submit a new 510K. In a PMA, you submit a supplement. But the review time for a 510K is 90 days. The review time for a PMA and many supplements is 180 days. So it's half the review time. The user fees for a 510K versus a new PMA and for a substantial supplement, the user fees are for a new PMA, I don't know what it is right now, but it used to be like $12,000. I mean, for a 510K, like $12,000. A PMA is $400,000. So the difference in cost to a manufacturer to submit and to maintain a 510K versus a PMA are substantial. It's a much shorter timeline for review. So there are a lot of benefits to a 510K versus a PMA. But when we reclassify a device from Class C to Class 2, as I said, a Class 2 device can be high risk. And that doesn't say anything about the impact of the condition that it's being tested for. Can we mitigate the risks of that device? It doesn't say anything about the actual condition. So my first challenge question is, does classification of a diagnostic test in Class 2 mean you consider the condition for which a test to be less serious than that for a Class 2 device? Yes, because Class 2 equals moderate risk. No, because Class 2 devices can be high risk or moderate risk. C, no, because device classification is based on the risk to a patient of a wrong result, and if and how that risk can be mitigated, not the seriousness of the condition. Anybody want to guess? Well, it's kind of a trick question because B and C are both correct, but C is more correct. And the reason I emphasize it is because I have heard people say, even within the agency as well as outside, if FDA reclassifies a device, for example, for HIV, it's because we don't think HIV is serious anymore. And that is not the case. But I've heard it said. So I just wanted to emphasize the point that does not mean that we don't consider HIV any less serious than it was before the device was reclassified. So when we looked, again, as I said, after 30 years, we decided, you know, we understand these devices really well, and we know how to mitigate the risks of a wrong result with these devices. So I think we should start thinking about if it's time to reclassify. This was within the agency. Outside, there were a number of stakeholders who thought it was well past time to consider reclassification. But in 2017, we had a couple of 510Ks, but most of the devices were Class 3 PMAs. And of course, we have BLAs for donor screening, biologics license applications. We put those aside. We don't even think about them. They're separate. But most of our devices were Class 3. So when we think about should we reclassify a device, again, we look at the benefits and the risks and the balance of the two. So the benefits, there's a shorter timeline, lower user fees, no annual reports or supplements. But the real reason that you want to reclassify is because we want an earlier entry to market. We want more devices on the market, more and better devices, ones that can maybe are more attuned to unmet, you know, the users with unmet needs. So we want more devices out there. And if reclassification can help accomplish that, that's a real benefit. But the risks that the new devices may not meet the performances of the old devices. And we don't review manufacturing. The reclassification process is lengthy and complex. Started in 2017 and finished in 2022. Of course, the shutdown in 2020 didn't help any. It also didn't help that we had a historic snowfall that put back our advisory committee meeting and then a fire. Alarm, an actual fire during the rescheduled meeting. But we brought the proposal to reclassify to our biologics product advisory committee. People are probably familiar with other advisory committees that FDA does, which is a panel of independent experts who give advice to FDA on our proposals. So we proposed to reclassify, and we had unanimous support for our proposal. Although we hadn't proposed reclassifying by the low test, the panel and other stakeholders also strongly recommended that we do those as well. So we decided that we would do that also. Now, as I said many times, special controls are special device type specific controls that must be followed because they are necessary to provide the reasonable assurance of safety and effectiveness of the device. So we wrote special control. One of the risks associated with the classification with a 510K where the paradigm is substantial equivalence versus the PMA is what we call colloquially predicate creep. So you don't have to show the independent performance of the device. You have to show how it compares. Is it substantially equivalent to another device? So one of the risks is that you could have a device that has a sensitivity of 99%. If that's so, and now it's a 510K. So device B comes in and it's using A as a predicate, and its sensitivity is 98.7%. Well, you know, is that substantially equivalent? It's pretty close. So then C comes in, and it's using B as a predicate. Now its sensitivity is 98.1%. That's substantially equivalent to 98.7. You can see that over the years, you can have a real degradation of performance where performance has really decreased compared to the original PMAs. And we wanted to make sure that wouldn't happen solely because the devices had been reclassified. So we put in the special controls, basement levels of sensitivity and specificity for the major clinical study for these devices. There's a difference between point of care and lab-based tests. Again, balancing slightly lower performance at point of care with vastly increased accessibility of point of care devices. But we put these special controls in there, these performance standards, and you can see they're 98% and 99% lower bound, or 95% confidence interval. So if somebody had a device and it has sensitivity of 96%, we're not going to clear it. It doesn't meet the special controls because it's not because the performance of these devices is so critical for making sure people are appropriately treated. And we can't have the performance degrade just because it's 510K and it gets more devices out there. We don't want more devices out there if they're bad devices. So we also reclassified our viral load devices. In these cases, we don't have the same performance standards. Lab-based viral loads have very exquisite sensitivity. Oh, sorry. Exquisite sensitivity. But we want to make sure that when you compared a predicate device to a new device, that the bias was very small and the performance was very similar across the measuring range. So the reclassifications were finalized in 2022. And here are the Federal Register notices and the regulations. And the special controls are in the regulations. So if you want to see what they are, you can look at the reg and that will tell you. We did reclassify. So in 2024, the status of HIV IVDs, we have most of our devices now are class 2 that require a 510K. We still have some PMAs. Those are self-tests and self-collection devices, again, POAs. We did reclassify viral load, lab-based and point-of-care tests, although we didn't, in the diagnostic reg, we separated out the two because we had different performance standards for the two. In the viral load, we didn't specify sensitivity. So we didn't specifically call out lab-based and point-of-care the same way we did in the diagnostic one. But in the viral load, our intention is to reclassify viral load point-of-care devices also. We've gotten feedback that there's been some confusion there because we want that explicit in the reg. Because some people think we didn't reclassify point-of-care viral loads. Our intention is that we did. However, we have not had any submitted yet. So our expectation is that they'll be able to meet the bias requirements and the special controls. If not, you may need to consider de novo. There may be, again, more of a balance of benefit risk than we were anticipating. If you are developing a viral load point of care and have questions about that, would you come talk to us about it so we can be thinking about it and see if this is going to work or how to make it work. There's a real need for point of care viral load tests, and we're really anxious to work with the community and the developers to try and make these happen. Self-testing devices are class 3 devices that require a PMA. There's only one self-testing device that FDA has approved, or
It was approved in 2012. And if people were around at that time, they may remember that the process for getting to approval of that device was laborious. It took years. There were multiple stages. And that's because at that time, there was a lot of concern about it was a good idea even to have an HIV self-test. And what would that look like if people got a positive result? But in the subsequent years, we found out it's a great idea to have an HIV self-test. And a lot of the concerns that we had have been alleviated by the experience of the community using these tests. So we agree there's an urgent need to have more self-tests. And while we haven't reclassified them, we have been working with manufacturers to streamline the process so the multi-stages and the multiple years is no longer necessary. We've been streamlining the process. We're considering, if you want to add a point of care claim to an already approved point of care device, that's a very straightforward way to go. We're working with them how to leverage existing outside the US data to support some of the requirements for demonstrating performance. Self-collection kits are IVDs and require authorization to be legally distributed. So self-collection means sometimes called home collection. The person gets a kit, they poke their finger or take a swab, put it in a card or a tube and mail it away to a lab. The lab sends you the results. It's a given that adequate and appropriate sample collection is necessary for appropriate device performance. But the lab, except for a few, is it hemolyzed or the dried blood's not big enough, has very little insight into if that sample was even collected correctly. So what we do with collection devices is we look at the labeling, we look at the instructions for use. Everybody's done a COVID test now and they know how the instructions, some of them are really clear, some of them are not so clear. And the manufacturers run studies to see if people can actually follow the instructions as they are presented. Oftentimes, when you already know how to do something, it's more challenging than you think to actually write it out. We had a case where in the instructions, the person had to open a lancet, and they had a drawing, they had one hand holding the lancet, opening it. When they ran the observed user study, they found people were following the instructions, using one hand to try to open the lancet, which doesn't work. So they had to change the instruction that showed two hands, one holding the lancet, one taking off the lancet. People were following the instructions were faulty. So we look at things like that to make sure that people can actually do the self-collection correctly. So we're improving access to testing by reclassification, shortening the timelines for review, much decreasing the cost. We've reclassified the viral loads, streamlining validation for self-tests, holding workshops, advisory committee, and public meetings to obtain community feedback. There's always the Breakthrough Devices program that prioritizes novel devices if somebody has a really good idea. So we're always interested in new devices to help protect and promote public health. In the 30 plus years since the first test was licensed, since we reclassified, we've had 3 new HIV diagnostics come in as 510Ks. They've all been exceptionally good devices. The performance is very high. We're very happy with those. We also have some concerns. Long-term use of PrEP, for example, seems to affect diagnostic devices. It may have some effects on sensitivity. We're not quite sure where that's going to go, but we're keeping an eye on that. So novel devices coming in to protect and promote public health. We're really keen to continue on this path. So we urge you to come and talk to us. There's a Q-sub process to come in, submit questions to the agency. Through this process, come in whatever questions you might have. We're from the government, we're here to help. And so thank you very much.
Thank you so much. I'd like to invite both speakers to come up for the discussion table. And I'd like to invite any of our audience members that might have burning questions to make their way to the podium. Time while you're thinking, maybe formulating your questions in the audience, we'll go to an online question.
All right, so the first question is for Orieji. Can you please give examples of disease associated with plasma source?
Oh, so in my talk, I talked about source plasma collections, like normal source plasma, disease associated, and disease specific. So the question is example of disease associated. Yeah, so I think a really close example would be plasma from donors who recovered from COVID infection, for example. So something like that would be considered that. But also there's plasma from donors who have recovered from other diseases that their antibodies might be of interest in making controls for IVD devices, for example.
Have another question from our online audience?
Yes, this question is for Julia. Julia, please, would you give us real examples of class 1, 2, and 3 HIV assay devices?
So there are no class 1 HIV devices. The class 1 device is one, I think some like cholesterol IVDs are class 1. I'd like to think of things like nitrile gloves, tongue depressors, crutches. Those are all class 1 devices. There are a few IVDs that are class 1, but not actually a whole lot of them. Class 2 are, as I mentioned, the reclassified devices. So those are the HIV diagnostic and supplemental serology and NAT devices, viral load. We also have disease-resistant mutation devices where when people are on treatment but appear to be failing, the treatment appears to be failing, you need to know what mutations they have because HIV is one of the most highly mutagenic viruses to look at if their therapy needs to be changed. And then class 3 are the self-tests and self-collection devices at the moment are class 3, but we are, considering, you want to consider, they weren't reclassified, but we want to consider if they should be de novo in order to bring them into class 2. But we haven't actually done that yet.
Any further questions from the audience just yet? We will leave your questions. So in that meantime, we'll go look at our online audience. Do we have another question from online?
Yes, we do. This is for Amici. The question is, does OBR also have oversight for blood collection devices such as blood bag systems?
Yes. So OBR oversees blood bag sets that are used to collect and process blood. The regulatory pathway for those vary. If they contain anticoagulants and storage solutions, those come in as NDAs where we look at the whole package. If they're just empty blood bags or they're part of an apheresis device, for example, those come in as 510Ks.
We'll take our next question from our online audience.
We have a nice question. This is for Julia. Can one test have more than one intended use?
Yes, they can. You can have a device that is both a diagnostic, for example, a nucleic acid test that tests for HIV RNA. It can have a claim both as a diagnostic and as a monitoring test. But both of those, just in one device, both of those intended uses, though, have to be validated appropriately. They are different populations. So it certainly can be done, and it can be a very efficient way to get a device with multiple uses on the market.
Thank you so much. And, do we have another question from our online audience?
Yes, we do. This is for Orieji. So is the CTD format used for blood BLA?
Oh, that's a good question. Currently, no. I believe right now it's optional, but typically, historically, we've provided the e-submitter process for submitting applications for BLAs or BLA supplements for blood and blood components.
We'll take our next question from our online audience.
Yes. This question is for Julia. Is there a user fee for pre-submission?
At the moment, there is no user fee, and there's also no limit to the number of meetings or presets you can have for a device, unlike CEDAR, which
I think it's very strict in limiting the types of test meetings you can request. In our IVD pre-subs, we entertain a lot of meetings, a lot of feedback. And there's no user fee, so they're free at the moment.
For the next question from our online audience.
Yes, this is for Orieji. The question is, after a BLA approval, is the FDA still conducting establishment inspections? If yes, at which frequency?
Yeah, that's a great question. So in my presentation, I outlined what we, OBR, do in terms of inspections. And our inspections focus on pre-license or pre-approval of the supplement. Once the BLA or application, once the BLA is approved, inspections do continue, and that's done by our Office of Regulatory Affairs, ORA. They conduct what they call surveillance inspections. Right now, they use a risk-based approach, I'll put it that way, to determine how frequently they should conduct the inspections. Historically, it's been about every 2 years, but that may vary. And of course, if there are issues that warrant an immediate inspection or an inspection closer to the time when they had their last inspection done, for example, we will work with ORA to conduct a for-cause inspection or other kind of inspection as needed.
And our next online question.
Yes, this is for Orieji. Does OBRR participate in industrial and international efforts to harmonize policies on the regulation of blood and blood components?
Yes, we do, and that's very important to the work that we do. We work with a lot of our customers and our other government agencies, both domestic and international, to develop blood policy or address issues related to blood. So I'll give some examples. We work very closely with our trade associations in the U.S., the AEB for blood and blood for transfusion, and also PPTA for source plasma. And, for an AABB, for example, actually we have FDA liaisons on many of their committees. For example, they put together what we call the Circle of Information, which is kind of the prescribing information for blood components. We have FDA participation in that, and we work together to put together the Circle of Information and review it so that it's acceptable for use for all blood establishments that wish to use them. We also serve, example, on the Donor History Question Task Force that is responsible for developing donor questions that are used to screen blood donors. So that has been a very useful relationship between FDA and our trade associations. We also participate in different, groups, WHO developing standards for some of our devices. We also participate in the European EDQM and their meetings. So we're very much aware of what other countries are doing in terms of policy, and we work together to try and harmonize as best as we can. So that's very important for us.
Well, thank you so much for fielding all of the questions from our online audience. And we thank you very much for your excellent talks. Both of you presented very interesting talks today. Thank you for taking the time to present with us here. And a round of applause for our speakers one more time, please. So now we're going on a break a little early. So everyone has some time to go stretch their legs, go for a walk a little bit, get something to drink. And then we'll meet back here promptly at 2:45 for the last session of the day. Thank you so much.
Good afternoon, everyone, and welcome to the last session of CBER Track. In this session, we will spend time with our experts from CBER's Office of Compliance and Biologics Quality and talk about Bioresearch Monitoring Program, or what is known as BIMO program in CBER. So BIMO program at FDA in general is a comprehensive program for on-site inspections, data audits, and regulatory assessments designed to monitor research conduct and reporting for FDA-regulated research. And so today we have Lieutenant Triet Tran, who will be talking about CBER's BIMO program. Dr. Tran is a regulatory officer in the Bioresearch Monitoring Branch in the Division of Inspections and Surveillance at the Office of Compliance and Biologics Quality in CBER. Prior to his work at FDA, Dr. Tran served as an inpatient pharmacist and nuclear pharmacist and radiation safety officer. Since joining FDA in 2021, Dr. Tran has served as a bioresearch and monitoring reviewer, collaborating with different product offices within CBER to review drug applications and with the Office of Regulatory Affairs to issue and review inspections. So Dr. Tran earned his Doctor of Pharmacy degree from right here from the University of Maryland School of Pharmacy in 2018. Dr. Tran, please.
Hi, everyone. My name is Lieutenant Triet Tran, and I'm a regulatory officer within the Bioresearch and Monitoring Branch within CBER. And today, I'm here to talk to you guys about the BIMO program that is administered within FDA, and more specifically within CBER, how it's administered within CBER. By the end of this presentation, you'll be able to understand the objectives and the goal of the FDA BIMO program, be able to recognize 3 common reasons for BIMO inspections, be able to describe the BIMO inspection process, understand the current trend and the frequent issues found in our BIMO inspections, and lastly, be able to identify effective strategies to ensure compliance with the FDA regulations. So the BIMO program, as mentioned earlier, is a comprehensive FDA-wide program of on-site inspections and data audits, and within recent year, also remote regulatory assessments that are designed to monitor all aspects of the conduct and reporting of FDA-regulated studies. The BIMO program in the different centers within the FDA and the different offices work together to develop policies and procedures to make sure that they're harmonized. And this is so that compliance is enforced the same way throughout the agency, and that we speak with one voice when we are talking to industry, and also including our international partners. So the objective of the BIMO program is threefold. The first, and arguably the most important, is to protect the right and welfare of subjects enrolled in these studies and FDA-regulated clinical trials. Second is to ensure the quality and the integrity of the data that is being submitted to FDA as part of the marketing applications. And lastly, we want to assess the conduct of these FDA regulations, assess the compliance of FDA regulations governing the conduct of the clinical trial. So here is a organization of the FDA, and the BIMO program is administered within the 6 products offices, the 6 product centers, and the Office of Regulatory Affairs, which are our boots on the ground. These are the FDA inspectors that are coming to the different sites to conduct the inspections and their assessment. Today's focus, of course, is going to be on CBERs, which is focused on biologics and vaccine products. Within CBER, there are 3 product offices that oversees the products that we regulate within CBER, the Office of Blood Research and Review, the Office of Vaccine Research and Review, which are responsible for the respective products, and a super office, the Office of Therapeutic Products, that oversees the remainder of the products under CBER's jurisdiction. In addition to those 3 product offices, there are 5 supporting office that works in collaboration with the product office to assist them in their review. And the BIMO program within CBER is administered within the Office of Compliance and Biologic Quality. This slide outlines the Code of Federal Regulations, or CFR, that governs FDA-regulated research, and such are the relevant one to the BIMO program. The CFR serves as a legally binding framework of regulatory enforcement by the FDA. So if we take any regulatory actions, if we cite any objectable finding, we will cite the Code of Federal Regulations in our work. But in addition to the CFRs, we also have 11 BIMO compliance program manuals. And these compliance manuals serve as a comprehensive set of guidance that has been developed by the FDA for our own personnel to its instructions on how to conduct our inspections, how to assess compliance during these inspections. So while these guidance are not legally binding like the CFRs, it is a great resource to understand how we conduct our inspections and what we are looking for in our inspections. So here are a list of the different BIMO inspections that issue within our program. The first 5 listed here, the sponsors, clinical investigators, sponsor investigators, contract research organizations, and institution review board inspections are geared towards good clinical practice. But in addition to that, we also do non-clinical focused inspection as well in our non-clinical and analytical laboratory inspections. So that was a different type of BIMO inspections, but why and when we issue these inspections really depends on the circumstance and the goal of the inspections. The first and the most common reason why we issue our BIMO inspections is for the marketing applications. So these marketing application-driven inspections are associated with our review of original or a supplemental submission. And with these inspections, we are focused on ensuring the data that is being submitted in support of these marketing applications are accurate and legitimate, and also comply with the regulatory standards. The second type of inspections are the for-cause or directed inspections, and these are typically initiated in response to a complaint that we have received. Complaints can come from consumers, can come from study subjects, the whistleblowers, or even within other departments within FDA. And lastly are the routine surveillance inspections. So these are the routine inspection program that BIMO within FDA issue every year. We work collaboratively with the other centers to identify programs and issue inspections as part of the annual surveillance. And in addition to the collaborative surveillance program, within CBER, we also issue our own surveillance inspection as well. So what is review and what we look for during the inspection really ties back to the objective of the BIMO program. So first, of course, is patient safety. We want to make sure that there are measures in place to protect the right, the safety, and the welfare of the participant in these clinical trial. And second, we want to look and assess the study conduct to make sure that the study is being conducted in accordance with the approved protocols. And then also, we want to make sure that the study is being conducted is adhering to the regulatory requirement. And lastly, and this is more so with the marketing applications-driven inspections, there is a data verification component. The data that the sponsor has submitted in their application is included in our inspection assignment memorandum, so that is compared with the source document at the site, just to make sure that the data are legitimate and are accurate. So that was the different type of inspections that we issue and why we issue it. And I want to take this opportunity to also go over the inspection process from the beginning to end. So in this slide, I've separated into 3 different levels. On the left is at the center level. There is the Office of Regulatory level, and then also activity at the inspected entities. First, at the center, we conduct our initial review. So whether that be a marketing applications, a complaint, or part of the surveillance program, we review and determine if inspection is warranted. And if so, we would then do a risk analysis to determine which site should be selected for inspections. After the site has been selected, we then draft an inspection assignment memorandums to include any background information for our Office of Regulatory Affairs inspectors, any questions and concerns that is not covered within the compliance program manual. And of course, for marketing applications, we include a data table component to it as well. After the memorandum has been put together, it is then issued and sent to the Office of Regulatory Affairs to conduct the inspections. Most of our inspections, especially with the marketing application ones, are pre-announced. So when the FDA inspector is ready to conduct the inspections, typically they'll reach out to firm to alert them that they will be coming to conduct the inspections, just to give a heads up and also an opportunity to pull any records that would be required during the inspections. After the inspection has been conducted, the FDA inspector will then issue an initial classifications based on the inspectional findings, as well as draft an establishment inspection report, or EIR, which is a narrative of the inspection process and any finding that they discovered during the inspections. And also at this time, if there were any objectable findings that were identified during the inspections, a Form FDA 483 is then issued to the inspected entity. If this form is issued, the inspected entity has an option to reply to it, and if they choose to do so, they will have 15 business days from the issuance of the Form FDA 483. Both their EIR, any exhibits that were collected during the inspections, as well when applicable, the Form FDA 483, and their response from the inspected entity is then submitted back to the center for a final review. Here at the center, we will review the narrative, all the exhibits that were collected during the inspections, and the response when applicable to make the final classification of the inspection. Any regulatory actions, any letters that are issued is then issued by the center to the inspected entity. And for marketing applications, the inspection result is then shared with the review team as well. So as I mentioned before, the FDA inspector will make a classification of the inspections, and then the center will make a final classification. So the inspection classifications are broken down to 3 categories based on the severity. The first one is no action indicated when no violations has been identified. The second one is voluntary action indicated when objectable conditions or practice were found, but the agency is not prepared to take any administrative or regulatory actions. And lastly, the more severe one is the official action indicated, or OAI. And this is when serious non-compliance has been identified from the inspections and regulatory and or administrative action is indicated. So so throughout this presentation, I emphasize a lot about the inspections are driven during the marketing applications, and a lot of what we do within the CBER Biomer branch is act as reviewer on these committees of reviewing the marketing applications. So as such, within CBER, we have a branch that's dedicated to administering the Biomer program, and our main function, of course, is reviewing the applications, issuing the inspection assignment, reviewing any complaints that come our way to determine if a directed inspection is warranted. We also serve to answer any questions regarding data integrity, good clinical practice or GLP, good laboratory practice or GLP. We collaborate with other centers and offices to administer the surveillance program inspections and also develop policies and procedures to make sure that they're harmonized. We also conduct internal and external education and outreach activities to stakeholders within FDA as well as attending conferences and giving presentations. So throughout this presentations, I've emphasized that a lot of our inspections are driven by reviewing the marketing applications. So I wanted to show the inspection process in relations to the life cycle of the marketing application as well. So above, you see that the marking above is the key milestone of marketing applications. In the middle is the progressions of the application through the life cycle. And at the bottom is the function that what we're doing within the CBER BIMO team. So even before submission, we participate in the pre-submission meetings to answer any questions that the sponsor may have regarding data submission or any BIMO related questions. And once a marketing application is submitted, we start our review to determine, one, if inspection is warranted, and two, if so, we would then do the risk analysis to do the site selection. We will put together the inspection assignment memo and then issue it. And in terms of the application timeline, we try to have this step earlier on in the application when possible, because based on the inspectional finding, it can determine what the next steps are. In some case, we could issue more inspections, or and if we do find any issues from the inspection that we did issue. Of course, so once the inspection is completed, we review and make the final classifications, and then we share that with the review team since it's a marketing application. In this case, we summarize the inspection finding in our final discipline review memo, and we do that before, of course, the action due date. So in this slide are the metrics of all the inspections, all the BIMO inspections that we've conducted in the fiscal year 2023. Something I really want you guys to take away, if anything, from this is publicly available, and every year we publish this on the FDA website. But as you can see, in the last year, we've issued over 1,000 BIMO inspections. And if you look at the top row and you see CBER, the most common type of BIMO inspection that was issued was the clinical investigator. And following that is the sponsor. And this really ties into what I've been saying, that because a lot of the inspections we issue are a result of the marketing applications, sometimes a sponsor will submit an application that has 10 up to 100 clinical investigator conducting in support of that marketing application. So I will be focusing on the metrics of the clinical investigator and the sponsors, but if you look at the metrics that are available online, it goes into details a lot more about the different inspection types that were issued by BIMO last year as well. So here are the metrics for the final classifications of the last 7 years for the clinical investigator. So as you can see, during the pandemic, of course, 2020, 2021, we saw a little dip in our numbers, but we're working back to what it was before. With our inspections, typically we see no action indicated, a handful of voluntary action indicated, but rarely do we see an official action indicated. But as you notice, it is trending up, so that is something that we are looking out for. And of course, the goal is for it to trend down, essentially go to zero, and have more NAI than anything else. For the metrics that were posted online, in addition to just the number of inspections that were issued and the final classifications, we also take into account and record the common violations that we see. So in this slide here is presenting on the list of violations that we see with our common clinical investigator inspections. The most common one would be failure to follow the investigational plan, inadequate study records, inadequate study subject protection, so issue with the informed consent process, whether that be the assent process or the informed consent missing required portion by the CFR, and then inadequate control and or control of their investigational product. And lastly, with issue with the safety reporting, particularly failure to report and or record an adverse event. So this is the metrics for the sponsors and the CRO. Similar to what we saw with the clinical investigator, during the COVID years, there was a little dip, but we're working, getting the number back up now. We see predominantly NAI as their inspection classifications. And with, unlike the clinical investigator, we see here is that the OAI is actually trending down, which is good. And the last 2 years, we actually have no official action indicated. So hopefully we see that trend moving forward, and even less VAI, and we see more inspections being conducted. Here are a list of the common violations that were found in the sponsors and the CRO inspections. Failure to ensure proper monitoring of the study and ensuring study is conducted in accordance with the study protocol or investigational plan. So this is similar to what we saw with the clinical investigator. The second one is failure to meet the abbreviated requirement for investigational device exemption, or the IDE. Failure to maintain and or retain adequate record in accordance with the CFR 31257. So that's accountability of the investigational product record, investigational statement, Form FDA 1572, and any financial disclosure. We also see issue that there are failure to submit investigation new drug applications, and lastly, failure to submit the current list of participating investigator to FDA at the 6-month mark. This timeline is for the IDE, and for the IND, it's actually shorter, but we don't see that as a common violation, which is good. So here are a list of compliance actions that the FDA can take as a result of the inspection finding. NIDPO, or the Notice of Initiations of Disqualifications Proceeding and Opportunity to Explain, it's a long one. And then NOOH, or the Notice of Opportunity for Hearings, are the steps that FDA would take if we are looking to disqualify a clinical investigator from conducting a study in the future. And we seek to take these actions when we believe that there has been repetitive and deliberative violations of the regulations and any intentional falsification of the data that is being submitted to the FDA. In addition to that, we can also refer to the Office of Criminal Investigation if we believe that there are potential for criminal charges. We can also issue warning letter, which are publicly posted online, and untitled letter, which are not posted. But these are these are advisory actions, so they're intended to give the opportunity of the inspected individual to still come back into compliance without disqualifying them. And the last two actions listed here are more so with the clinical reviewer and clinical review team, where depending on the inspections result from the site, we can choose to reject the data from that site. If we see that the issue is just an isolated into one site, we can request that the sponsor remove the data from that site and reassess, do their analysis again. However, if we see that the issue are occurring at multiple sites or even at the sponsor level, then we can also move to put the application on hold. So that, I know that sounds a lot scary, where a lot of regulatory actions, but there's a lot of ways that are to avoid getting to that step. I think when you guys look at the metrics, most of their inspections are not official action indicated, so most inspection doesn't get to the stage, but it can happen, and taking steps to stay in compliance is the best way to avoid being on the receiving end of these compliance actions. So we wanted to end on a good note and included some suggestions on what you can do to prevent non-compliance. So before starting the study, you would definitely want to understand your responsibilities and what you're signing up for. Any training that is required by the sponsor, definitely, we want to make sure you complete and fully understand it. And those are the bare minimum. So if there's any additional training or optional training, it's, we strongly suggest it so that you are aware of what you're signing up for. In the clinical trial, we see that a lot of times duties are being delegated to other people. We just want to make sure that this is documented, and that any duties that you're delegating to another person, make sure that they're qualified to perform those duties. When you're reviewing the protocol and you see that there's a lot of tests and a lot of assessments, a lot of steps in a process, developing a form or a checklist to make sure that all the tests and all the steps are being performed would be strongly recommended. Sometimes the sponsor will provide this, sometimes they don't, but you as a clinical investigator, you can develop it for your site as well. Definitely understand the protocol. The most common violation is protocol violation. So please make sure you understand the protocol. Anytime that there's new amendments that are coming through, understand what is being changed with each version of the amendment, and any new training that comes with the amendment, make sure you and your staffs are being trained as well. Here, the Code of Federal Regulations, again, is what we use to take our regulatory actions. So being familiar with the different codes is going to be important to avoiding those violations. Developing a plan to keep all your record organized is very important, especially a lot of these clinical trials span over the courses of years. So you want to make sure you keep everything organized, and especially when there are change in clinical investigator, change in study staff, you want to make sure you keep their record in one place and organized. And any times that there are new staff being onboarded, make sure they're trained and brought up to speed before they perform any tasks of the clinical trial. Know your limitations, know your team's limitations. Don't take on too many clinical trial if you know your team is already at max capacity. This will lead to a lot of errors for sure. And then also, in addition to the Code of Federal Regulations, we also have guidances that we make publicly available as well, like the compliance manual for the different BIMO inspections that we do. So those will be good ideas to be familiar with. During the study, now that the study has started, definitely keep track of the dates that you have to report to the IRB and the sponsors. Anytime that there's a protocol violations or any serious adverse event, you want to make sure you report that to the sponsor as required by the protocol and the regulations. Anytime that you're doing something that is not in the protocol, you want to make sure you communicate that with the sponsor, and you want to make that documented. Even if it's a planned deviation, having it documented can help you in the future. Back to delegating duties. Now that the study has started, you definitely also want to follow up to make sure that people are only doing the duties that they've been delegated to and nothing more. And then also, if you guys identify any small issue, try to fix it, because it's definitely going to get bigger. Again, checklists and dashboards to track procedures and date, and keep that updated as you determine new things. And when you conduct the study, you can also fine tune those as well. So after the study, again, you want to keep all your record organized. I think I've emphasized a lot where a lot of our inspections are marketing applications. So a lot of times the study has been finished, the sponsor has done their analysis and they're submitting the application. So this could be years later. So, but that doesn't mean we don't issue our inspections. So we could come after you complete your trial to conduct our inspections. And this is when the ORA reaching out and alerting you guys a heads up that they're coming. Having an action plan on being able to pull those records back before they come, or knowing where to get it is going to be important. So please keep your records safe, secure, and of course, away from harm. And before I end, I have two challenge questions for you guys. So the first one, if a Form FDA 483 is issued to the site at the conclusion of the inspection, the site can submit a written response within 60 business days, 30 business days, 15 business days, or 10 business days. Yes, C. So if a Form FDA 483 is issued, the inspected entity has an option to respond, so you don't have to. But if they do, they have to do it within 15 business days of the issuance. And the second question is, which of the following is not a compliance action we can take from a BIMO inspection? Issuing a warning letter, referring to the Office of Criminal Investigation, rejecting the data from that site, or granting an inspection waiver. So the answer is D. So we don't grant inspection waiver from the BIMO inspections. So in summary, the FDA BIMO program aims to protect participants' rights, safety, and welfare, as well as to verify the accuracy of the data that is being submitted in support of the marketing applications, and to assess the compliance with FDA regulations. BIMO inspections can occur as a result of marketing applications, complaint, referral, or part of our routine surveillance inspection program that we do every year. It's a collaborative effort to select the sites, conduct the inspections, as well as to implement any regulatory actions. And BIMO inspections and the inspection observation metrics are publicly available and posted on the FDA website every year. And lastly, focus on building quality into the clinical trial process through proactive compliance is going to be the best way to avoid non-compliance and any violations with the regulations. I've included here our contact informations. The top one is just the general CBER contact information, but if you have any questions for the BIMO team, our email is listed below. And then I've also included resources that I used to put together this presentation, as well as other BIMO-related resources that are useful. That's all my presentation.
Thank you so much, Triet. Thank you for presenting, and thank you for including a lot of actionable information in your presentation. Don't go too far because we still have Q&A at the end of the session. And our next speaker in the session is Seth Schulte. He is a biologist in the Division of Biological Standards and Quality Control in the Office of Compliance and Biologics Quality in CBER. Prior to his work at the FDA, Seth worked in various industry settings in quality control, analytical development, and small-scale production. Since joining FDA in 2021, Seth has been involved in regulatory review of microbiological methods for multiple FDA offices. And in his today's presentation, he will focus on current regulations and requirements for validation of rapid microbial methods for biological products.
please join me in welcoming Seth here? Thank you.
Hello, good afternoon, everybody. Very excited to be here. This is my first time presenting at this conference, so I hope everyone is really interested in this topic. So yes, my name is Seth Schulte. I am a biologist in the Office of Compliance and Biologics Quality in the Division of Biological Standards and Quality Control. So today my presentation is going over CBER's perspective on the evaluation and implementation of rapid microbial methods. First, I'll just go over some learning objectives from this presentation. So today, I hope to help you understand the regulations that describe rapid microbiological methods, or RMMs. Learn about implementing RMMs for your specific product. Understand the guidances that surround rapid microbiological methods. And I also have some case studies at the end to hear some common deficiencies from implementing rapid microbiological methods. So before getting into this, I do want to take a quick step back and talk about the Division of Biological Standards and Quality Control. I won't read the top part, but it is summarized in the graphic below. So of this, I want to talk about the two main missions that our division has, one of which is obtaining and preparing reference material. This is filled and lyophilized at the FDA as needed, depending on the nature of the product. We test potency and calibrate to assign standard values, and we also distribute these reference reagents to other manufacturers to control their processes. Additionally, and the foundation for today's presentation, is analytical method review. So in addition to that, we do in-support testing at the FDA for regulated biologics. We develop lot release testing plans, and we also perform lot release protocol review and testing. So first, before getting into rapid microbiological methods, I wanted to quickly go over compendial method suitability. This on its own is really important to understand when testing your products, but it's also equally important when we talk about implementing rapid microbiological methods. So compendial tests are standardized methods that are used for testing a variety of different samples. These could be pharmaceutical raw materials. These could be manufacturing and process samples. These could be drug substance or active ingredients. And these could also be your finished product. When demonstrating suitability for compendial methods, laboratory records are a regulatory requirement. So if the method that you're using is employed in a recognized standard reference, the method must meet standards of accuracy and reliability. And this does include a description of the sample that you're testing. And ultimately, the method that you're using must be verified for suitability under the actual conditions of use, so in the presence of your product. There are several components of method suitability. Here I just have three. So performance of your reference standard, which includes your R value, your slope, or your intercept. This also includes the performance of your controls, as well as the precision of your replicates. Some examples for this would be if you're performing compendial sterility testing, so direct inoculation or membrane filtration, recovering not more than 100 CFUs. And for endotoxin, that would be evaluating your positive product recovery and the R value of your standard curve. And like I said, method suitability is a really important aspect of rapid microbiological methods. So rapid microbiological methods, also known as alternate microbiological methods, are technologies that allow for the end user to receive test results faster than your traditional compendial methods, which take 14 days. There are many different methods out there that use a variety of different platforms, and these can be used for testing many diverse types of products. Some methods do tend to be more straightforward than others. However, most alternate methods do often require proprietary technology or platforms. Some common examples of rapid microbiological methods for sterility would be respiration-based assays or ATP bioluminescence. And for mycoplasma, a very common method is the nucleic acid amplification technique. In order to implement rapid microbiological methods, there is additional work that has to be performed in addition to method suitability. And the big one is method validation. So method validation of an alternate or a rapid method is a regulatory requirement. I'll go over 2 CFR references, one of which is testing and release for distribution. So this just states that the accuracy, the sensitivity, the specificity, and the reproducibility shall be established and documented. And this can be done in accordance with CFR 211.194. Additionally, and this one we will visit a few times in this presentation, is equivalent methods and processes. So this states that modification of your test method will provide assurances of the safety, purity, potency, and effectiveness of the product. And this insurance for your alternate method has to be equal to or greater than the method that's or process that's in the general standards. So that's comparing your alternate method to your compendium methods. So this is a lot to evaluate. So on the next slide, I have a very high-level overview of CBER's expectations for rapid methods. And I will go through all of these through the course of the presentation. But the first bullet point, and I would say one that is very helpful, is going to be your compendial references. So USP 1223 and European Pharmacopoeia 516. Both of these are really useful chapters when you're looking at validating alternative microbiological methods. And I would highly encourage anybody that is exploring implementing an RMM to read both of these chapters. There's a lot of really great information there. The next one, as we discussed on the previous slide, is your comparability study. So this is a regulatory requirement as part of method validation. The third one, which kind of goes along with the next two, is that the end user must validate their alternate method in the presence of product to really provide assurance of that test method, especially when comparing it to your compendial methods. It's also the responsibility of the end user to understand the limitations of the potential alternative microbiological methods that they're pursuing and ensuring that they are appropriate for their specific product. We also recommend that the end user test this using worst case scenario conditions to really understand those limitations of the alternate method. Additionally, it's the responsibility of the end user and the expectation of CBER that microorganisms are used on the panel that are relevant to the product and the manufacturing environment. I will go into that more detail in a little bit. And also, if the end user has any questions before implementing these, rapid methods can be very time consuming and use a lot of resources. So we do highly encourage reaching out to a CBER representative, whether that be a Type C, a pre-BLA, or an IND meeting to request feedback on their proposed process. So first I'll start with USP 1223 and European Pharmacopoeia 516. I will get into more detail in these chapters, but first I kind of want to just go over the basic parameters of method validation that CBER looks at for biologic products. So first I'll talk about limit of detection and specificity. So the limit of detection is the lowest number of microorganisms that can be detected in a defined sample volume. CBER does recommend using a serial dilution approach because the LOD of your rapid alternate method is really important for subsequent validation steps. And it's also important to note that the detection limit of your alternate method should not be more than that of your conventional. Specificity is the ability of the test to detect a wide range of microorganisms in a sample. And like I said previously, these microorganisms should be carefully selected by the end user. So these are going to be microorganisms in addition to your compendial indicator microorganisms that pose a risk to the patient or the product, those microorganisms that are commonly found in the manufacturing environment through environmental monitoring, or those microorganisms that have been associated with previous product failure. And this panel should also be sure to include slow-growing microorganisms. The emphasis for specificity is generally the detection of not more than 100 colony-forming units, but CBER does recommend evaluating specificity of your alternate method at the limit of detection. Additionally, ruggedness and robustness. So ruggedness, also called intermediate precision, is the reproducibility under a variety of normal test conditions. And this is assessing the same sample. So this can be done, the end user can determine the most appropriate path forward for this. But this may be using different analysts, different instruments, different lots of reagents, or performing the test on different days. Robustness is the ability of the method to remain unaffected by small but deliberate variations in the method's parameters. And this is really helpful because it provides a really good indication of the method's reliability during normal usage. And this can be done by modifying the sample preparation or the incubation conditions, for example. And I would just like to point out that ruggedness and robustness are both parameters that can be assessed during the study for LOD and specificity, which will make things a little easier on the end user. The last parameter I'd like to discuss is equivalence. I will actually spend quite a few slides talking about this. So equivalence in general is the level of agreement between your compendial standardized test method and your rapid alternative test method. This can be evaluated using a variety of different test parameters that I have here on the slide. Initially, equivalence is demonstrated using standardized microbiology cultures. So this really assesses the equivalence of the method's platform itself, and then later it's separately performed using the actual test sample, which is really going to help you understand the limitations of your test method. And to evaluate equivalence, the end user should identify test samples throughout the manufacturing process that would be expected to contain microorganisms, or those test samples that are used for release throughout the manufacturing process to really demonstrate that the test method will provide equal or greater assurance than the compendial standardized test method. Like I've said before, during the test for equivalency, this is not only performed in the presence of product, but CBER expects that this is also performed with an inoculum at the level of detection that has been determined for your rapid alternate method. Your alternate and compendial methods should also be run in parallel. The end user can determine the most appropriate strategy, whether that be a specified period of time or a certain number of product batches or test samples. And so while we're talking about equivalence, I would like to talk about equivalency or comparability testing in the context of USP 1223. So USP 1223 has a really detailed section about equivalence testing. It essentially infers the equivalence of the outcome between your two test methods that you're comparing. And like I've said before, equivalence testing should be done at a low level or your limit of detection for microorganism. With this bullet point, I really just wanted to cover that at least one product type should be assessed during equivalency testing. And I would like to point out that if a manufacturer has a rapid method that is validated and approved for one of their products, a repeat equivalency study can be avoided if they have a product with a similar matrix that they plan to use the same rapid method for testing. And also when we're looking at USP 1223 for the demonstration of equivalency, it does make reference to USP 1225, which is the validation of compendial procedures for validating alternate methods. And the one thing I want to point out is the description they provide of LOD in this chapter, and that they do say that subsequent validation testing should be performed at or near the limit of detection for your alternate method. The chapter does provide 4 options to demonstrate equivalency. Today, I'll be focusing on just performance and results. So essentially, both of these are looking to evaluate that your alternate method provides assurance that is equal to or greater than that in the standardized chapters. For performance, this is done using validation criteria. And if you're looking at results equivalency, that is going to be comparing numerical results. So like I said, in either case, CBER's expectation is that the alternate method provides assurance that is equal to or greater than your general standards. And I would just like to point out that regardless of the option to demonstrate equivalency, CBER does require that your equivalency studies be performed in the presence of product. So in addition to USP 1223, European Pharmacopoeia 516 is another really valuable resource when you're looking at understanding or implementing rapid methods. So they describe a variety of different methods and different platforms in great detail. And one section in this chapter is risk-benefit analysis. So like I said previously in my overview slide, it's really important that the end user examine the different alternate method platforms that they have available to them to understand which is best suited for their specific product and the limitations of that test method. And this chapter also provides a lot of really great detail about levels of validation. So there are 2 main levels. There's primary validation and validation for intended use. So these are done by different users and for different purposes. So primary validation is going to be a validation that is done by the equipment supplier or the method supplier or whoever has that proprietary technology. This testing is not done in the presence of product, which is one important thing to note. But this does cover validation criteria such as LOD, specificity, robustness, precision, and if applicable, prerequisite treatment of the samples that they may be using. We also have validation for intended use. So this is going to be the responsibility of the end user. And this is, I would say, a very important validation. So this also covers several parameters of method validation. But this is done in the presence of product. And it's the responsibility of the end user to understand the design qualification. So making sure that the equipment that is necessary for their alternate method is suitable for the performance for what they plan to use it for. And this also requires the end user to perform an IQ, OQ, PQ. And I would like to talk about the performance qualification. So as we were discussing with primary validation and validation for intended use, validation data that is provided by the supplier has been accepted by CBER for primary validation, but because validation for intended use is done by the end user, this is done in the presence of product. And so this is going to cover all of your validation parameters that CBER is going to look at, which include suitability testing, LOD, specificity, ruggedness, robustness, and equivalence testing. And again, I would just like to point out the importance of limit of detection, and that equivalence testing per European Pharmacopoeia 516 states that the method should be compared at a low level of inoculum, such as your LOD. And while we're talking about method validation for a variety of different products, there unfortunately isn't a uniform approach for all product types. And especially recently with the large number of cell and gene therapy products that we're seeing coming to market, the path to validation isn't as straightforward. So I do have a couple slides talking about risk-based approaches to validation. So this first slide just has some really useful references for those types of products. So USP 1071 describes short shelf life products, and European Pharmacopoeia 2627 is for those cell-based preparations. There's also a CFR for negative to date early release of product. So for any products that fall into these categories, I would highly recommend reading these documents. But with these products, they tend to be either non-cryopreserved preparations that need to be given to the patient before a traditional test could be completed. And with patient-specific products, these cell-based preparations, they tend to be really limited manufacturing quantities, which ends up resulting in a limited sample size that is available for your sterility test. So while there is no one-size-fits-all approach to these, I would just like to point out that the focus tends to be on method suitability for these products, but validation is still required for these products regardless of the alternate method route that is taken by the end user. But everything is examined on a case-by-case basis. And this would definitely be a situation where it's important to reach out to the FDA for guidance. And because there is no universal approach to this, I do have some case studies, and this kind of leads me into my first one. So with limited sample sizes for these products, compendial documents, both European Pharmacopoeia 2627 as well as USP 1071, they state that 1% of batches that are 10 to 1,000 milliliters needs to be tested among all media bottles. The issue there is going to be the big decrease in sensitivity of the test method. So CBER does expect that in situations like these, the sample to media ratio remain appropriate. So generally decreasing the amount of test media to match the amount of sample that's being inoculated. In addition to that, this may also require that additional in-process tests on samples collected during the manufacturing process be needed to support the final release test due to the limited sample size. The next common issue that I wanted to talk about is incubation conditions. So as most people that do sterility testing probably know, compendial testing is performed using 2 media types. The importance here is that FTM is used for both aerobic and anaerobic incubation, while TSB is used for aerobic incubation. But what we're seeing with rapid microbial technologies is incubation for aerobes at 20 to 25 and anaerobic incubation at 30 to 35. But there tends to be a lack of aerobic incubation conditions at 30 to 35. And slow growers don't necessarily do well at lower temperatures of 20 to 25. So generally for rapid microbial methods, especially those respiration-based methods, CBER does expect 3 incubation conditions to really cover the growth requirements of a variety of microorganisms. So that would be aerobic incubation at 20 to 25 and 30 to 35, and also anaerobic incubation at 30 to 35. Additionally, and I have spent a couple slides going over this, is a lack of clarification between primary validation and validation for intended use. So like I said earlier, primary validation is performed by the supplier of the method. These are often submitted to CBER as a master file that we are able to reference. But again, these are performed without the test product. So data that's published in literature can be used for primary validation because that is assessing the equipment or the platform itself. Whereas validation for intended use is performed by the end user in the presence of product. And this does evaluate all of the traditional validation parameters that we would consider. So LOD, specificity, ruggedness, robustness, and equivalency. And data published in literature or from the method or the method supplier cannot be used for validation for intended use. This is really important for CBER to be able to understand that the test method provides assurance and that we understand the limitations of the test method. Lastly, this case study is over environmental isolates. So compendial indicator microorganisms are obviously going to be part of a validation, but they may not always represent real-world scenarios. So it's important that the end user, whether they're performing the test or whether they're using a contract laboratory to perform the test, that they evaluate the environmental isolates that they are finding, whether that be through environmental monitoring or sterility failures, and especially those slow growers, because with these rapid microbial methods, we have incubation times that are half, sometimes even less than that of compendial incubations. So we really want to make sure that it's able to identify a variety of microorganisms, especially those that you are likely to encounter during testing. So with that, I do have 2 challenge questions that hopefully I have covered. So the first one, what is the difference between primary validation and validation for intended use? A, primary validation is done in the presence of product. Validation for intended use is not. B is the exact opposite of that. Validation for intended use is done in the presence of product. Primary validation is not. C, there is no difference. Both are done in the presence of product. Or D, there is no difference. Neither are done in the presence of product. Yes, B. So validation for intended use, this is done by the end user in the presence of product. Primary validation, again, is done by the method supplier. My next question, which regulation requires equivalence testing between current standards and an alternate or rapid microbial method? USP 1223, European Pharmacopoeia 516, CFR 610.9A, or USP 1071? So it is CFR. USP 1223 and European Pharmacopoeia 516 provide a lot of really great detail when it comes to equivalence testing, but it is the CFR regulatory requirement that for any alternate method, equivalency testing is performed. So here I have some resources. Like I said, if you have not read USP 1223 or European Pharmacopoeia 516, I do highly recommend reading those. They're really valuable resources. And for cell-based products that either have a short shelf life or are patient-specific, USP 1071 and European Pharmacopoeia 2627 also have a lot of really valuable information. So in summary, rapid microbial methods are really valuable for the end user because it allows them to get test results much faster than traditional compendial methods. The method suitability is really important because it demonstrates that the method is suitable under the actual conditions of use, so in the presence of product. Method validation for alternate or rapid microbial methods is a regulatory requirement, which does require the assessment of multiple validation parameters. And once again, USP 1223 and European Pharmacopoeia 516, highly recommend reading them. And lastly, for my closing thought, if you are interested in implementing a rapid microbial method, please reach out to CBER with questions. We are more than happy to provide feedback and to provide guidance to make sure that you are on the right track.
Thank you very much, Seth. Please don't go away. I would like to invite both of our speakers to come here to the stage. And we are now going to proceed to the Q&A part of the session. Any questions from our in-person audience? If you have any questions, please come up to the microphone. Okay, we have a question.
Yeah.
Please go ahead.
Thank you both of you for the presentations, very insightful. The question is for Dr. Tran. I think will be regarding the data verification component that you mentioned in your presentation. What concerns specifically about data integrity and accuracy of clinical data do BioResearch address during an inspection, and how those concerns are addressed?
Yes, so thank you for the questions. So in terms of our inspections, a lot of what we're doing is we're comparing it with the source data that is being recorded at the site just to make sure that it matches up. But in terms of during the analysis and we see any outliers and stuff like that, that is what triggers us to select the site to be inspected. But as far as when we're at the site, we're just looking at the data that is recorded and making sure everything matches up and lines up.
Historical tracking and all.
Yes, like audit trail and stuff like that, who made the edits and, stuff like that.
Okay, thank you. Yeah. And just a quick comment for Dr. Schulte. Thank you for sharing the CBER expectations on your presentation as well.
Thank you. Do we have any questions from our online audience? Yes, all right.
Yes, hi, this goes to Seth. What are your manufacturing isolates or past sterility failure microbes suspected for stability and or validation study?
Can you repeat the question? Sorry.
Sure. Why are manufacturer isolates or past sterility failure microbes inspected for suitability and or validation study?
Oh, okay. Yeah, so as I was kind of referencing, compendial microorganisms are really important to use in those studies, but they may not necessarily represent the real world scenarios that manufacturers are running into. So by including these isolates that are most likely to contaminate their product or those that they're finding that are slow growing, it really provides additional assurance of their alternate test method that it will be able to detect those in real world practice.
Thank you. Any other questions from the in-person attendees? All right, so we'll move on to the online questions again.
This next question is for Tran. Tran, is there a systematic BIMO inspection with each new marketing application, or is it done more on an ad hoc basis?
I'm sorry, can you repeat the first part again?
So is there a systematic BIMO inspection with each new marketing process or marketing application?
Or is it done on an ad hoc basis?
So in terms of how we conduct the inspections, that's outlined in the compliance manuals. So we follow that, but in terms of the process is pretty similar regardless of the different applications. Yeah. Depending on the protocol, we might be looking at different things, but other than that, we follow the compliance manual.
All right. Any other questions from our online audience? Yes, okay.
This is for Seth. Can we contact the agency prior to starting any method validation studies for any feedback?
Yes, and I definitely encourage that, especially seeing how validation studies for alternate methods can be very time consuming. So please reach out, whether that be a Type C, a pre-BLA meeting, an IND meeting. CBER is more than happy to provide feedback to make sure that your approach is on the right track.
Thank you. Do we have more questions? Yes, okay.
Yep. This next question is for Trent. Trent, CBER has issued a guidance document regarding a standardized format for electronic submissions for NDA and BLA content for planning of BIMO inspection. Does CBER have plans to issue a similar type of guidance document, or is there any additional information that needs to be included in any original BLA to assist in CBER BIMO inspection plan?
So yes, we are working to develop the guidance similar to what CBER has published. So it's going to be harmonized, so it'll be very similar to what CBER has right now. But we're in the process, the final process of getting it approved and then getting comments on it before we publish it.
Thank you. I see that we have more questions from the online audience. All right, please go ahead.
All right, this question is for Seth. Can published literature be used in lieu of validation for intended use?
So no. So that type of data is appropriate for primary validation, but when the end user is validating for intended use, that needs to be generated from them using their specific test product.
Thank you. Are there more? Okay, please.
Yes, there's. This next one is for Tran. From a compliance perspective, what is the best recommendation or approach? A planned deviation or temporary change control?
So in terms of compliance, it's never ideal to do anything that's not in the protocol, of course, but in real life circumstances, moments like that happens, and we just want to approach it and make sure that everything is documented before it happens. So when we review and we come to the sites and we do the inspections, we want to be able to understand what happened and that the reason why this planned deviation occurs happening. I think just having it documented, well documented, is the best way to avoid any issue that comes up. But having it documented before it happens is much better than, of course, catching it afterwards.
Thank you. Are there any more questions from our in-person audience here? No, not at this time. And online audience? One more? Okay.
Yes. This question is for Seth. During method validation testing of a product, does the FDA have a standard allowable amount of variance?
I'm not sure that I understand the question.
Can I ask it one more time? During the method validation testing of a product, does the FDA have a standard allowable amount of variance?
Is there an allowable amount of variance during the method validation of biological products? That's a question, right?
Yeah, so I'm not entirely sure I understand what they're asking, but I would just say that when you are performing validation, you're really just wanting to make sure that in the presence of your product, the alternate method is going to be performing the same or better than what is outlined in the compendia. So I would say that we're not necessarily looking at the variation, we're just looking at the performance of the alternate method compared to the compendial method to ensure that it is performing either the same or better based on the validation parameters that I outlined in my presentation.
Thank you. This is helpful. Thank you. No more questions from online? Oh, other? Okay, please go ahead.
This one is for Tran. How much notice is a sponsor given before inspections of their facility or site?
How much notice does a sponsor give?
Yes, is a sponsor given by the FDA before inspection of the facility or site?
So when we pre-announce our inspections, we communicate that with the inspected entity. We don't communicate that with the sponsor. So whatever arrangement that the sponsors have with the clinical investigator, that's between them. But we only announce the site that we will be inspecting. So we would announce to the clinical investigator, but not the sponsor, unless it's a sponsor inspection.
No more questions? Okay, we've exhausted all of the questions from online audience. And any questions from the in-person audience? Not at this time. Well, thank you very much, Triet and Seth. The topic of compliance with biologics quality and safety is always a topic of interest for our conference attendees. And thank you so much for highlighting several very relevant areas that can be obviously used by our biological product manufacturers. So thank you. All right, well, let's give a round of applause to our presenters. And thank you. This will conclude our last session of the day. And in fact, we are at the end of the second day of the CBER track and at the conclusion of the annual Regulatory Education for Industry Conference 2024. So since we're conducting this conference as a hybrid event this year, each center concludes and closes their own track, not in the previous years when the conclusion of the conference was done for all of the tracks at the same time. So at this time, I would like to thank all of the CBER speakers for sharing their expertise and experience and perspectives, as well as the FDA Planning Committee and the Small Business and Industry Assistance Program that every year helps us organize this event and deliver updated educational lectures to help product development and to facilitate future regulatory submissions. And of course, much appreciation should go to the conference support team that every year does all of the preparations and behind the scenes work to help us to run the conference smoothly and without major problems. So most importantly, many, many thanks to all of you who attend the conference. Thank you for your engagement, for your interest, for your attendance, and for your attention. And thank you for sending us questions. And this year, as every year, we ask people not to forget to please complete your final evaluations and provide your feedback. We greatly appreciate all of the comments and suggestions that you may have for the future topics of this conference. And a few reminders, of course, if you have not had the chance to listen to some of the presentations that you wanted to listen to, particularly if they were from other tracks, the conference presentations are all recorded and they will be posted about 10 business days after the event. Please do not forget to download the certificate of attendance and complete the forms, and make sure that you do it on time, because you have to complete the evaluation forms and get the certificate of attendance within 14 days since the last day of the conference. We hope that the contents of this annual event will continue meeting your needs for the regulatory educational experience. Although we keep track of the post-event viewing, it's not really the subject of any time restrictions, so you may view the presentations at any time and at your convenience. And given that the REdI conference is the annual event, we hope to see you and perhaps other participants at our annual event next year. And so once again, I would like to thank everybody for your participation and for attendance, and wish you all a great year ahead. So thank you.